So I'm currently working in Saclay, which is a small town near Paris, where I'm working closely with Alexandre Camfort, who created actually MNE Python, I don't know, 8 years, 10 years, something like that ago. And yeah, it's actually my full-time job to work on MNE Python and related tools, and to at the same time stay in close contact and do collaborations with scientists who actually use the software. And additionally, I have used it myself in my research in previous years, so I still know where some issues, difficulties, quirks of MNE are. And I guess I still remember some of the the difficulties one typically experiences when one is a beginner. So with that said, the idea of this workshop is to give you it cannot be a thorough introduction into MNE, because it's like, it has so many features and you can do so many things with it. So it's going to be a brief touch upon the most important topics you will need to get started with MNE Python when you want to analyze your MEG and or EEG data, both on the sensor level, but then also going maybe taking it a bit further to the source level, source estimation. Maybe, can I share my screen? Yeah, I can. Now I just need to find the correct desktop. Okay, so I will assume that you signed up for this workshop, so I will already assume that you know what MNE is, but let me still try to briefly summarize it. MNE is a software which was initially designed for MEG and EEG processing, analysis, and visualization. And in recent years, it has also gained the possibility to allow you to analyze intracranial recordings. This is still, in a relatively early stage, but some people already use that in their research. The website, the address has recently changed. So it's now mne.tools. And this is actually, whenever I work with MNE, I have a browser tab open with this website, because I will constantly check the documentation. So there's tutorials on pretty much anything. There is a glossary, because sometimes you don't know what the heck are they talking about. And there is, of course, also instructions on how to install it, but also change logs, what's new. So there's about two major releases of MNE a year. So every 6 months, approximately, there's going to be a big new release, which fixes lots of bugs, adds new features, and sometimes also breaks backward compatibility. So whenever a new release has been published, you may want to check out the changelog, and you can see it's extensive. Like it's really, which is good and bad at the same time. But you may want to change. You may want to check out the changelog to see if maybe a bug in a function that you're regularly using has been fixed, or if the API, this is the programming interface, that is which options does a certain function offer, or maybe a function name has been changed. This is sometimes adjusted, and you may need to adjust your code accordingly to keep working with the latest versions if you upgrade. So it's always a good idea to check out the changelog before installing the new version of MNE. MNE is developed by lots of, here you have a brief overview, right? This is just for the last release. Lots of people all across the globe. It's a free and open source software, and the development takes place on GitHub, actually. The GitHub repository you can find under github.com/mne-tools/MNE Python. No, there's a typo. MNE Python. There we go. If you think you have run into a bug or an issue with MNE, there are certain ways to get help. One is you go to this GitHub repository and create an issue. And you can say, hey, something is not working. Here, for example, then it will get tagged as a bug, and hopefully the developers will fix it afterwards. There's also a mailing list. Also, I do not want to promote it now because we are soon going to switch, hopefully, we're soon going to switch to a modern bulletin board or a forum, which will be more visually pleasant, and it will offer many new features. Yeah, nobody really wants to use a mailing list anymore once we have used this board, which will happen within the next couple of weeks. Okay, with that said, I think we should get started. Before we get started, I want to ensure that everybody can actually follow along. So I'm just glancing over to the Slido poll where we ask, have you already installed MNE Python on your machine? And there's actually a few people who say no, and I need help. Okay, so I just clicked on the link that Johan shared on Zoom, which leads to this GitHub repository. Okay, this is where I share notebooks and also README. The first thing you will need is, well, that's almost trivial. You need a Python installation. And we recommend that you use the Anaconda Python distribution. Now there's different versions available depending on which operating system you're using. For example, I'm using a Mac. So I'm going to follow this link. And I'm going to click here. It says here, download the graphical macOS installer. Just going to click there. It should actually download now. Here, there's the download link. It's the download link, and I'm clicking on 64-bit graphical installer. So that's what we want. You always want the graphical installer. It makes things easier. I just download that thing. Should be the same on Windows. It's a bit more complicated on Linux, but it's explained on the Anaconda website. Fortunately, I have a rather fast connection here. It's just my computer, which is a bit slow. So as you can see, it's already being downloaded, but the computer was still processing the file. So I'm gonna open this installation package. I'm gonna say, yeah, please go ahead. And I'm just gonna follow through, and I'm gonna leave everything with the defaults. So here, for whatever reason, it says I cannot install it into this location. However, if I click, so that seems to be a bug in the installer. So if I click then down here on install on a specific disk, and then click again on install for me only, it suddenly works. And I would click continue, etc. I'm not going to do that now because I already installed Anaconda before we started the workshop. But you would just follow through, clicking continue, etc. And then it will take a few minutes because it's actually quite a big number of packages that's going to get installed. And after that, on the Mac, you open your Finder. Let me just close that so we can start from scratch, essentially. Oops, messages. You just open your Finder, go to Applications, and open the Anaconda Navigator. I'm sorry? Was there a question?
No, sorry, it wasn't. Sorry.
Okay, just gonna double click here. Gonna take a bit because my computer is rather slow. And I can see this thing starts up. So this means that the Anaconda installation essentially worked. We downloaded the installer. I just view it in my file browser. And well, I'm just going to run the installer here. Click continue. We're just going to follow the default settings here. Continue. So it's the same on Windows, right? Just follow the defaults. And here, this is what I wanted to point out. Somehow it says, so by default, it selects install for me only, which is great. But then down here, it says you cannot install it into this location, which to me seems to be a bug. But now if I click on install on a specific disk, and then click again on install for me only, suddenly it works. I would now click on continue and follow through. But I'm just gonna stop it here because I have already installed this thing, okay? And this is gonna take a few minutes, even on a fast computer, to complete because there are so many packages involved, okay? So to verify that the installation succeeded on the Mac, you open the Finder, go to Applications, and click on Anaconda Navigator. On Windows, this will be in the Start menu. Just click here, and it fires up, which means, yay, the installation succeeded. Okay, I'm gonna close that again. It was just to verify that the Anaconda installation actually worked, which it did. What we're going to need now, so I'm going back now to the installation instructions I published earlier. It says here, follow the MNE installation instructions. I'm gonna click here. And here it says you need to download a certain environment file and need to run a bunch of commands. So this is pretty much similar both on macOS, on Linux, and on Windows. Just trying to move around the on-screen controls of Zoom's screen sharing. They always get in my way. Okay, so I'm going to follow the instructions for the Mac. Actually, there might be an easier way. So we can actually skip the first line here. We don't need to run this conda install base whatever thing, but we're going to run this line, curl remote name, blah, blah, blah. This is essentially just gonna download a file, this environment.yml file. Okay, I'm just gonna copy that. I'm gonna open a terminal and I'm gonna paste this, okay? And this is going to download this file. If I now look into my home directory, it will be here. You see, environment.yml. Okay, it's similar on Linux. You also run pretty much the same command. And on Windows, you can just click on this file and download it, or probably you want to right-click onto this file and click on download linked file or whatever your browser offers. You want to be sure to keep the extension .yml. And then we're going to copy this line here. Oh, by the way, on Windows, obviously, you want to run the Anaconda prompt. Okay, so this terminal that I opened here on the Mac is just the terminal app. Whereas on Windows, you can find a similar terminal if you go to the Start menu, and then it says Anaconda prompt. Okay, so then you just run, I'm just going to make this a bit bigger. You just run this command I just copied from the website, which says conda env update, and file then points to the file we just downloaded. I'm now, I might as well run it. Okay, I'm just going to run this thing. It's going to take quite a bit on my computer because it's old and slow. And what this is going to do is it's going to create a virtual Python environment with all the dependencies we're going to need for running MNE Python. Is there anyone still having issues following through until here? Because there is an alternative way we can achieve the same thing. Okay, there's an alternative way that might work for you. Have you figured out how to run the Anaconda Navigator, the one we started earlier? So on the Mac, I just go to Applications, Anaconda Navigator. On Windows, there should also be an Anaconda Navigator. You should find it in the Start menu. And I have several tabs here on the left. One of them is called Environments. Yeah, okay. So you click there. I have so many environments here, but so for you, it should probably only say base and then in parentheses root, and the rest should probably be empty. Okay. And what you can do now is you can click on Import. Click on Import, and then it says down here, Specification File. You click on this Browse button to the right, and navigate to your environment file, to the one you just downloaded. So you navigate to it, you select it, open or whatever it's called on Windows, and you see by default it inserts a name for the new environment. In this case, just MNE, because that's the default we set in this environment file, but you can enter anything else. You can also just call it MNE Pybrain 2020 or whatever. And you click Import, and then it starts to get busy, and it may appear to have stalled for several minutes actually, because the progress bar may just seem to have gotten stuck, but it's actually downloading and installing stuff. So let's move on for now, and for anyone who hasn't installed MNE bits so far, and JupyterLab, I'm just gonna show you how to do that. So you're going to open your terminal. On Windows, it's going to be the Anaconda prompt. On the Mac, it's just going to be a terminal. You have to activate your new environment. Okay, what you do is you do Conda activate MNE, because that's the default name of the environment. If you assigned it a different name using the method I showed you earlier, you would have to use the name you picked. So I'm going to use Conda activate MNE. And then it says here MNE, which tells me, okay, the environment has been activated. Great. Okay, we're going to need to install JupyterLab. And after that, we want to install MNE bits and some of its dependencies. To install JupyterLab, we just run Conda install JupyterLab. And it's gonna process a bit. And here it offers to install all these things. And we're just gonna say yes. And again, my computer is slow, so it takes a bit. Hopefully, it's not as painful on your computer. Okay, perfect. So for me, the installation is done. Now, the only thing that remains to be installed is MNE bits. So I'm going to go back to the installation instructions. I don't need to run the Conda activate command because I'm just leaving my terminal open. But I do want to run pip install MNE bits NI-bubble, or whatever, however it's pronounced, and PyVV, which is a software for writing brain vision data. Gonna paste this here, click enter, press enter, and again, my computer takes a break and I'm set. So I just received the questions, is there any limitations if I use Google Colab instead? Yes, there is. There is a reason why we didn't choose to just dump these notebooks we're going to use on Google Colab. And the reason is that we do want to use the interactive visualization capabilities of MNE. We want to demonstrate them. And they don't work properly with inside the browser yet. So they only work locally, and they won't work properly on Google Colab. You will only see static images with which you can not, or yeah, I think you cannot interact with them at all. Or if you can interact with them, the possibilities you have of interaction are very limited. So I highly recommend to run these notebooks locally and not in Google Colab. Okay, what's the status? Are we all ready to get started and have some fun? Just checking Slido. Anyone still experiencing issues? This is your last chance. We're going to move on after that. Okay. Okay. So I got a message. It says no module named MNE for me. Where does that happen? And are you sure you ran the activation command, Conda activate MNE? Let me try to download the sample data. Okay. You do see MNE. So you're trying to run this command, right? This should work. If the MNE installation has been successful, this actually should work. Okay, so what you can do to verify that the installation worked is you activate your MNE environment, Conda activate MNE. In my case, actually, it's MNE-new. Then you type Python, and then you just type import MNE. And this should work. Ashley, does that work for you? On which platform are you, Ashley? Is that Linux, or is that Windows, or is that a Mac? I am a bit clueless, because if you have this MNE environment, then all the dependencies, including MNE, should have been installed. Okay, what you can do is, can you open the Anaconda Navigator? And you go to your MNE environment, like you click on environments here on the left, and there you select your MNE environment. In my case, it's MNE-new. In your case, it will just be MNE, I suppose. Then it produces a long list of packages. And if I scroll down here, at one point, it says MNE. Can you see that? If you cannot see that, this means that something went wrong with the creation of this MNE environment. Ashley, you still with me? You cannot find MNE in this list? If it's not in the list, then something went wrong when creating this environment. I would suggest you do this again. So first, you remove this environment. So you click on the environment, MNE, you click on, it loads a bit. Yes, exactly. Remove the old environment and create an environment again. Okay. And I think we'll move on for now. Yeah, no worries. It's important that everybody has a working system. This already demonstrates one of the difficulties when working with Python compared to, let's say, MATLAB, where you install MATLAB and you're set. Here it's typically a bit more complicated. Okay. Whew. So now I'm going to quit the navigator. I'm going to go to the Anaconda prompt again. I'm going to activate our environment. In my case, MNE-new. In your case, MNE. I'm going to run Jupyter Lab. This will open up a browser. And on the left side, you can actually navigate your files. So first thing you need to do is, obviously, you need to download the notebooks we're going to use today. There is a Google Drive. The link, I think, was shared in the Zoom chat. I think that's the easiest way to retrieve the notebooks. Just select the files, right-click, click on Download, and Google Drive creates a zip file, which may sometimes take a while for whatever reason. Okay, I'm not sure why this is taking so long now. Just going to cancel that. I'm going to try again. Now it's done. I'm just going to save that to wherever I can find it, downloads or whatever. Just going to put it here in the downloads folder. Going to save it. Going to go there and unzip the files. Okay, and here I have them, all 4 of them. Now, back to Jupyter Lab. We're just going to navigate to the folder in which we saved the just downloaded IPython notebooks. And in my case, actually, I have prepared something. I have it stored under projects, MNE workshop, my notebooks. And I'm just going to open the first one. Just double click there. Did everyone manage to get there and open the notebook? There's a yes. There's one yes. That's great. There's two yes. Okay. I'm starting to gain confidence. 3 of them. Okay. Okay. Is there anyone still lots of yeses coming in? This is great to hear. Is there still anyone having some difficulties? Maybe somebody who cannot find the notebook file from within JupyterLab or anything? Okay, doesn't seem to be the case. So now I don't know what these yeses refer to, but I just assume that it's working for you. Whew, what a ride, what a ride. Okay, but here we finally are. The first thing we're going to do, have you ever worked with a Jupyter notebook? I'm just going to show you the basics. So essentially what we have here, it's essentially a mixture of, if you will, a text document and code snippets. And it's arranged in so-called cells. This is a cell, this is a cell, this is a cell. And these cells can be executed. And the easiest way to execute them is by just pressing Shift and holding pressed Shift, pressing it down, and then pressing Enter. So this will run the currently selected cell and move to the next one. And you can see here, it added a number, which means that, okay, this was the first cell we executed. Okay. Can everybody execute the first cell and tell me if things break and the computer starts to burn down? Or is it working for all of you? Let's check the chat. All good. All good. Perfect. Oh, I'm so relieved. No fire there. That's great. I was already about to call the fire department. But okay, now we can calmly move on. Okay, so the first, what did we do there? We just imported matplotlib, which is a library that MNE is going to use for plotting. I'm going to explain in a bit why we need to explicitly import it here. We imported pathlib, which is just a package that makes it easier to work with paths, meaning directories and files. And finally, yay, MNE. This is going to be our working horse today. Okay, we imported matplotlib so we can execute the matplotlib.use command. What it does here is it tells matplotlib to use a very specific, they call it a backend, to actually draw and arrange our plots. And so there's different backends available. And by default, if you run the Jupyter Notebook, all figures that MNE creates are going to be embedded within the notebook. This is going to happen, for example, if you run the notebook on Google Colab. And this comes with the issue I mentioned earlier, that these embedded figures are not interactive at all, which is not super convenient if we want to explore some data interactively. So for that reason, we run, this line is really important. It ensures that whenever we create a figure, or MNE creates a figure, it will actually pop up, where you get a new window, and we can actually click there and interact with stuff, which is going to be super neat. You will see that in a second. So we need to run this cell as well. Okay, so now we want to work with some sample data. MNE comes with a bunch of data sets that you can use to, test certain analysis or just explore the functionality of MNE. And one of these data sets is called sample. It's an audiovisual data set that contains MEG, EEG, and EOG recordings. And we're just going to ask MNE to find this data set and to tell us in which directory the data set is located. Now, if MNE hasn't already downloaded this data set, it will start downloading. If it has already been downloaded, MNE will just point us to this directory where the files have been saved. So I'm just going to run this. So in my case, the data had already been downloaded previously, and I just get this path. So it points me to this directory. We can have a look at this. So it's going to be Richard in my case, MNE data, and then it's going to be MNE sample data. And this is, whoops, so this is the data set, lots of files. We're only going to use a few of them. Now, finally, let's load some raw data. To load raw data, we're just going to use the mne.io.readRaw function. Okay, and it accepts as an argument the path to a raw data file we intend to load. And in our case, it's going to be, I just showed you this directory, and it has subdirectories and lots of files, and I just picked this one. Now, if you're working with actual experimental data that you acquired, of course, you would have to point MNE to the data file within your project directory that you want to load. But here we're going to use the sample data, and we're going to load it. And this was pretty quick. So MNE tells us some things about the data. It says which time range we can find in the data and how long it is. And it also says here data not loaded. So this is something we are going to look into in further detail later on. But for now, yay, we just accessed our first raw data file. Oh, there's a question. Somebody is getting an error message which indicates that the file cannot be found. This shouldn't happen. So what I would suggest in that case, what you can do is you open your file browser, and you go into this directory where it says MNE data, MNE sample data. You just delete this entire directory, just delete it so the data is gone. Go back to the notebook and run this cell again, which should download the sample data once more. Okay, somebody had a similar issue. Borja, what was your solution? Where did you move the folder? So by default, MNE saves all the data in your home directory under in the subdirectory MNE_data. Borja, you said you moved it to your root path. I'm not sure what you mean by that exactly. Your home directory. Yes, it should be in your home directory. Is it not in your home directory? For Iona, it's in the home directory. Iona, can you just try that again? So it seems for some, the download actually fails. Is there anyone experiencing issues accessing the data, downloading the data? Okay. Daniel, how did you download the notebook? From GitHub. Can you try fetching it from Google Drive? It seems that GitHub may do weird things when you fetch it. Iona, can you try that? Can you try again to restart the download? What you're seeing seems to be an issue with OSF, the open, what is it? I forgot. And they seem to have some server issue, I'm afraid. Open Science Framework, I think that's what the name means. Great, so Daniel has a working notebook now. Borja, where was, that would also be of interest to me. Where did the data end up previously before you moved it? Because that seems kind of uncommon. It should always be in your home directory. What you can do is you can try to click on kernel, restart kernel, and execute the first 3 cells again. This should reinstantiate the download. What you can also try is you can pass this force update equals true. Run this line. See if that helps. Ayana, are you still with me? Ayana, last chance. Otherwise, I'm going to move on. I'm going to check Slido. Okay. Okay, Ayana, I suggest you restart the IPython or the kernel in Jupyter Notebook and run the first 3 cells, but modify this command, this get data path, such that you pass force update equals true and see if that does anything for you. Otherwise, this seems to be an issue with the Open Science Framework servers, but typically these are only short-lived, and it should work again, should start working again very soon. Okay, so let's get back to our data. So we loaded the data, hopefully. And okay, and now let's get fancy and visualize things. So I'm gonna run raw.plot, and it's gonna take a few seconds on my computer. And then you should see a plot, a figure popping up. Is that the case for everybody? Does anyone not see a figure popping up? Okay, there's no one saying anything. Okay, so I just assume it's working for all of you. Okay, so what we see here now is the raw data we just loaded. You can navigate there by using your cursor key. So I'm pressing right, which moves right in time. It's a bit slow and lagging, especially on my computer. You can press the up and down keys to move through sensors. And there's many more things. For example, you can press the button or the key B, like butterfly, which will calculate a bit. And you see butterfly plots. Well, I mean, they don't exactly look like butterflies here, but you essentially see an overplotting of all channels for different channel types. And here you can already see that we have different channel types, magnetometers. So for those who are not familiar with MEG, there's typically 2 channel types, magnetometers and gradiometers. We have these 2 here. So these are MEG channels. There is an EEG channel, or actually a bunch of them. We have EOG and something called MISC, which are channels that code for triggers. There is a question. The command was B. And because it's difficult to remember, there is a help button down here. Just going to click here, and then it tells you, okay, if you press B, it toggles butterfly plots on and off. And you have many other things you can do. Let's toggle butterfly off again. I'm going to change the time scale a bit. On my Mac, I'm doing that by pressing the function key and left or right arrow keys. You can change how many sensors you can view at once. On my Mac, again, this is the function key and arrow up and down. So arrow up means more channels, more sensors simultaneously, and arrow down means fewer of them. As you can see here on the right side, this scroll bar actually comes in different colors. So this is like a light blue, a dark blue, black, and yet another black. So in this case, the light blue refers to the gradiometers, which you can also see here on the left. There is this, this unit. This is femtotesla per centimeters, which indicates it's a gradiometer because it's a gradient. It's a change of magnetic field power per centimeter. Whereas these down here, the dark blue, are magnetometers. It's just femtotesla. The black ones are our EEG channels. And down here, we have stimulus channels. If you scroll to EEG channel number 53, you can see it's gray. If we click on it, it turns black. We click on it again, it's gray again. What's going on? Well, this channel, as you can see, it's obviously very flat, so probably didn't produce any useful signal. So we would assume that this channel is broken. And by clicking on a channel, we can actually mark it as bad. So try that out. Click on a few channels. And you can revert this by clicking on the channel again. Another cool thing you can do here is you can add so-called annotations. Annotations allow you to, well, annotate your data. For example, if you discover a segment of data that seems to be bad, maybe there is lots of noise and you want to exclude that from further processing, you can just mark it as bad. To activate annotations mode, you press the A key, A like alpha. And then this not so intuitive interface comes into play. You can just type bad, whatever, assign it a label. In my case, I'm just going to call it bad noisy. Click on add label. And now we have here a list of potential labels. Currently, there's only one, bad noisy, the one we just added. It's selected. That's why there is a red circle around this. And now, if I click and drag my mouse, I can actually mark segments of data, in this case, as bad. If I right-click, the annotation goes away. Isn't that cool? Back to the help, we can actually see that there's a few more things we can do, but this was, I think, the most important stuff that you needed. Okay, I'm just gonna close this. Gonna go back to the notebook. Could everybody follow until here? When you plot raw data, you can press B to toggle the butterfly plot. If you press A, you can add annotations. If you click on a channel, you can mark it as bad. If you click again, you can mark it as not bad. That's the most important things. And there's always the help in the bottom left corner that brings up this reference of which keys and buttons to press. Okay, perfect. So, one thing we always want to be certain about when we're actually working with our experimental data is we want to, or at least when we're working with evoked designs, we want to be sure that we actually have access to all the experimental events that happened. And on many MEG systems, these are called in a separate stimulus or trigger channel. On EEG systems, there might be different approaches, but common approach is to have a separate channel which contains your events. And to extract events from these channels, or this channel, sometimes it's one, sometimes it's more than one, we can use the MNE function find events. So I'm gonna run this. Oh, so there's a question, where is the data stored? So the data is stored in your home directory under MNE_data. But you actually shouldn't have to worry about that because it's a default location that MNE creates automatically when you try to access this data set. So I'm not sure which issue you're currently experiencing. Does that help you, Hamid? Okay, just get back to me if you, oh, your system ran out of space. Your system ran out of space. Okay, so are you on Windows or are you on Linux or Mac? Windows 7. Actually, I'm not. Is there any Windows user here who can tell us where this sample data is stored? The value of sample data here is what we're going to need, because I don't know where it's stored on Windows, honestly. Ah, perfect. So Hamid, yeah, just delete this directory and start downloading again. Okay, back to our events and triggers. So to find experimental events in the trigger channel or channels, we use MNE.findEvents, and we pass in the raw data, we just read. And it tells us, okay, found 320 events. Sounds good so far, right? And with the following event IDs. Now, event IDs is the way that events are typically encoded, but they are not very human readable. So what we typically want is we also want to map between these numbers and an actual event name. So what we do is we create a dictionary which maps an event name or maybe an experimental condition to event numbers. Okay, and we're just going to call this event ID. Ta-da! Okay. So this events thing we created up here, if we have a look at it, just going to insert a new cell here, just going to print this thing. It's actually a NumPy array. It has 3 columns, and in our case, this would be 300. That's why I didn't include it in your notebook because it's so long. It's actually 320 rows. Okay, and the first column is always the sample number. The center column we can ignore for now, and the last column is actually the event number. Collapse that here. That's too much, taking up too much space. Okay, so now let's see if we can figure out how many events with event number 32 we have in our data. So I told you it's a NumPy array. Okay, so we can actually index this NumPy array in the following way. So we say, okay, give us from our event array all rows where the third, this is the one, the last column contains the value of 32. Okay, this would give us all rows where event number 32 occurred. Now to figure out how many rows that is, we just say len for length, call this on the result, and we get 16. Does that make sense so far? This means we have 16 in our data, we have 16 events of type button. Okay? So now I would like to ask you to figure out how many events of the visual type we have. Can you manage to do it? Or are you going to run into issues? Do let me know. Just gonna give you a minute, and then I'm going to show you one potential solution. So remember, the event codes for the visual stimulations are 3 and 4. You can also drop a line in the chat when you're done, or if you're having issues. Okay, somebody post. Yeah, perfect. So Anna posted a solution. I'm just gonna copy that here. I'm gonna copy that to my notebook. So let's see, number of visual events. So what she's doing is she's just summing up. She's extracting first the visual left events, then the visual right events, and just summing that up. There is another solution by Johan. This is neat. So he's using a logical OR. Actually, personally, I didn't know this was possible. So he selects all events where the last column equals 3 or 4. So that's also quite neat. That's very elegant, Johan. Perfect. I would like to slightly modify that. Instead of using the plain event numbers here, why not take advantage of this event dictionary we defined up here? So what I'm going to do is I'm going to use this event ID for visual left, and here instead of 4 I'm going to say event ID of visual, and this is already a bit more readable, I think. Make sense? Okay, going to move on. So now we have our events. This is great because we can now visualize our raw data with the events superimposed onto them. Let's plot this. It's not the prettiest figure, I have to admit, but you can see we have all these vertical lines here now. And not only do we have vertical lines, but these lines also have a label. And the label equals the events we extracted from the data. So visual right, auditory right, visual left. So this is already super neat. Okay, close that. I want to show you some more important bits about raw data in MNE. So one of the most important structures is actually the info structure. You can always access it via raw.info, or when you have an epoch.info, when you have an evoked later on, it's going to be evoked.info. It contains lots of valuable information. For example, it tells us here, in my data set, there is currently 2 channels marked as bad, 1 MEG channel, 1 EEG channel. It has a list of all human readable channel names. It tells us whether high pass or low pass filters have been applied to the data. Super important to know sometimes. And importantly, it tells us the sampling rate. Also information like the measurement date. So these data were recorded on December 3rd at 7 p.m. UTC. So that's London time, essentially. Sampling frequency is about 600 hertz. And we can also, all this info we can see here, we can extract programmatically, okay? So to get the bats, which we can see here, that's the bat sensors, we just do raw.info and then in brackets, bats, and there we get a list of our bat sensors. Channel names, this is going to be a long list because we have so many channels. You can see so many. Oops. And just for demo reasons, I'm just going to slice that. It's just a list. So to get the first elements, the first 10 elements, I'm just going to use column and 10. I'm going to run this, and this is going to be the names of the first 10 channels. And lastly, there's much more information we can get about each individual channel. This is stored in the this is something, I think it's supposed to be short for channels. I'm not sure why it's not simply called channels, but and it's a super long list, and we're just gonna extract the first element just to demonstrate what it does. So it encodes which channel type is it specifically, information about calibration, which coil type it is. That's important for MEG, the physical location of the channel, in which units were the measurements taken, and the channel name, obviously. So this typically you will not need. I just wanted to show you it is there. So MNE knows quite a bit about the data it's working with, and it needs to know so much to do the right thing, depending on which operations you're asking it to do. Okay, I just just had a brief glance at my clock, and it's already 12:05. Took a bit long to get everybody up and started. I think we were going to do a lunch break from 12 to 1. Is that good with everybody? So because I would then say we're going to go on a break now until 1, would be a bit shorter than, oh, you're 1 hour behind. Perfect. Yeah, right. UK time. Yeah, okay, it gives us another hour. That is great. Yay, okay, absolutely. Okay, so then let's just proceed. That makes me happy. I was already worried. Okay, so let's just visualize the locations of our sensors, because that's super important. We need to know where the sensors are or where located in respect to our participant's head. And let's just do that with the function. It's called plot sensors. And here in this example, we're going to pick the EEG channels. If I run this line, I get this pop-up. Again, not super pretty, but it does do the job. So each of these filled in circles or dots or however you want to call those depicts one EEG sensor. One is red. The red one is the one that was marked as bad before. We'll get back to that in a bit. If you click on any of these, you actually get their names. You see that? You can see their names popping up here. Okay, let's close that. We can also get a 3D visualization of these channels. Now, this doesn't look too useful without a head, but yeah, it's sometimes may help to actually figure out what this was supposed to look like in 3D in real life. And again, if you click on the channel names, you get on the dots, you get their names. And obviously, there seems to be a bug because I'm getting names for random channels, but not the ones I'm clicking on. Okay, so there's a bug that needs to be fixed. But you get the idea, I hope. Okay, so as you saw in the first sensor plot, sensor visualization, I'll bring it up again. We have this channel marked as bad here. Let's just decide to add another channel as bad. Let's have a look again at our info structure. So an info bad stores the information about which channels, which sensors have been marked as bad. Let's just add another channel. Maybe, you know, we inspected it and we figured, no, it's only producing noise. We want to throw it out. To do that, we just add a list of these channel names to the existing list of bads. So you can do it using this syntax. It's quite simple. If you wanted to add another channel, you would just do something like this, okay? To add 2 channels as bad. But we only want to add 1 for now. It's important to have these brackets here, because otherwise all hell will break loose. So mind the brackets. So we're going to add this channel to the list of bads, and we're going to visualize the sensor locations again. And as you can see, we now have not only have 1 channel marked as bad, but 2. It's EEG 053 and EEG 051, which is Yes, which is hopefully the one we just added. Perfect. So this is working. So the data set we have here, let's have another look at the infrastructure. Raw.info. The data set we have here comes with, I cannot find it right now. Ah, here it is, this line. It tells us how many channels of each type are actually available in this data. So we have 204 gradiometers, 102 magnetometers, 9 of these stimulus or trigger channels, 1 EEG channel, and 60 EEG channel. These were all simultaneously recorded. Let's assume we only want to look at a subset of these channels. Maybe we only want to look at the EEG channels and the EOG channel. We can actually select a subset using pick types. So what we would do is we would first create a copy of our existing raw data, because the function pick types we're going to use afterwards is going to work in place. So if we were to run raw.picktypes without the copy in between, it would modify our just loaded raw data. That may not be what we want to achieve, actually. So we insert this copy thing here, okay? So we take our raw data, we create a copy, we pick certain channel types. By default, MNE will always include the MEG channels. This will change with the next version, but for now, to exclude the MEG channels, we have to specify MEG equals false. Okay? So meaning no MEG channels. We do want the EEG channels though. So we say EEG equals true. And we do want to include our sole and lonely EEG channel, so we pass EOG equals true. This last bit here is only important to tell MNE, hey, please do not remove the channels marked as bad. Normally it will remove them, it would just drop them, but for the sake of this demonstration, we do want to include them. So we say exclude is an empty list, meaning please do not exclude anything. Please give us all the channels, whether they are bad or not. Okay, and after we've run that, we can have a look at the number of channels that actually remain, 61. And if we look at the infrastructure of this newly created raw object, we can see here, channels, it says 60 EEG, 1 EOG. Remember, when we scroll up again, the original, it used to be gradiometers and stim channels as well, but now we only have the EEG and EOG channels left. And there is questions coming up. Okay, so there's multiple questions. Why are the sensors outside of the head? Which sensors are you? Which visualization are you referring to? Are you referring to the? I think I can already tell which one it is. Are you referring to this visualization? So it's a difficulty of the visualization of sensor topographies, because obviously the head is three-dimensional and the sensors are placed on something that's on the head, which is pretty much like a sphere or approximates a sphere. So now when you plot that on a 2D surface, how do you actually do the transformation from 3D to 2D? You could try to squeeze it all inside this head circle, or you could just say that, okay, this head circle is, for example, the horizon of the head, and everything below the horizon you plot outside the head circle. Does this make sense? So assume this electrode here is at it's like OZ, you know, the electrode at the far back of the head and far down. It's very low, and you would plot it outside in this kind of transform. And therefore, it can make sense to also have a look at this 3D visualization, although it's not the. Okay, so for example, let's do that briefly again. So the sensor that was, oh my goodness, that's so choppy here. The sensor that was plotted, so maybe we can open both, or will my computer just die? Here, this sensor that was EEG 60, that's plotted outside of the head circle. This should be this one. Again, I don't get the proper label. And you see it's in the far back of the head. And it's just the difficulty in how do we do the 3D to do the 2D, 3D to 2D transform. It's also a recurring question when we later do the topo plots and topo maps. Lots of work has gone into that, and there's still, it's still not an optimal solution we've achieved there. Okay, so there's another question. There's a runtime error. Yes, please restart the notebook. I mean, this runtime error thing should not happen. The command you pasted into the chat seems to be perfectly correct. So this should work. Okay, if you try to add multiple bad channels at once, it should work like this. You have this list, and you need to quote them. Okay, you need to remember the quotation marks, and then have a comma between them. I'm gonna run this, and here it worked. Okay, so this should work. Where were we? Oh yeah, right. Totally lost track here. Okay, what did we do? We selected a subset of our raw data which contains only the EEG and the EOG channels. We did that by using the function pick types, and we ran this function on a copy of the raw data as to not modify the original. We then realized, okay, we have 61 channels now, and if we look at the infrastructure, we can see 60 EEG channels and 1 EOG channel are now present in the data, which is great. So let's plot these data. Et voila. You can see that on the left side here, it only says EEG, EEG. On the right side, everything is black. So this gray thing here is just a scroll bar. Okay, we scroll down. And here is just the 3 channels I just marked as bad. Well, actually, it's 2 channels I marked as bad. And down here, there is the EOG channel. And you can see that that's actually super nice. You can see these blinks here happening, and you can see how it relates to the EEG signal. Okay, this is something that we later will want to correct for. Okay, was that working for everybody? In that case, I would like to encourage you to go back to our raw data again, and instead of selecting just EEG channels, please select all MEG channels, and visualize them. And after that, please only select the magnetometer channels and visualize them and see how that goes. And please post any questions to the chat. Oh, and by the way, I mentioned this earlier, it's super helpful, and for me, it's priceless to always have a browser tab open pointing to mne.tools and the documentation. If I want to do something, for example, I want to use pick types. I'm just gonna search for that. And then it says here, whatever, MNE pick types or raw pick types. And then you can see which parameters it actually accepts. And there is even links to examples demonstrating the use. So in this case, it's quite a bunch because pick types is a function that we constantly use. You know, almost all examples actually make use of it. So, and you can see it has this MEG option, and it tells you specifically what to do if you want to include all MEG channels or only gradiometers or only magnetometers or whatever, which is what you're going to need now to solve this exercise. So there is a question, are there any efforts to give this package a graphical interface? That's a difficult question, in the sense that the idea of MNE was always to be a Python package that allows you to programmatically do your data processing and analysis. However, there is, I mean, as you already saw, there are ways to visualize the data or analysis and analysis results. Actually, the most sophisticated visualization is when it comes. It was just added, I think, half a year ago, with regards to source estimates. We have the so-called time viewer, where you have a 3D visualization of the brain, and you can see how the activation changes over time. And you can, click with your mouse and drag, and it's super cool. And we will see that later. But if your question was whether there's going to be like a graphical user interface that makes everything click and point, this is currently not in the works as part of MNE itself. However, there is some effort going into, let me see what this was called, MNE Lab. It's called MNE Lab. This is by Clemens Brunner, who is also a core contributor of MNE. I'm not sure if they have a proper website yet. Okay, but it's essentially, let me open this screenshot. It's essentially a graphical user interface that allows you to open your data and conduct some very simple processing and visualization just by clicking with the mouse. But it's in an early stage, and functionality is pretty limited. But maybe that's something that could at some point develop into something bigger. Does that answer your question? I was just wondering if they have, I think that's all the documentation they have. Okay. Is everybody done with the small exercise, or were there any issues, or did any new questions come up? No questions? Everything working properly? Okay. So let's just move on to cropping and filtering the data. This is almost too easy. So to crop raw data, we just call its crop function. Sorry, this one. We just call its crop function. Again, crop works in place. So we first create a copy, and then we call crop on the copy. Crop accepts a, oops, why is this not? Crop accepts a tmin and a tmax argument. So typically we only want to cut something off at the end, so we pass tmax. So, and this line here would crop the data to a duration of 100 milliseconds. And we can actually have a look at the time points that are retained after cropping by looking at the last element of the dot times attribute. Okay, so getting the last element of a list, it can be done by indexing with -1, and that's what we're going to do here. So first going to crop to 100 seconds, and then we're going to look at the latest time points in the cropped data. And we can see it's not exactly 100. It's close to 100. This is because MNE takes into consideration the sampling rate, and it doesn't just crop halfway between a sample and another, but it just tries to squeeze as many samples into the time period you specify, and therefore you may end up with something that is slightly shorter than the than the Tmax value in seconds you specified up here. Okay. So now let's filter the data. Again, filtering works in place, but this is okay. We're just going to work on this crop data set here. So now we're just gonna skip the copy maybe, and there's a function called filter, and it takes quite a number of arguments. If you place your cursor right after the opening parenthesis and then press Shift and Tab, you can see the so-called function signature, which means which parameters does the function accept, and below that there is documentation. So this is also super helpful. This allows you to have a brief look at the documentation without having to switch tabs constantly and going back to the MNE website and searching for the docs there. Okay, so but of interest to us for now are two parameters, lfrec and h bound, and h-frec is the upper frequency bound. So what we're going to do here is essentially we're going to run a high-pass filter with a cutoff of 0.1 hertz and a low-pass filter with a cutoff of 40 hertz. And if we run that, we're going to see, what's going on there? It's a runtime error. Well, this is strange. This shouldn't happen. Well, maybe let's read the error message for once. And it tells us about MNE does not load data into memory to conserve resources. And maybe you still remember back in the very beginning when we loaded our raw data, it said here, data not loaded. So what it did there is it only read some meta information like how many channels, how many, what is the duration, and maybe some information about our participant. But it didn't actually load all the data into memory. So, but now filtering obviously needs the data to be loaded. Okay, so what we're going to do is we're going to load the data by just using the function load data. It's very easy. And then we're going to, actually I'm seeing here, I'm creating a copy. So let's just create a copy of the data, of the loaded data, and then applying our filter should work, no problem. Okay, let's run this. So it tells us here, reading almost 100 seconds. This is all right, yeah, because we crop the data to 100 seconds. And then it tells us that it's going to apply a filter. It's a bandpass filter, so it wasn't entirely correct what I said previously. I said we're going to apply a high pass and a low pass. No, it's actually going to be a bandpass. And we can see the frequency range that is going to be preserved. Here's some more information about the filter that gets created and applied. Okay, so now to get an impression of the difference between the filtered and unfiltered data, what we're going to do is we're first going to plot the unfiltered data, and then we're going to plot the filtered data. So it's going to open two plotting windows now, and it's probably gonna make my computer want to cry. Oh, it's working. It's number 1 and number 2. It's difficult to see much of a difference on this small screen, but on the left side you have the unfiltered data. There's lots of high frequency noise, and on the right side you have the filtered data, which looks better. Now, just looking at the raw data may not be the best thing to do to investigate how well a filter worked. What we can also do is we can plot the power spectral density, or PSD. To do that, one would just call the plot PSD function on the data of interest. Here, it's going to be a bit more complicated, because actually what I want is I want to create a single plot with two subplots. And the first one shall be the PSD of the unfiltered data. The second one shall be the PSD of the filtered data. There's a question. Is there a way to label figure windows to keep track of what is what? Yeah, yes, I just realized this exact same issue. I think, so by default, I mean, they do have a title by default, right? So let's run this again. And the default title is their file name. Now, obviously, since both were based on the same file, the filtered and the unfiltered data, they have the same title, which makes things difficult. I think we can pass a title parameter. I'm not entirely sure. Yeah, title. Let's just call it title unfiltered and title filtered. Let's see if that works. I'm not sure. Perfect. That makes life easier. Does that answer your question, Johan? Okay, perfect. So I think most MNE plotting functions accept that. You can just pass a title. That's a very good point. Okay, where were we? The PSD. So we want to create a single figure with two subplots. First subplot shall contain the unfiltered data, the PSD of the unfiltered data. The second subplot shall contain the PSD of the filtered data. And for that reason, so I'm not sure how familiar you are with matplotlib. Plotting is similar to what MATLAB does. So I haven't been using MATLAB in years, so maybe I should take that back. So the way you do it is, here we create two subplots, essentially. These are going to be stacked upon each other. And then the plot PSD function actually takes an argument x for axis. And we're going to tell it, okay, please use the first axis for the unfiltered data and use the second axis for the filtered data, and then make things a bit prettier. So let's just run this. Let me close these other windows because they're a bit distracting. So, and here we have both PSDs on the top before and on the bottom after filtering. And we can see here, like this vertical dashed line actually indicates the high pass filter cutoff, which we set at 40 Hertz. Oh yeah, almost forgot about that one. So you can actually click and drag, and if you then release, you get the average power for a certain frequency range. Okay, so this should be only noise, also in the front. Okay, well, so be it. And if we look at before filtering, it all looks pretty similar, but so far we didn't do a proper artifact rejection anyway. So, okay, so this is how you apply a filter and then later on check the result, right? It's really important to check the PSDs to see that the filter actually did do what you expected it to do, but also to have a look at the raw data again. Because I mean, there are some horror stories out there about weird filters doing all kinds of odd stuff. So always do visually inspect your data. And here I get lots of red because my computer can barely handle it. Okay, and now I have a super simple and small exercise for you. Please apply a different filter. We want to use a 1 Hertz high pass and 30 Hertz low pass now, and then want to see the PSD of that. So essentially, you have to go back here and create the PSD plot. This is just so that you use this function yourself for once, otherwise it's difficult to memorize. And again, if you need help, there is documentation. If you look for filter, there's mne.filter, it's a function. So currently we're working with raw data,
which means that you typically want to look for something that goes like MNE raw.filter. You will get used to that after a short while. And here you get all the nicely rendered documentation. Just gonna grab myself a glass of water. Okay, are we all good? Oh no, there's questions in the chat. Can you repeat the instructions? Okay, so the instructions are actually right here, right down here in the green box. So so we just applied a filter to our cropped EEG data. And the idea is that now you simply apply a different filter and visualize the PSD and see. And what you should see is actually that the vertical, let me just plot that again. What you should see is that this vertical a dashed line should be moved to the left in direction of, it should actually be positioned exactly at 30. So if you hover with the mouse, I'm not sure if you can see in the top right here, shows the coordinates. And if I place my cursor on top of this dashed line, it's about 40. And if you apply a 30 hertz low 30 hertz low pass, then actually the dashed line should be at about 30. Actually, exactly 30. Okay. Okay, it's clear now. So then the second question, how do you get the figure window to open separately and interact? So yeah, that's to open. So if your window opens inline, you probably haven't run this line here. This is important. This matplotlib.use qt5ag. This will ensure that windows actually pop up, like you can see on my screen, and do not open inline in the notebook, because if they open inline, we don't have any of the interactive features. Does that help you, Saskia? You run that line, but it still doesn't work. Can you restart the notebook? So what you can do is you can do run kernel. You click on kernel, and then you click on restart kernel and run all cells. Because sometimes, for whatever reason, if matplotlib has decided to use a certain backend, it cannot be switched until you restart the kernel. Try if that works. It's really important because, like I said, the interactive features are otherwise not available, and that's just painful. I mean, you cannot browse your raw data, for example, and you cannot mark channels as bad and the like. Is it working for you now, Saskia? Okay, so Saskia, yeah, the figures might still be loading. It may take a while. There was a question whether this can also be run in Spider. Yes, you can also run all of this inside your favorite integrated development environment. So I typically use Visual Studio Code. I used to be a big fan of PyCharm Professional, but we thought that for this tutorial specifically, it would make most sense to run the stuff in JupyterLab. But if you want to run it elsewhere, that's totally possible, and it should work. Yes. Although I do remember an issue that a colleague of mine once had with interactive figures and Spider, but I'm not sure exactly what it was, and may have been resolved meanwhile. Okay, and Saskia is all set now. Okay, last step. Now that we've filtered the raw data, we want to save it. And well, to save anything, you typically just use the variable name, append.save, calling the save function. You pass the output path. And I always have this override equals true, because typically I want to run my scripts multiple times because they change small details, and by default MNE won't overwrite existing files unless you specify override equals true. Okay, so what do we do here? So we're going to tell MNE to store the file inside the directory called out data relative to our notebook. And inside this directory, it shall create a file EEG cropped filled raw pip. Yeah, these file names are always a bit funny, long and a bit odd. Okay, but if we run this, it will throw an error message. Why is that? Well, because this directory out data doesn't exist. So I'm just going to create this one. I'm just going to frozen. Yeah, just going to right click here, new folder. It's going to be called out data. If I run this line, this cell again, actually, we don't get an error message anymore. So maybe things worked. So let's have a look. Let's inspect this folder. Yay, there is a new file. Ta-da! And if we open our regular file browser, navigate there, we can see it's actually, in my case, almost 18 megabytes in size. So that's 100 seconds of EEG data and an EEG channel. Did it work for everybody? Oh, there's more questions. Let's see. Okay, so there's a number of questions, so, or issues. So Anna had an issue where it says SVD failed to converge for MEG channels. Saskia says figures have still not appeared. They should. If they still haven't appeared, then there's something off. So are you saying you're getting blank figures popping up, or what's happening there, Saskia? They are blank. That seems peculiar to me. They're still loading. Did you try restarting the kernel? Actually, not sure what that could be. Which platform is that? Is that Windows or is that Mac? It's Windows. Anyone else on Windows having this issue? Blank figures showing up? Windows 10 or Windows 7? Or Windows 8, for that matter. Windows 10. Okay. Olaf had the same issue, right? This is super odd. What you can do, okay, what you can do is you can try insert a new cell and just type %sign matplotlib qt and see if that changes anything for you. Like have this maybe at the very top and restart the kernel, like restart and run all cells and see if that helps. Okay. I'm super sorry. Otherwise, I'm a bit clueless right now. Okay, but so now it's noon, right? Even in the UK. So I hope all or most of you actually got to the point where we actually saved the file. Is it working for you? Maybe time for a quick recap. So what did we do? We ran into lots of issues, obviously, but also we imported some raw data. We plotted this raw data. We worked with these stimulus channels and extracted experimental events / conditions. We counted these events, which can be super helpful, and to verify that you didn't, you don't have any spurious events, which sometimes happens. You end up with too many, or that you didn't lose any. We added these event markers to the raw plot. We looked at the infrastructure, which tells us a lot about the data at hand, for example, channel names or the types of channels involved. We learned how to actually plot channel or sensor locations. We marked channels as bad. And then we selected only a subset, EEG channels at first and later MEG channels, and only worked with those. We cropped the data and we applied a bandpass filter, visualized the results as PSD plots, and eventually we stored the pre-processed continuous data. I mean, even though we were interrupted quite a few times and started a bit late because there were a number of technical issues, I think we're off to a good start, and actually quite happy. We managed to work through the entire notebook. And now it's lunch break. After lunch, we will continue working with, it's a bit laggy here. I will give you a very brief introduction into bits and working with bits data. And if you've never heard of bits, I will be sure to attend because I will explain it to you. And then we will finally use the data and cut it into epochs and see which fancy things we can do with these epochs. So now, so we finished the first notebook. Woo! What a ride. And now we're going to start with the second one. It's called Working with Bits Data. So I would kindly ask you to open this notebook. If I remember correctly, and if I did everything right, it won't depend on anything that we did in the first notebook. It will depend on knowledge that we gained, obviously, but not on output files or anything we created there. So what is BITS anyway? What is this acronym standing for? BITS is short for Brain Imaging Data Structure, and it was, or it is, a specification that was originally invented to describe how to organize MRI data sets. You know, you acquire a bunch of data and then you give the files a name and you put these files into a folder, or maybe not. And then you have a certain folder structure and a naming scheme. And your colleague obviously uses a different one, and another lab uses one. And this makes it very difficult to interoperate and to write portable code, right? Or to have a portable analysis. Maybe somebody has written a great analysis script and a colleague of theirs thinks that they could make use of that. So they send them the script and they try to run it, and it won't work because the way the data is organized is so different and it's always painful. And I think we've all been in this situation. So BITS was invented to solve this issue, or at least to make it manageable. Let's put it that way. And it was more or less recently amended to also specify EEG and MEG data storage. So this makes it a good option for us to also use it when working with MEG and EEG data. Let me maybe quickly these Zoom controls are again blocking my view. Okay, there's a question in the chat. 1 second. It's just the installation instructions. Thank you, Joanne, for sharing the installation instructions again. So the BITS specification can be found at bits-specification.readthedocs.io. Not sure if you really want to read it because it's quite long and technical, but just wanted to point you there so you know where to find it should you ever need it. To interact with BITS data, we're going to use MNE bits. MNE bits is essentially it's a package, a collection of functions that under the hood enable MNE to read and write bits data. Okay, let's have a look at an example. I made this graphic here, and it's on the MNE Bits website. Oh, by the way, MNE Bits you can find if you go to mne.tools/MNE-bits. So let's assume that's the data you acquired. It's from one participant. It's MEG data. You give it a more or less random name, and these files here, MEG people may know those. This is a crosstalk file, and this is the fine calibration file for Elekta Neuromag Mag-in systems. It's just stored somewhere. And then we use MNE bits to convert this data set into a bits compatible data set. And what will be created more or less automatically for us is this. This is a folder structure with folders and files that are named and organized such that it follows the bit specification. So you have a participant, essentially a table of all participants in your study. The subject naming is predefined, so we always have a sub prefix followed by the subject name or number or ID. Then you can have multiple sessions. You can have multiple data types, as BITS calls it, MEC or EEG, or it could also be ANAT for anatomical scans or FUNC for functional scans. Could even be BHEF or something, or is it BEH? I don't remember, for behavioral data. And in here is the actual data, plus some, so this .fif file is the actual data here. And then there is lots of TSV and JSON files, which essentially contain metadata, participant age, handedness, and the like. And these here are the fine calibration and the crosstalk files in case of MEG. Okay, so why should you, in case it hasn't become clear why it's of advantage to go from here to there, let me just repeat that again. So if you have data that's organized according to the bit specification, you will be able to run tools that expect bits data on your data. One of those is, for example, the MNE study template, which is a collection of scripts essentially that we wrote, and it's gonna, you feed in bits organized raw data, and then it will epoch the data and filter it and run artifact correction and eventually run the source estimation. It's an entire pipeline. And building something like that requires us to know in advance how data is organized and how and where to find certain information about the data. And with bits, this is solved because all of that is specified. We can just look up in the specification. Okay, where can I find the sampling rate? Okay, the specification says, okay, in the following file, and then we can extract that. So this is for writing the MNE bits data, and for reading, we will also use MNE bits, and it will read the raw data files, it will read all these sidecar files with meta information and compile the respective MNE internal data structures accordingly. Okay, but it sounds probably very theoretical, so let's have a hands-on example. So this is notebook number 2 now. We're just running our imports. Almost trivial at this stage. There's a new one here, MNE bits, because we're going to use MNE bits. Is that working for everybody? Because if it's not working, you maybe forgot to install MNE bits. So, okay, it seems to be working. So we're going to read the input data, which is going to be our raw data. We're going to do exactly the same thing as we did previously. So we're going to find the location of the raw data, yeah, the sample data there. We're going to specify which file we intend to read. We're going to read it using MNE's read raw function. And we're also going to find the events. Okay, let's run this. Okay, looking good. No error messages at least. So one thing that bits requires is that the line frequency, the power line frequency, must be stored somewhere in the data. MNE stores that in the info line frec setting, and this value has not been set in the raw data we're using here. So we need to set it manually. Okay, and we know that the sample data was recorded in the US, and the power line frequency is 60 hertz there. So we're going to set this to 60. This is an exception here. I think in most cases I have seen with real data, like from an actual lab, this is already set correctly. But if we were to skip this step here, we would get an error message in the next cell. So we're just going to set this. We're just going to tell MNE, hey, the power line frequency in our lab where we recorded this data was 60 hertz. Trivial. Okay. Now, how do we write this raw data and the accompanying events to bits? Okay, so first thing, we need to define into which directory should that stuff go. So I here specify out data should have another subdirectory called sample bits. I think I need to create this directory manually. I'm not sure if it will be created automatically. Don't want to take any chances here, so I'm just going to create it. Okay, so now I have, so what I did was I went to this out data directory, right, selected new folder, and called this folder sample bit. Okay, then MNE bits need something which is called a bits path. A bits path is essentially an object that specifies the location of bits files. And to specify this location, you don't actually put actual path names or something here, but instead what you do is you specify what bits calls entities. You see, a bits file name, or essentially the bits folder and file naming structure, is based on these entities and their corresponding entity values. One of the entities is subject. This is just our participant's or subject's name or ID. Another entity that is optional, by the way, but which we're going to add here, is session. So we're just going to say here, it's going to be the first session. If you only have one session, you can actually skip that one. But just for the purpose of demonstrating how things work, we're just going to keep that. The entity task is not optional. You always have to specify it. And we did an audiovisual task here, so I'm just going to call it audiovisual. And another entity that we're going to use here is run. This is essentially a block or I think in EEG terms I would call it a block, but I guess MEG people would disagree. It's an acquisition run. This is also optional, but if you have multiple runs, you can use you can assign different run values. And the only time an actual path appears is when we specify the root. Bits calls it the bits root or just root. This is essentially the
the directory in which all of our bits data shall reside. Okay, so we create this bits path, and then we're going to call the MNE bits function, write raw bits, which takes as arguments the raw file or the raw data, the bits path, which is created here, and optionally the events and event IDs. If you don't specify those, no events or conditions, or however you call that, will be written. Events or triggers, not conditions, will be written if you don't specify that. So typically you do want to specify it. Okay, so I'm just going to run this line, this cell, and you can see it first opens a file, writes stuff, writes a README, and then there's lots of output here. Ooh, so much output. So much output. Okay, now maybe let's explore the output directory and see what happened. So this is the output directory I created. And all of this content here is what was just created using write raw bits. Look at all of this. Quite a bunch of files. Actually, I think we can also explore them from inside the Jupyter lab. So I'm just going to go here. Let's maybe have a look at the README. Just add some references to MNE bits. And, oh, there's a question. If you get the attribute error, MNE bits has no attribute bits path, it means your MNE bits is too old. You need to update it. You need to do, just gonna type this here. You need to run, actually I think if you prepend it with an exclamation mark, you can actually run it in the console. Let's see if that works. Yes. Does that help you, Pia? But this shouldn't happen. So, okay, what you can do is you can do import MNE bits, print, and do MNE bits __ version __. What do you see? MNE bits on Conda Forge should actually be up to date, but yes, do install it via pip. You should have version 0.5. That's the latest. Yes, you need to have 0.5. Okay, Pia, can you try to update? Okay, I'm just gonna continue for now. I'll delete that. Where were we? Ah, yeah. The README currently only consists of some references to MEG bits and I think MNE bits. So that's not really interesting. But here is, for example, participants.tsv. Let's have a look at this one. Computer is a bit slow. Okay, so here, I mean, it's not super fancy now because it only contains one participant without much of any info. But we do get a table of our participants. And normally you would also get some metadata if the data is not anonymized. We'll get some metadata, age, sex, and handedness. Another question in the chat. Okay, it works now for Pia. Great, great. Okay, let's look into participant or subject 1's data. So we're going to go to subject 01, session 01, MEG. You can see the file names are super long. But here, there's two interesting files. One is called subject1 blah, 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 _ channels.tsv. Let's open that one. Just double click and it typically takes a while until my computer actually shows something. So, and as the name of that file suggests, what we can see here is an overview of all the channels that are in the data we just converted. So this is the channel name. This is the type of the channel. So these are gradiometers, planar gradiometers, and magnetometers. If we scroll further down, we should also see the
EEG data. Yeah, there's EEG data. As you can tell, I'm more an EEG person, so I like them more. That's my preference. We see which unit is used in case of EEG. This is saved in volts, which is interesting to know, right? Because I mean, typically EEG signals are in the magnitude of microvolts, but here we store everything as volts. This is what MNE does by default, whereas for the magnetometers, it's Tesla. We see filter cutoff, so there was a low pass filter applied. This was applied during recording already. 172 hertz and a high pass of 0.1 hertz. Get some more information on these channels and sampling frequency, obviously. And also, and that's important, channel status. Is it good or is it bad? So there was a bad EEG channel, if I remember correctly. So let's see if there it is. It was, I think, channel number 50, yeah, 53. 53, maybe you remember it from our first session. 53 was marked as bad, and it is automatically marked as bad here as well. Status description can be used to specify why a channel was marked as bad. But since MNE, or we don't know why this channel originally was marked as bad, and we didn't specify any further information, it just says NA here, meaning there is no info. Okay, so we can close that. Let's have a look at this other interesting file. It's called sub01, lots of stuff, underscore events.tsv. Again, waiting for my computer not to die. Ah, there it is. So, as the name suggests, again, this is now the events, the experimental events. Okay. So, what can we see here? The onset in seconds, the duration. Since these are instantaneous events, their duration is 0, obviously. We get info about which kind of event or trial type was that. So this is essentially the mapping we created here. Yeah, this event ID. The value is just the trigger code, if you will, and we also get the sample number. So not only the onset in seconds, but also which sample number was it. And I think we had 320 events. So let's see if we have 320 rows here. Yes, we do. Perfect. So when we see the last event was, cannot move the zoom controls out of the way. Ah, no. The last event was a button press at about 238. Yay. Okay. So yeah, and these other files we're just gonna skip for now. Oh, we have our first bits data set. Isn't that great? Let's scroll further down here. So in case we're working with MEG data from an Elekta, Neuromag, or there's a question. 1 second. Yes, hooray. Yeah, hooray. Yes. Thank you. There was no question. Thanks. I like celebrating with you. Okay, so if your data were EEG data, you would be set by now. If your data is MEG data acquired on a Mag-in, Elekta, or Neuromag, or whichever name they have these days. I mean, this company gets bought and sold off so often. If it's data acquired in a system of these, you will also want to store the fine calibration and the crosstalk files. For our data, for our sample data, the fine calibration and crosstalk files are stored in these locations here. I can briefly show this to you. So I lost, there. Couldn't find my own name there. Okay, so I go to MNE data and then it's the MNE sample data. And there is a subfolder SSS, and here a crosstalk file and the fine calibration file. And we want to tell MNE bits, hey, please store these files along with the data of our participant. Okay, and we're just going to do that. So we specify the paths, and then we're going to use MNE bits.writeMac calibration and MNE bits.writeMac crosstalk. So super simple. So we're going to write those, and yeah, just took a second to refresh. And here now in the participants folder, we can see we have 2 new files with the correct bits naming. Okay, so just to recap, this file was originally called sss_cal_mgh.dat, and now it's called sub-01_ses-01_acquisition-calibration. It's difficult to actually read those file names, but you can see it was mapped to the correct bits file name. Perfect. Okay, so now what I would like you to do is I would like you to essentially repeat what we just did, but, I'm scrolling up, but act as if the raw data we have here was from participant number 2, or with a different name than 01. So that we end up with another participant in here. Okay? And drop me a line when you're done. I mean, it's kind of trivial, but if you're running into issues, just ask me. Okay, is it working for all of you? I'm just going to show how I would do it. I would simply go up here to the cell. I would change subject to 02. Okay, so then just gonna run this and gonna, oh my goodness, so much output. And and gonna run this, verify that everything is in place, and yes, it is. Okay, so MNE bits has a convenience function that prints out the directory tree, and the function is it's just called print_dir tree, and we call it on our what we also call bits root directory. It's the output directory we created here, and let's see what happens there. And you can see this is a nice and helpful, I was about to say visualization. Well, it's it's not super visual, but it's a way to, at one glance, quickly see how the data is structured. You can see there's also these annoying .ds_store files, which the Mac automatically creates, and they end up everywhere, and you can't get rid of them. Yeah. Okay, so now we have 2 participants. If we look into participants.tsv, again, we have 2 participants now. Perfect. The latest release of MNE bits has added a nice functionality. The idea was, if we know so much about the processing or the acquisition of your data, and how many participants are involved, and how many sessions there were, and how many runs, and the duration of the recordings, etc., can we not simply automatically generate a section for the methods paragraph of your next Nature paper? And that's what we worked on. And the function is called mnebits.makeReport. And again, as input value, it needs the path to your bits directory. And it generates this. Oh, there's a question. Okay, how can we change the age, etc.? Okay, we will look into this in a second. I did not mention that so far. Okay, but so it generates the summary, and it still has more than just a few rough edges, but it's a start. And so it tells you which bits version that is, which MNE bits version was used, how many participants, handedness, etc. This is all unknown here, and sampling frequency and all of that stuff. So it's super neat. So standard deviations here are always 0 because we wrote the same data twice, right? Okay, yeah, good question. So how do we actually add handedness, age, and sex? I totally forgot that when preparing this workshop. So it's good up. So the easiest thing you could do, obviously, is you could open the participants.tsv and you could just change that. Okay, but it's error prone, and when you run the script again, the data will disappear again. So what you typically do is, scrolling all the way up, what you typically do is you need to add this info to the info structure. Let's have a look at the info structure. Before, and I'm talking about the infrastructure of the raw data, before writing it to bits, okay? So let's have a look at the info structure. And there are so many things going on here that I, again, cannot find what I'm looking for. No, I think it's not even there. Okay, I will have to consult the documentation, and you can see how I do that. So I go to MNE.tools, and I want some info on info. Let's see what MNE tells me. And here there's a link, the info data structure. Maybe that helps me. See if I can find what I'm looking for. Looking for something that's related to subject. Not here. Okay. So the info or documentation is sometimes spread out across different places. I think here's everything we need. Janets, that's not what we want. Comps, not what we want. Dig is not what we want. HBR result is not what we want. Subject info, that's what we want to add. Okay, we can add a participant ID, last name, first name, we don't care about that. We can add a birthday, sex, and handedness. So let's try to do that. I'm not so sure about the birthday format, honestly. I would have to try. But sex seems easy, male and female and unknown. Just so you know, there were some discussions on adding more options there. And the only, also in the wake of current cultural debates and the like, the only reason why there is still only 2 sex options plus unknown is there is issues with backward compatibility in the data format. So it's not so easy to add something like inter or trans or whatever. Yeah, but there's people looking into that, so maybe we will get that. And for handedness, we have left, right, and ambidextrous. Okay, so let's try to add sex, handedness, and birthday for our participant. So we need to create a dictionary. It's here. It's a dictionary called subject info, and it needs to have a few keys. One is going to be birthday, one is going to be sex, and one is going to be hand. Birthday is going to be in year, month, day format, a tuple of int. A tuple birthday. Let me change the indentation a little bit. Birthday. A tuple is what you create when you use these just parents and then put stuff with commas inside. Okay, how was that? I've already forgot. It was year, month, day. I've never done that. So year, I don't know, 1998, month, October day first. Not sure why this is showing up as red now. Ah, probably can be skipped. Okay, birthday. Next, sex. Let's set it to female maybe. See, it's an integer and it should be 2 for female. And handedness is just called hand. And let's make it left-handed. It's 2. Or let's make it ambidextrous. It's a 3. And 3. Okay. And what we want to do now is we want to assign this subject info to our to the correct slot, if you will, of our infrastructure. So that's raw.info. Checked. I'm doing things correctly here. Subject info. I'm going to set this to info. And then we're going to print out the info. Let's see if that works. Okay. Did everybody follow until here? So just repeat. We created a dictionary with participant information. So by the way, I suppose this info has just been removed from this data set for reasons of anonymization. I think these systems typically do record this information and store it with them. But yeah, I mean, you might always encounter files and acquisition systems that create files that do not contain this info, so you have to add it manually later on. Okay, it's working. Okay, so we added that, and as you can see here, subject info shows up down here. Okay, so now let's try to write this again for subject 01. So I'm just writing the bits data again. And let's have a look at participants.tsv once more. Age 4. I mean, okay, there's something wrong with the age. But I mean, sex and handedness works. I have no idea what's wrong with age, honestly. What did I do wrong? Does anybody know? Year, month, day. I mean, the only thing I could imagine is that maybe it has to be without the 19. Oh yeah, right, obviously. Yeah, okay, so the recording date was in 2002, and if the birthday is in 1998, of course, the participant is only 4 years old. Okay, it doesn't make too much sense. So I'm going to change this to 1988 now. And it says 4. Did I do something wrong? No, 14. It's just 14. Thank you, Stefan. Okay, so this is how you can add this meta info to MNE bits, to your bits data using MNE bits. Oftentimes, we want to go the opposite way. We want to remove this info, and there is an option for that when writing the bits data. Here, write raw bits. There is an option called anonymize, and anonymize allows you to shift the recording date and remove information like, I don't want to give you any false info because there was a debate on which information is actually anonymizing information. But I think it removes birth dates as well, and maybe sex, but I'm not entirely sure. Yeah, but I don't want to go into details there. There is documentation on that on the MNE Bits website. So if you go to the MNE Bits website and click on Use, and then navigate down here to examples, there's lots of info on how to actually work with MNE Bits. And I think there was something regarding anonymization. Cannot find it now. It's not here. Okay. In that case, I just want to point you to the API documentation. And here we have write raw bits. And here it tells us about the anonymization option. Okay. This is what we will typically use. So this stays back, like I said, means that it will shift the date. So if you have multiple participants, for example, or same participant and multiple recordings, you do want to shift their recording dates by the same number of dates to keep the relative difference in recording dates without revealing the actual date. Okay, but maybe that's something we can have a brief look at later when we're going to talk about group analysis. Okay, everybody happy so far? So let's go further down here again. If we now create the summary, there should be some more info. 2 participants, 0 men, 1 woman. Obviously, it doesn't tell us that 1 participant was unspecified. 1 ambidextrous, and yeah, you get all the info you need, hopefully. Okay, so now we have written bits data. Let's read it back in. To read it back in, again, I'm just repeating myself here. We need this bits root thing. So just gonna point it to the root folder of our bits data. In our case, this is out data /sample_bits. And we create this bits path object again. So we just tell it which subject, session, task, etc. And data type, this is important. So the data type is always, so if you go into a session folder, and then there is always a folder like MEG, or if you have EEG data, it will be EEG. If you have simultaneous recordings of MEG and EEG, it will also be MEG, which is the case here. That's the data type. So in our case, that's MEG and the bits root. Okay, so now let's read that. And we can see it opens the correct file. Luckily, we didn't have to construct this file name manually because it would have been painful. And it also reads the events and channel info. We get this warning. This is related to something that happens with the stimulus channels, but this can be ignored. Okay, just ignore that. Okay, and let's visualize the raw data we just imported. Doesn't look too bad, does it? So we have our gradiometers up here. We have our magnetometers. We have our EEG data, yay. And we have our trigger channels or stimulus channels. Perfect. We also have at the top here labels for the different events, the different experimental events, and there's vertical lines. But you may notice that they look different than they used to look in our before lunch session, where all vertical lines were in, I don't know, turquoise or something. Whereas now everything is so colorful down here. What's going on? Well, it's very easy. If we toggle annotation mode by pressing A, we can see that there's lots of annotations going on here. So what happened? When MNE bits reads data and encounters any events, it will add these events as annotations to our data. So what we have now is, thinking back of before lunch, we had our raw data, and then to get to our events, we called MNE.findEvents. And this would extract the events from the events channel. Whereas now, if we're reading data with MNE bits, we don't get any events immediately, but instead, MNE bits will parse the events GSV file and add all events that it finds as annotations to the raw file. Okay, that's why we end up with so many annotations here. That's an implementation detail that hopefully isn't too confusing because it comes with a number of advantages. Okay, so just keep in mind these are annotations. Let's have a look at the first annotation. So all annotations are stored in raw.annotations. We look at the first annotation. It will tell us, okay, this annotation has a certain onset, a certain duration, which is 0. A description, that's a description we just saw in the raw plot. And then this ORIC time thing that we can ignore for now. And we have one of these for each individual event in our data. Let's look at the last one. So we're just picking via -1. So that happens at 280 seconds and it's a button press. Sometimes, meaning in the next notebook, we will need events to do what we want to do. For example, if we want to cut the data into epochs, MNE expects us to pass events. It won't just work with annotations, which, by the way, I personally think is something we should change. But it's an ongoing discussion. So this just to say that sometimes we may need events, but how do we get events if all that MNE bits provides us is a raw data structure with annotations? Well, it's easy. We got your back there because MNE comes with a function, events from annotations. Events from annotations. Oh, by the way, this is something I wanted to show you. So if you are unsure about what a function does and how it works, I mean, obviously you can add these parentheses, and then you can press Shift and Tab. Sometimes it works, and right now it doesn't. Oh yeah, opening parentheses, Shift and Tab, and then you get this small window, and it's a bit painful to scroll around. What you can also do is you can just type the function name followed by a question mark and press Shift Return, and then you get the entire documentation. It's not as neatly rendered as on the MNE website, but it comes in handy once in a while. Okay, so we have this function raw from annotations. Okay, which input does it need? Okay, it needs raw, obviously. Event ID, okay. And then the rest we can probably skip. And it returns events and event ID. Okay, so it returns event ID and it also accepts event ID. What the heck is that? Oh, it seems we can just skip that one. Because that's the default. This is just to select a subset or to change the mapping. Okay, so we need to pass raw data and it will return events and event ID. Let's just close that. And let's run this. So it's telling us it used the annotation descriptions. You see, each annotation has a description, and it used auditory left, auditory right, button, smiley, visual left, visual right. These are actually all the event names we initially specified before creating the bits data. And that's all the events we saw when we plotted the raw data. So everything seems to be here. We can, I'm just going to insert a new cell here. We can again look at the events object that we got back. So it's this NumPy array again, big NumPy array with 3 columns. I can maybe get some info about its shape. 320 events. Perfect. None has gotten lost. That's great. Now I want to show you a convenient way to graphically get an impression of how many and in which order events occurred throughout the experiment. And it's a function underneath the vis module, and it's called plot events. And it just accepts as the first argument the events array, the one we just looked at, the event ID dictionary. So we got this back up here, okay? And it needs the sample, the sampling rate. Otherwise, so if we specify the sampling rate, we would get the time axis in seconds. Otherwise, the time axis would be in samples. But in seconds, it's easier for us to read. So the sampling frequency we can get from raw.info sfrac. Okay, enough talk. Let's have a look. So we get this figure. It's a bit cramped, but you get an overview. So you see, okay, there's 72 auditory left and 73 auditory right events. And you can see the order here of these event names corresponds to the order on the y-axis. So this here's auditory left, auditory right. Again, I have to say we have been discussing enhancing this figure. I guess it can use a bit of love, but I think it can be quite helpful. So you see that everything is kind of evenly distributed. Perfect. That's a valuable figure for inspection and sanity checks of your thing. Close that. Okay, and the last thing. So remember, we also saved these fine calibration and crosstalk files. So assume you need those. For example, because you want to conduct some sort of Maxwell filtering. Oh, by the way, we have skipped Maxwell filtering so far. If there is any demand to show you how that works, we can certainly squeeze that in somehow. Just drop me a line. Yeah, if you need to know where these files are located, you can just use the bits path object. You know, the same we used for reading our file. Where is it? Up here. It specifies which participant and task and session, etc. And it has an attribute or a property. One is called MEC calibration, F path, and the other is called MEC crosstalk, F. And it just tells you the path to the fine calibration and the path to the crosstalk for further processing over for whatever you may need to do with it. Okay, are there any questions until here? Something popping up? Mm-hmm. Yeah, so I just got a question on max filter, on how to fix bad channels and basic usage of max filter. We need to see how it works out with our schedule to squeeze that in. Otherwise, I have just the tutorial you're going to need. Where is it? This one. So, okay, let me show you how to find it. If you go to the MNE website, mne.tools, click on tutorials, scroll down, scroll down, scroll down. No, I cannot find it anymore. Ah, here it is. Scroll down to pre-processing. And the previous to last tutorial, signal space separation, SSS, and Maxwell filtering. This actually has all the info you need to perform Maxwell filtering. It's quite a bunch of code, but do not despair. Most of this here is really just for plotting. I wrote this a while ago. So this is first applies an automated method for detection of bad channels in MEG, and later we do Maxwell filtering and TSSS. Okay, but if we have some time, maybe tomorrow morning or so, I can also give a quick demo of that. Okay, so we're done with this one. Yay! I'm gonna close that. And we can move on to notebook number 3. Does everybody have the notebook open, number 3? So far, we've only worked with continuous data. We've loaded it, we've filtered it, we've cropped it, we've saved it, we've extracted events, we've converted it to bits, we've read it back from bits. That's all great, but well, I typically have to deal with evoked designs, and there we have to create epochs, meaning we have to slice the continuous data into quite a bunch of small time segments, typically just a second or 2 seconds or so long, and we analyze those. And the next part of the tutorial is going to focus on just that. We're going to create epochs, and eventually we're going to average these epochs to create evoked responses, meaning ERPs in EEG, and ERFs, evoked. Oh my goodness, what is it called? Is it called evoked response fields? Evoked responsive fields? Let's see what the chat says. Evoked fields, just evoked fields? Event related, of course. Sorry, yeah. Event related potentials in EEG and event related fields in MEG. Oh, how embarrassing. Okay, so let's get started. I'm gonna run the first, that's trivial, right? You already know that. Gonna import what we need. Now we're going to work with the bits data we just created. Okay, so I hope, that's why I was asking repeatedly whether you could successfully run the previous notebook. So we want to use the bits data now. So we're going to specify that input folder again, and the participant info. Sorry, not the participant info, the info of the recording. Okay, so we're going to read that. We're going to load the data. You remember we cannot filter without preloading the data. And then we're going to extract the events from the annotations as we did before. So this is essentially just this is more or less a summary of the first 2 notebooks, if you will. Okay, let's see if that works. I think, oh no, it's still busy. Okay, so we created a bunch of new variables. We have raw, we have events, we have event ID. Let's have a look at event ID. Seems to make sense. Okay, and now, actually, I just realized I forgot to insert some more explanatory comments here. But I'm just going to explain to you right now. Okay, so what we have now is we have the raw data and we have the events and description of these events. To create epochs, we need a few things. We need to specify where does the epoch start relative to an event onset? Where does the epoch end relative to an event onset? And also, should we apply some form of baseline correction? Okay, where do we specify? How do we specify where the epoch starts? It's just a parameter that's going to be called tmin. We specify time in seconds, right? It's important. It's seconds, not milliseconds. This has caused quite some headaches for me when I started working with MNE. So in this case, we will start the epoch 300 milliseconds before the event. Tmax is the end of the event in seconds, end of the epoch in seconds after event onset. Oh my goodness, I'm twisting my tongue here. So this is going to be half a second. And baseline, this is what do we consider a baseline period if we want to have any at all. And it's essentially a tuple that consists of two values. The first one is the start of the baseline, and the second one is the end of the baseline. So in this example, the end of the baseline, I mean, it's clear. That's time point 0, that's the event onset. None doesn't really make much sense at the first glance, but it has a special meaning to MNE. None means beginning of the epoch, from the beginning of the epoch. So this baseline means the baseline period extends from the very beginning of the epoch to time point 0, meaning the event onset. Okay. To actually create these epochs, then, we use the MNE.epochs object. So I just placed my mouse, my cursor here, and pressed Shift Tab to get an impression of all the parameters it takes. As you can see, we're skipping most of them. Okay, so we're going to pass the raw data we intend to cut into epochs. We're going to we have to tell MNE which events shall be used to actually cut these epochs, right? Because epochs, epoch durations, epochs beginnings, epochs end are always relative to a certain event. So we're going to pass in the events NumPy array. We're also going to pass in the events dictionary. This will be used to assign human readable names to the epochs. Tmin, Tmax we already talked about, and the baseline we also talked about. So if we pass a baseline here, this means that MNE will automatically apply baseline correction. We could also opt not to pass a baseline or pass baseline equals none. Then MNE will not apply any baseline correction, and we can do that later on. There might be situations where you want that, but most of my analysis, I don't need that. So I would ask MNE to perform baseline correction on epoch creation. And we pass, in this case, preload equals true. This will ensure that the data gets loaded into memory. Okay, finally, let's run this. Computer takes a nap. Okay, so it tells us it has found 320 events. That's exactly what we expected. Sorry. It applied baseline correction. By default, it uses the mean. I mean, typically you don't want anything else unless you have very special needs, so that's okay. It talks about this SSP thing, which when I was still working with EEG data only, I had no idea what the heck that is. So if you're an EEG person, just ignore. If you're an MEG person, you probably know what it is. We will talk about this later. And yeah, eventually it tells us, okay, we have created an epoch with 320 events. Epochs extend from minus about 0.3 seconds to about 0.5 seconds. There is a baseline from the start of the epoch until time point 0. And here we get an overview of how many epochs have been created. Okay, enough talk. Now finally, let's look at some nice visualizations. So we're going to run epochs.plot. And my computer is struggling. What? Help. Okay, I think we got there. Okay, what you can see here is down on the x-axis and here, that's the epoch numbers starting at 0. MNE starts counting at 0, like Python does. Can be confusing at times, but it's better to be consistent, so everything starts with 0. up here, you can see the event codes. Now, I personally would prefer, something is, right, I cannot properly interact with this. 1 second. Ah, okay. Now, I personally would prefer if it were to say the experimental event name and not the number, but for whatever reason, it only displays the event number here. Let's have a look at the help. Okay, we can, for example, press B to get the butterfly. We see some things are seem to be a bit off the charts. Going to press B again. Let's change the time scaling factor. So on my Mac, I'm going to hold down function. I'm going to press the left arrow key a few times to make the epochs wider. Okay, that's good. And so assume we encounter an epoch of which we think that looks really, really bad. I'm not sure if I can find any here now. Let's just assume this one, epoch 66, just why not? And we think, okay, we should exclude that from our analysis. We just click there and it's gone, okay? It's marked as bad now. If we scroll further down, we can see that this expands beyond all channel types, or across all channel types. Okay, and if we close that again, this one too, we get some output here, dropped to epochs, and channels marked, I mean, these channels were marked as bad previously, but the important info here is that it dropped to epochs. Interestingly, these are gone now, okay? Like you see here, it says 318 events, where we used to have 320 before. So this is maybe something important to mentally note. When you browse raw data and mark a channel as bad, this info is then stored inside the raw data. And if we mark epochs as bad, then these epochs, as soon as we close the plot, these epochs are actually removed from our data. Okay, so if you play around and just click at random epochs like I did now, and then close the plot, and then just continue processing as if nothing happened, you might end up with undesirable results because you just removed some random bits of data. Okay. So now I have a small exercise for you. I want you to create epochs from our raw data, from the same raw data we just used. I want you to create epochs that start 200 milliseconds before stimulus onset and end 800 milliseconds after stimulus onset. And I want you to apply baseline correction, but not for the entire range of 250 milliseconds to 0, but from minus 200 milliseconds to 0. I'm just clicking on Zoom because there is a request. Hamid is in the waiting room and I cannot admit him. Oh, can I let him in? Johan or Olaf? I'm trying to admit Hamid to the chat. Ah, it works now. Okay. Okay, is the exercise clear? There is a question. Juliet, yes, this seems like your laptop has too little memory. Doesn't have enough RAM. So it could be that, do you have lots of other software open? Maybe close stuff. If you have lots of browser tabs open, maybe close those. If you have like Word or Excel open, maybe close it. There seems to be a lack of RAM in your computer. Okay, yeah. If that doesn't help, maybe try restarting the computer and starting up the Jupyter Notebook and only the Jupyter Notebook and just running it again, then it should work, hopefully. Yeah, Juliet was experiencing some memory issues. Okay, so let's see. So what we wanted to do is we wanted to start at 250 milliseconds before stimulus onset. We wanted to continue until 800 milliseconds after stimulus onset. And now we didn't want to take the entire negative time range as baseline, but we wanted to conduct baseline correction from with the baseline period of -200 to 0 milliseconds. So just gonna add -0.2 here and gonna run this again. And did everybody get so far? There's another question. What does not setting metadata mean? Not setting metadata, actually, I'm not sure why this message is emitted. MNE supports having metadata attached to your epochs, or maybe even to raw data. I'm actually not sure. So I personally have never used it. I do know that a colleague of mine does use it. It can be used to store additional info about your experiment, like, I guess, certain info about responses, or I would actually have to look it up. Is anyone here using metadata in MNE? The simple answer is, for now, you can just ignore this error message. And let me quickly look this up. MNE metadata. Aha. Okay, so it's an epoch thing. Yeah, right. So you can just assign additional metadata to your epochs. How that works in particular, I cannot tell you right now. It's all right here. I haven't done that myself. There was no need to. Okay, does that answer your question? Okay, perfect. Where were we? Oh yeah, the exercise. Did you manage to finish the exercise? Does it work? I mean, I essentially gave you the solution up here, right? So I just assume that it worked for all of you. And this was just to demonstrate, oh, look at this. I made a mistake. Huh. No one told me. Unbelievable. So let's see what happens now. I can only emphasize again, time is specified in seconds with MNE. Yeah, and this was just to demonstrate that baseline, or specifying a baseline period of MNE, is not bound to, it's not necessarily bound to the entire period before time point 0. It can also be a subset. It could also be, so here it's from -0.2 seconds to onset of the stimulus. I also had a paradigm once where we used, I think, -700 to -500 milliseconds as the baseline period. Okay, so this can come in handy. Okay, so now we have these epochs, and they have numbers, experimental conditions. How many? 1, 2. Sometimes you just want to work with one of those experimental conditions. You know, you want to have only the epochs for auditory right or auditory left or whatever. And you can actually very easily select only a subset of these epochs by passing a name of the condition in brackets. So if you want to have only the auditory right epochs, we just do epochs, auditory right, and there we go. We get a new epochs object, 73 events, all of them being auditory right. This is neat, but it can get even better. If you look up here, you will see that, or maybe you still remember, that we not only have auditory right, but we also have auditory left. What if you want to analyze all auditory epochs in one go and do not want to differentiate between auditory left and auditory right? MNE introduces a syntax here when it comes to event naming or condition naming, it allows to include a forward slash. You see here, we have visual left, visual right, auditory left, auditory right, always separated by a slash. And this allows for grouping operations. What we can do here, for example, is we can select all auditory epochs. Let's just do it by just passing auditory. And it will automatically select all auditory left and all auditory right epochs. Okay? We can also, so this was now the left, so what was left of the forward slash. But we can also pick what's right of the forward slash. So we can say we want all epochs that were associated with left stimulation, left side stimulation. So essentially we want to have auditory left and visual left. So we just subset by passing left, and we end up with auditory left and visual left. Isn't that cool? So now, and let's have a look at a visualization. I think it's called an ERP image in EEGLAB of all of our visual epochs. Just going to run this. Taking quite a while. This is because we have 3 channel types involved up until now: magnetometers, gradiometers, EEG. So we're getting 3 figures now. So this is for EEG, for magnetometers, and this is for gradiometers. I just wanted to show you that these images, these figures can be created. Stuff looks kind of noisy because we still haven't applied any proper artifact rejection here. But so you get the GFP, and on the x-axis there's time, and on the y-axis there's the epoch number. Okay, from 0 to the max. Okay, just gonna close this. I just wanted to show you that we can create this kind of figure. Okay, and now I would like to ask you to first extract all epochs from the auditory, from the right condition, meaning we want auditory right and visual right. I cannot hit the correct line while selecting auditory right and visual right. And I want you to create an ERP image, like the one we create with plot image. Oh, and maybe to make it even more difficult, let's do that just for EEG. Okay? Extract all epochs with the right condition and plot the ERP image for the EEG channels. The baseline isn't 0 in these figures because it's the GFP. That would be my explanation. Just gonna grab myself a glass of water. I'll be right back. Stefan, does that seem like a reasonable explanation? So Stefan asked, why is the baseline not 0 in these figures? Yeah, it's because it's the GFP here and not the ERP that you can see at the bottom. Unless I'm mistaken, I'll be happily corrected. I too make mistakes. Okay, Stefan said he didn't pay attention to the image descriptions. Okay, let's see if we can solve this exercise. So we're going to use the right epochs. We can use the same function as we, at least I hope we can use this. I haven't tried that. We should be able to use the same function as we used with the raw data or continuous data. We're going to use pick type, MEG equals false, and EEG equals true. plot image. Oh, and we want to have a copy of that. Okay, and you can see this gets super long here. This is a bit annoying. So what I typically do is I put this into parentheses and I split commands like this across multiple lines. This way it's easier to read. Maybe even like this. Okay, bear with me while my computer explodes. Okay, okay, damn it. I should have tried that first. Okay, let's have a look at the documentation. Maybe plot image. Pick types. Pick types. Oh yeah, and Pia has a different solution. She just posted it to the chat. So let me first. Thank you, Iona. What a typo. So let's see. Yay. Okay. But maybe another comment, another remark again. So I mean, look at the activity in the baseline period. The GFP is super high. This also shows that there is still something off with the data. It's just too noisy. Okay, we need to apply some sort of artifact correction or rejection. Okay, but this was just for demonstration. Okay, and Pia suggested we can use this command. So apparently plot image accepts a pix parameter, which should yield similar results. Yeah. Okay. By the way, I think if you use plot image with pix, you don't need to create a copy first, because I think plot image will internally create a copy. Yeah, that's just a minor detail. Okay, so we can save these epochs using the common way. We just enter the name of the variable, in this case epochs, and we call the function save. And the first parameter is going to be the output file name, and followed again by override true in case we want to run this multiple times. So let's save it. Takes a bit. Oh, by the way, so I always use _epo for epochs and _raw or _MEG for raw data. And the reason for that is that MNE would actually complain if you use different names. So if I were to use epo, but it would call it, I don't know, _xxx, try to write it, MNE would tell me, hey, doesn't conform to the MNE naming conventions. And it tells you what you probably should use. It suggests to use a dash followed by a certain suffix, but I prefer an underscore, which is accepted as well. Okay, so now we had epochs. Maybe it's time to look at evoked data. Evoked data is really just an average of epochs. And now that we know how to subset or select a subset of epochs, we can actually create evoked, in this case, in this example, for auditory and for visual stimulation. Okay, so this here selects all auditory stimulation, left and right, and this here selects all visual stimulation, left and right. If we run that, average them both, we will have some evoked. We can actually get some more info if we just type the name here and run that, and it will tell us, okay, so that's essentially the name that MNE gave this evoked object internally. It just shows us that certain weights were used for combining auditory left and auditory right. In this case, equal weights. There is actually ways to tell MNE to use different weights, but that's that's advanced stuff. We don't need that for now. It tells us how many epochs were actually averaged, and it tells us the duration, how many channels, and the size. Okay, so let's plot these epochs. We're going to use these evokeds, I'm sorry. We're going to use the evoked name, and we're going to run the function .plot. And we also want to pass a parameter which is called spatial colors equals true, which will make a colorful plot. So why did we pass this spatial colors equals true? Because it tells us through color coding of the traces the location of the respective sensor or channel. So green is more like, you know, frontal left. You can see so these traces here must be more frontal left, whereas these must be more right, and the like. So it can make it easier to figure out where certain activity takes place spatially. What we can do is also, we can, again, click and drag, and then wait, and then it will create topographic maps of the averages, or of the average activation of the selected time period. Now, if you ask me, there is a bug here because the MEG topo maps should look different. But yeah, okay, that probably needs to be fixed. Or maybe it has been fixed and will look better in the next version, but this looks a bit odd to me. So I would expect to see the MEG helmet here, but it's just a circle, which doesn't make too much sense. Okay, so what I selected here is essentially the region of the peak, or of the first big peak, and we can see that there is a strong negativity in the frontal electrodes on the EEG, and we can see there must be some sort of dipole here on the gradiometers, and we can also see it on the magnetometers. So, and this was clearly auditory stimulation. I mean, okay, but what we can also do is we can ask MNE to produce a topo map. By default, it picks 4. I thought it's 5. Well, it picks a number of time points and plots a topo map at these time points. And now we get the correct shape. You see, that's a gradiometer, magnetometer, and we can see this the shape of the MEG helmet here. Okay, and so you can see how activation changed over time. Well, actually, you cannot see it so much here because the time points are so sparse, but you get the idea. You can actually, tell MNE which time points to pick. So you could also go like times equals, let's say 0.050, 0.1. No, wait, 50 milliseconds, 100 milliseconds, 150 milliseconds, 200 milliseconds, just like that. Let's see if that changes anything. Yeah. See, now we have our custom spacing and also a different number of topographic maps, 5 instead of 4. We can also create a so-called joint plot, which is a combination of the traces and the topo maps, and MNE will automatically try to detect the interesting portions of the data, which is the peaks of the GFP. Here, I don't know. So this one totally makes sense, but these, I'm not sure if there's actually any meaning to them. But but yeah, that's a nice visualization. And again, here one can also specify at which time points to place the topographic maps. I think that's nice to at least get an idea of the data. Okay, so here, click and drag also brings up a topo map. All clear up until here? Seems so. Okay. And the last thing I wanted to show you is there is an easy way to plot GFPs of different conditions to help you contrast them. Well, maybe not contrast them in the sense that we actually calculate a contrast, but to do some visual inference, as I like to call it, which is by using the function this.plot_compare evoked, and it accepts as its first argument a list of evoked. So we have two here. We have the auditory evoked and the visual evoked. And then you can also select which channel types, which sensor types to include. And here I'm going to confine myself to magnetometers. So let's run this one. And again, that's one of the plots that could be prettier, but you can already see how this might be helpful. Okay, so we have the GFPs of both conditions and superimposed, and you can see that for both visual and auditory during the baseline period, meaning before stimulus onset, the GFP is, I mean, there is quite something going on there, clearly, looking at these numbers, but it is rather constant, and after stimulus onset, it goes up, okay? And it goes up quicker and much higher for the visual stimulation than for the auditory stimulation than for the visual stimulation. And then after a while, it goes down again, but it doesn't return to the baseline level, which probably is due to the fact that we just end here. You know, if the epoch was longer and would go until, I don't know, 2 seconds or so, we would see how the signal goes down again. At least that's what I would expect to happen. Okay, and I would like you to visualize, like to make the same plot as I did now for the GFPs, comparing the GFPs, and I would like you to compare the visual left and visual right conditions, but only of EEG data. Can we do that? I think you can do it. I'm just gonna wait a minute or two. And if there's any issues, do let me know, please. So hint, you need to create 2 new evokeds, okay? One for visual left, one for visual right. And then you need to call plot compare evokeds and select the EEG sensors. Okay, anyone having difficulties, experiencing issues, or is it working for all of you? Do you know what to do? Did you get it done? Or is it totally unclear and you have no clue where to start? Don't be shy, just drop me a message. Okay, I would say then we just move on, if that's all right with you. So MNE can also save evoked data. I rarely do that personally because it's so cheap to just regenerate that from epochs. But what you would use to save evoked data is just the function write evoked, and you pass again the file name, the output file name, and then you pass a list of the evoked you wish to write. So MNE then doesn't write just one evoked, but it will write multiple evoked. So let's just run that. And to read back these evoked, we use, well, who would have thought, MNE.read evoked, and it uses it accepts as parameter the file name again. So now we get quite a bit of output here, and we get back a list of 2 evoked. That's a 2 which is saved, and you can see. So when you get a list, obviously you don't know immediately which one is which, right? When reading it in. Therefore, we have this comment field here, where MNE stores which epoch contains which evoked contains which data. And to grab the first one, we can just index this list like this, or to grab the last one, you use -1. If you have an evoked file on disk that contains multiple evokeds, and you only want a very specific one and you know what it's called, you can pass in the specific condition of that evoked. So in this case, it would be this, right? This won't work in my code now because I dropped some epochs. So I would have to adjust that. 0.51 visual left and 0.49 visual right. And it will only read this one. Okay? I'm just demonstrating that because you may get into a situation where somebody shares with you their evoked file and then you need to know how to open it. Okay, so that's it here. Let's close that. And let's move on to the next notebook. If that's all right with you. Are you still here? I can't hear anyone. All good, all good, still there. Okay, perfect, perfect. I already thought maybe my headphones, my speakers aren't working correctly anymore. Okay, so far we've worked only with awfully contaminated data. We didn't perform any sort of artifact rejection except for filtering, right? Which can remove quite a bit of the power line frequencies, but I mean there might be participants blinking, or there might be ECG artifacts. So we probably should look into removing those as well. Okay, so first we're going to run our imports again, and we're going to add another line here. MNE.set log level, we're going to set it to warning. This will mean that all MNE functions we run will only spit out output if they believe they have reason to warn us of anything. As you could see in the previous notebooks, sometimes output was quite extensive, and we just want to cut down on this now so we can focus on what we're actually doing. And so if we set the log level to warning, we will still get warnings and error messages if something goes wrong. We just won't get so much info on things that went well. Okay, so I'm going to run this one. And I'm going to load the epochs we created in the previous step. So in my case, it's the epochs that go from -250 to 800 milliseconds, and with this odd baseline. So what we want to do, what we probably should do first is we should reapply a proper baseline correction. So what we want to do is we want to do epochs.apply baseline, apply baseline. So and I'm pressing Shift Tab again, and it says accepts none or tuple of 2. And what we're going to do here is we're going to pass the tuple non-zero. This is going to apply baseline correction for the period, for the entire period before stimulus onset. I mean, that's not the way you should normally do it, okay? It's just to get something useful out of the data we saved previously in an odd manner. Okay, so we now have this baseline corrected. Maybe visualize. Let's visualize them. Oh, looks like epochs, right? Lots of artifacts here. Let me maybe scroll down. There should be an EOG somewhere. Where's the EOG? Oops, I think my plot is frozen. Oh no, it's good. So, okay. But so what I wanted to demonstrate is, okay, I just wanted to check that after applying this odd baseline correction here again, which you normally shouldn't do. You shouldn't simply pick a different baseline interval and do stuff. Even though it might not, I think it wouldn't actually make your analysis incorrect, but it's just odd. It's just something I have never seen before, so maybe we shouldn't do it. And I just wanted to verify that everything still looks kind of fine after doing that. Yeah. Okay, so there's a question: where do I get this fifth file from? The epochs. We created that on the previous notebook. So if in doubt, just run the previous notebook again, and it will produce this epoch file. Okay, so now we can ask MNE to drop some of our epochs based on the amplitude of individual channels. This is called specifying a rejection criterion. So we could, for example, say that if we have an EEG channel in our epoch that shows an amplitude of 150 microvolts or above, we're going to reject that epoch. If we have the EEG channel and it shows an activity of an amplitude of 200 microvolts or above, we're going to drop that epoch, and the same for magnetometers and gradiometers. We also have flat criteria, meaning that if now I'm not sure if it's amplitude or variability, I would have to look that up, is below these values, we will reject the epoch as being too flat. Okay? And to actually apply these criteria, we use the drop bad function, and we pass in the reject criteria using reject equals, and the flat criteria using flat equals, and run that. Oh, I'm sorry. I didn't mean to run that immediately. Okay, so if you go up here, where we loaded our epochs, it's at 320 events. Now, after running, there's a question. There is an E missing in the EEG reject. Oh yeah, oh yeah, right. There's an E missing. Here, there is an E missing. Yes, thank you. 150 microvolts. Okay, let's run this again. Actually, to rerun this, we need to load the epochs again. Okay, so gonna go back up here. Gonna load the epochs again. Gonna apply our new baseline. Gonna specify the corrected EEG rejection thresholds. So just to repeat, in the file I gave you, this E here was missing. Looked like this, but there needs to be an E. Gonna run this. And now we're going to drop the bad epochs. Okay, and what you will see here is that we now have 251 events left, and originally we had 320. So it's we lost quite a number of events. We can actually ask MNE, okay, so how did that go, and why did you remove events, like based on which criteria, and we can have a visual representation on that. And we just use epoch.plot_drop_log. So this function only works after we've run drop bad. Okay, let's do that. And you can see here, there's quite a number of bars. Up here, it says unknown 21.6%. So this 21.6% means that 21.6% of our epochs were dropped. So we lost a fifth of our experimental data, which is horrible, if you ask me. Unknown here, I didn't know what that meant until I figured that's the subject name, participant's name. Since we haven't specified one, it just says unknown. So it doesn't mean that 21.6% are unknown. No, participant named unknown, colon, 21.6% of the epochs were dropped. And here we can see on the x-axis, we can see which channel contributed to which amount of epochs being dropped. Okay, and we can see that's definitely EOG, and also this EEG channel, a few EEG channels here, which we might speculate is also related to ocular activity. But definitely the EEG channel led to lots of rejections. Okay, is that clear so far? What happened here? Okay, let's just work with that. Let's just accept that we lost a fifth of our data. And let's plot this herb image again for the visual stimuli. And we can see, we still cannot see too much, can we? By the way, I forgot to mention, to tell you earlier, this graphic is actually interactive. And if you click on this color bar, you can actually start to adjust the scale. We can see a little bit, but it still looks super messy. Okay, so maybe, so we lost lots of data and we still cannot see that much. So maybe that wasn't the best approach. Maybe we can look at this herb image in a different way. So for now, we had a combination of across all channels. What if we pick a channel that is closer to the primary visual cortex? So some channel at the back of the head. Now, which channel name would we have to pick? Well, let's just plot the sensor locations again, just to remind us. Maybe this one. Oh yeah, it's channel EEG 060. Maybe we can look at an herb image of that channel. And to do that, we just call plot image, and we pass, as pix, we pass the name of the channel, EEG 060. And this already, I mean, this looks like something, doesn't it? I mean, I'm not an expert on visual EEG, but this looks nice. I mean, the timing looks good. So it's like at, I don't know when it starts, at 100 milliseconds, and then the peak is at 180 or so, and then it goes up again. I mean, that is cool. And you can see here, this signifies the peak across the epochs. So it always occurs approximately at the same time, which is neat. Anyone disagree? Anyone who knows more about visual stimuli and visual activity than I do? Isn't too difficult. Okay, no one disagrees so far. So, okay, this is great, but we got rid of so many epochs. I mean, maybe that's even acceptable if you have a visual or auditory experiment, because it's easy to acquire more data. But I used to work with taste data and olfactory data, and we typically have like let me think, 2 to 3 stimulations per minute. So we have really just so few trials and so few epochs, meaning you don't want to lose them all. So you might be interested in ways to preserve these data and to somehow get rid of this artifact without actually having to drop these epochs. So if there was a way for us to remove the blinking artifact from our epochs without having to remove the entire epochs, that would be awesome, wouldn't it? And there's multiple methods to do so. One approach that is commonly used in MEG, but can also be applied to EEG. I tried that one, but it's not so common, is called SSP. And that's short for signal space projectors or signal space projection. I don't want to go into details. You can find the details on the MNE website. If you go, so we go to tutorials and then pre-processing, repairing artifacts with SSP, signal space projection. It essentially uses PCA to get a hold of your artifact and then tries to project it out or regress it out of your data. I hope that wasn't so much simplified that it got wrong, but that's essentially what it does, okay? And MNE has some convenience functions to automatically help you detect EOG and ECG artifacts, create SSP projectors to remove these artifacts from your data, and be happy. So let's see how we can do that. First, we need to start again with the raw data, because what MNE is going to need is it's going to need the entire the continuous data to work properly. Okay, I think I'm not entirely sure. I think internally it will create epochs which are centered around the EOG and ECG artifacts, and will then try to capture these artifacts and try to remove them using an SSP projector. Don't quote me on, it's probably only like 75% correct, but that's essentially the gist. What's important for us is that to create these projectors, we need to start with the raw data again. So we're going to read our raw data. We're going to filter it again, just the way we pre-processed the raw data before cutting it into epochs. Okay. And then we're going to use these two functions here. One is called compute proc ECG, and the other one is called compute proc EOG. As the names indicate, the first one will try to find and correct for ECG artifacts. The second one will try to find and correct for EOG artifacts. Now, if you remember, I hope you remember, you probably do remember, we didn't have any ECG channel in our data. So how the heck would MNE know how to find the ECG? Well, apparently, I haven't tried it myself so far. I'm embarrassed to admit that, but apparently you can use the magnetometers to get a pretty clear signal of the ECG. So if you don't have a proper ECG channel, didn't have an ECG electrode attached to your participant, but you did record MEG data, you can use the magnetometers to reconstruct the ECG. This is great. We don't need to do the same thing for EOG because we have a proper EOG channel. Okay, Now let's just run that. So we're going to import the raw data, and we're going to create these projectors. And running that now. Just don't know how long it's going to take on this computer. I thought it didn't take so long yesterday, but I'm working with different epochs today, so epochs are a bit longer than the ones I tried it with. Oh, okay, it's done. It's done. We can have a brief look at these projectors. So we just type in the name here. Oh yeah, what I forgot to mention is, so these functions, compute proch ECG and compute proch EOG, they accept several arguments. I have to admit I'm not an expert there, but these are the parameters we typically work with and that have worked best for me in the past. Okay, so this is a good start. if you don't know where to start, this is a good start, certainly. Okay, so we look at the ECG projectors. Actually, we get a list of 5 projectors, which seems to be a bit odd at first. But it's just these 3 projectors. They were already in the data when we loaded it. I'm actually not sure when or how they were added, but they are already in the raw data in the sample data set. And these 2 here, they were added, ECG planar and ECG axial. I hope I pronounced this correctly. So one is for the gradiometers and one is for the magnetometers. Let's have a look at the EOG projectors. And here we have 6 now. We have the 3 that we inherited from our raw data. And there is now EOG planar, EOG axial, and EOG EEG. This is neat. So now we have one for the gradiometers, one for the magnetometers, and one for the EEG. Okay, but how do we make use of that stuff? I mean, it's so theoretical up until now, right? So first, we're going to combine these projectors. Okay, we're essentially going to concatenate them. I say, okay, please add up the EOG projectors and the ECG projectors and save this as the variable proch. So here, now we have this combination, okay? So we have the ECG projectors down here, and we have the EOG projectors down here. We have these 3, and these, they are now duplicated. I'm honestly not sure if that's a problem. I guess not, because I haven't run into issues until now. Okay, so we have this, essentially this list of projectors, and we're going to add these projectors to our epochs, okay? Using the add proch function. And after that, we're going to plot the epochs again. Let's see what happens. Okay, so I'm going to use the function key and press left to change the timescale a little bit. Okay, now I want to bring your attention to this. It's super tiny on my screen. There is this button on the bottom, right, called proch. This can be used to activate and deactivate projectors. If you click toggle all projectors will become active, and we can see the effect of these projectors immediately on the data. If we click toggle all again, it's noisy again. Okay, let's see. Okay. So these artifacts here, this looks a bit like ECG. Okay, it looks very much like ECG actually. We can remove them from the data by activating the, always clicking the wrong one, by activating the ECG projector for the correct data type or correct channel type. Can you see that? And you can play with that. Activate it, inactivate it, activate it,
inactivated. Isn't that cool? That's something that's super neat about these SSP projectors. You can just activate them and the artifacts will go away, hopefully, and then you just disable them and then you're back to square one. And you can play with them. You can see which ones actually make sense and which don't. Okay, but I want to go down here. It's my EEG. It's our EEG data. Let's see what happens if we remove the EOG artifact. So let's hear EOG, EEG. Wow, isn't that cool? I mean, it still could still be better, but I think it's quite impressive already. So let's see if we can get a better view here. Oh, my computer is a bit. Okay, this is without artifact correction. With artifact correction. Without. And now let's just activate all. And activating the rest of the projectors doesn't change anything for the EEG data because the other projectors only apply to MEG. Okay. So, but I think that's already quite impressive. So I'm scrolling further to the right to later epochs here, and again, let's talk at the projectors off, on. It looks much cleaner now. Yes. So Olaf just mentioned these projectors may also remove real brain activity. Absolutely right. This is always the case when trying to detect and remove artifacts. What is an artifact anyway, right? And I don't have a good answer or solution to this problem, except for always being cautious and inspect your data visually. And Olaf just suggested to have a look at the signal-to-noise ratio of your data.
Sorry, can I just briefly clarify? I mean,
yes, that would make total sense if you could.
I mean, I don't want to confuse too much. I just want to say, I mean, the best way to remove artifacts, of course, is to delete the whole data. So
yes.
There's an optimum somewhere, between removing your artifacts and removing part of your data.
Yes.
So I think with some artifact, it's obvious, if you have eye blinks or your heart. But I mean, heart, for example, in evoked studies, I don't think it's necessary to remove heart activity if it's not time locked or phase locked to your stimuli.
Mm-hmm. Because it will disappear as you do the average.
Yeah, the averaging.
That's a good point.
Systematic. In the end, you subtract two conditions anyway. So if it's the same in both conditions, it will subtract out. But for resting state, it might be an issue. If you have old and young participants and they differ in heart activity, that could be a confound. But what I'm suggesting is you can always add your projectors and see if it really increases the SNR, let's say for the N170 or something like that. So then you can check. How would
you assess the SNR in that specific case?
Yeah, compared to baseline. So amplitude versus standard deviation and baseline. So I would say,
okay.
So I would say if you remove brain activity as well, it would remove signal and noise, and the signal to noise ratio might not change.
Okay.
Or it could even get worse. But if you remove noise but no signal, then you should get a better SNR. So that is one, I think, at least quick way of checking whether you're removing too much or potentially not enough.
It's a good point.
Yeah, so I mean, that only works for event-related designs. I mean, again, resting state might not be, there you are on your own, basically. So, and you might want to remove too much rather than not enough, because obviously eye blinks and heartbeat might be an issue there, especially if you compare different participant groups who might differ in eye activity and heart activity. But sorry, that was just a side remark, so I'm happy to disappear again.
No, it's all right. It's thank you for sharing your thoughts on this. I mean, as I said initially, if you can afford to just remove 20% of your data, maybe you don't need to apply any of these methods. But it's not only a waste of data, but it might also raise questions on the side of the reviewers. Like, did you maybe cherry pick? I mean, you might overlook things if you just drop data
However, as Olaf mentioned, if you're too strict about your artifact rejection using SSP, and later, we will also explore ICA, you might also remove actual brain information, which is not noise, and it's a difficult. Okay, so you had a bit of time to play with that stuff. What was? Oh yeah, and the last bit I wanted to show you is, so when we did this plot now, we had to navigate to this projection button, and then we could toggle the projectors on and off. For certain visualizations and certain analysis, you want to have these projectors permanently applied. You want to have them on because, for example, this plot image function, it won't allow you to toggle the projectors on or off. And I think by default, if the projectors haven't been explicitly turned on before you run this function, by default, the projectors are off. So what we want to do is we want to turn these projectors on by calling the function apply proj. This will apply the projectors. There is one caveat though, which is that once projectors have been applied using the apply proj function, they cannot be removed anymore. So you're stuck with them. Therefore, we create a copy here, apply the projectors on the copy, and I'm just going to call the copy epochs cleaned. And then I'm going to create the herb image again. Let's see if it looks any better now than it did before we did the artifact correction. Well, I don't think it looks so much better, but what I can see immediately is that the signal amplitude is much lower and is in a much more sensible region. Okay, so now EEG goes up to, what is that, 30 something microvolts. That makes sense. Okay, let's have a look at the electrode at the back of the head. OZ or something. I'm not sure if that actually changed something here. So, which makes sense, I suppose, because it's at the back of the head. So it's probably not, there's probably not too much influence by eye blinks. If we had picked an electrode further at the front, we might have been able to see more of an effect. But this was really just to show you that it is possible with MNE Python to automatically detect some sorts of artifacts, namely ECG and EOG artifacts, and to try to remove those artifacts from your data in an automated fashion. However, as we just discussed, you should always be super careful about what you consider an artifact and whether and how aggressively you try to remove those artifacts or those potential artifacts. Yes. Okay, so that's it for today, right? Right? It's 3:40 in London. So I think the schedule said the workshop lasts until 3:30. What I didn't have enough time now is to show you ICA, which is another method for artifact detection and correction. I'm not sure how widespread ICA is in MEG actually. Most of the people I know use SSP, but since we're also talking about EEG in this workshop, and everybody I know who does EEG also uses ICA, I also want to introduce you to ICA, but we should probably do that tomorrow morning. Yeah, thank you. Is there any? Oh, you could do this all day, Olaf. Yeah. Okay, let me try to address the questions that came up. Is it possible to import an epoch data set already pre-processed with another software? Yes. That's what I have done in the past. Yes, it is very well possible. There is an MNE function called, oh my goodness, read epochs. I'm just gonna Google, okay? Read epochs EEGLAB, I think. Just that. Yeah, MNE.read epochs EEGLAB. Okay. So yes, it is very well possible to import data that was maybe pre-processed with another software. It's been a while since I've last used read epochs EEGLAB, so it takes a number of parameters, so I'm not entirely sure. I think you need to pass at least this event ID thing for it to make sense, if I'm not mistaken. But yeah, just try it. And if you run into issues, you can also drop me a line and we can see how to resolve that. Olaf says they're using ICA for both EEG and MEG. Yes, I've been looking into that too. And one colleague of mine actually does the same thing. However, everyone else uses SSP for MEG. So, okay. So that's it for today. Here we go. Are you all ready? I would like to ask you to open notebook number 4. Hopefully the new one you just fetched. Drop me a message in the chat if you're experiencing any difficulties. Okay, so yesterday we looked at how to reject artifacts solely based on the amplitude of a recording channel. And then we looked at signal space separation, or called or short SSP, which is commonly used by MEG folks, if you will. And now for those who are interested in ICA, independent component analysis, we will look into that. To do that, we first need to scroll up to the first cell. Because I had shut down my computer and restarted it now, all the variables we had created yesterday are gone, so we need to recreate them. We need to load the data again and do our imports and all that stuff. So I'm going to navigate to the first cell. I'm going to run it by pressing Shift and Return. So this is done. I'm going to import the epochs we created yesterday. And as you may remember, they had a kind of weird baseline set, which didn't include the entire pre-stimulus period. We want to change that now. So we're going to call epochs.applyBaseline to set a baseline in a way that is more commonly used, meaning that we consider the entire pre-stimulus period as baseline, and we use that for applying baseline correction. To see and verify that everything worked correctly, we just visualize that, hopefully. So I was experiencing some issues with my plotting environment yesterday, but it seems to be working now. Okay, so here we have the epochs. So the reason why I just for your info, you may have noticed that I just did a quick whoop and switched back. Switch here and switch back. This I did because I realized this window was somehow not activated properly, which sometimes is an issue on Mac OS. So let's see if I can recreate that. I'm going to close that, call the plotting function again. Now it's activated. You can see the title bar has the correct color. In my first attempt, the title bar was kind of grayed out, just like that.
as if the window was in the background. And even by clicking on it, I couldn't properly activate it. This is a bug, not in MNE, but I think in Qt or PyQt, which is one of the graphical user interface frameworks that MNE uses. But if that happens to you and so the window looks inactive and you cannot properly interact with it, you can just switch to another app by pressing on the Mac, it's Command and Tab, and then switching back, and then it should be active. And yes, I can properly interact with all of them. Okay, so we have loaded our epochs. Just a quick recap. On the right side here, we can see there's different channel types coded by different colors, MEG channels, and down here there's EEG channels. Okay, I'm going to close that. Let me get rid of all of these windows here. I did some programming and never cleaned up. Okay, nice background. Okay, so, and now let's do our ICA. I'm going to scroll down here to where it says ICA. First, we need to load the raw data again. Why is that? When we prepared our epochs, I'm not sure if you remember, but I'm going to give you the recap. So to get to our epochs, we loaded the raw data, we applied a certain set of filters. I think we used a high pass filter of 0.1 hertz and a low pass of 40 hertz or something in this range. And only then we cut the raw data into epochs. Now, it has been shown that ICA works best if you apply a high-pass filter with a higher cutoff frequency, not 0.1 hertz, but more like 1 hertz. This increases ICA performance. Since the epochs we have here have been processed with a different filter, we don't really want to feed them into the ICA. We would like to produce a new set of epochs with filters applied that are of advantage to the ICA algorithm. And to do that, we have to start with the raw data again. So as you remember from yesterday, hopefully, we're going to import the raw data from our bits directory. We're going to load it, and we're going to apply a filter. This time, it's a bandpass with the lower cutoff at 1 hertz and the high at 40 hertz. Takes a bit on my computer. Yours is probably already done. Okay, there we go. Data is loaded into memory. Okay, now, this is a bit of a tricky part, but I think it's important. The epochs we produced yesterday, let's just load them first, so you can see what this is all about. Going to load the epochs we produced yesterday. If you browse the epochs, I'm just going to show you again. If you call epochs.plot, you get this visualization, and if you click on an epoch, it becomes red. And then if you close the window, these red epochs will be dropped, will be removed from the data. Okay. So this means that if we were to take epochs produced from our ICA, produced for our ICA data, let me start that again. This means that the epochs we produced yesterday and saved to disk may not contain the exact same information as new epochs we're going to produce today based on raw data that is specifically filtered for ICA. Does that make sense? So we have raw data here. We have filtered it for ICA. We want to cut this raw data now into epochs. But we also want to ensure that we keep the same epochs as we as we kept yesterday, we want to exclude the same epochs we dropped yesterday. Okay, and this selection of epochs that we actually kept yesterday, we can extract from the epochs.selection attribute. So we load the epochs from yesterday, and we have a look at epochs.selection. I'm going to do that again. And we get an array, goes from 0 to 319, because actually I didn't remove a single epoch yesterday. Okay, so this is all 320 epochs are still included there. But had I decided to remove an epoch or several, some of these numbers here would be missing. Okay, to create the new epochs, which we want to keep and which we want to use for ICA, we first need to extract the events again. So we call events from annotations. And we do this additional step here. Let me maybe demonstrate that. Let me take these two lines apart. Let me put them into different. Okay, so first we extract the events. Just to recap, this is just a NumPy array. 3 columns, and the number of rows is equal to the number of events we have. You can also look at the shape, it's easier to grasp. So 320 rows, 3 columns. Now we can use this epochs selection we extracted from the data we produced yesterday. We can use that to index and select a subset of these events. We're going to do that by just putting the this NumPy array that contains the numbers of the epochs we intend to keep into these square brackets. And this is going to be the events the epochs or the events corresponding to epochs we intend to keep today. Okay, and in this case, it's all of them. Does it make sense so far, or did I totally confuse you? Just raise your hand or let me know in some other way if you're lost. Okay, just going to continue then. So now we're going to use this subset of events. In our case, it's the full set. It's an exceptional case. We're going to use this subset and we're going to feed this subset of events into the epochs constructor. So we're now going to create a new set of epochs, hopefully with the same properties as the epochs we produced yesterday. And I can already see that this is not actually the case because tmin and tmax were set differently by me yesterday. So I guess what we should rather do here is, instead of providing like a raw number like this, we should probably use epochs.tmin as the minimum and epochs.tmax as the maximum, because just to demonstrate that to you, this contains the actual beginning and end time of the epochs that we loaded, Tmax. Okay, so we're going to use the same. There is a question coming up. So the question was whether you're supposed to type this as well. So this part here would make sense to type this, okay? The part I'm highlighting right now. Setting tmin and tmax to the values that were used for the epochs we produced yesterday. And doing it this way will ensure that you don't accidentally pass in a number, a value that is different than what you used today. Okay, and baseline, again, we're going to use the time from the beginning of the epoch till time point 0, which is event onset. And that's it. Let's just run that. Okay, that's it. So I will, because that's maybe a bit confusing. I will try to recap again what we just did. We had a bunch of epochs produced yesterday that were pre-processed in whatever way. And we decided to run ICA or to clean artifacts of these epochs using ICA. Now, research has shown that ICA performs best on data that is pre-processed in a slightly different way, meaning with a different filter. There's a question. Okay, the question is, can we also retrieve the baseline of an epochs object after we had created it? So what you can do is you can always do epochs.baseline, and it will tell you which baseline was used. Okay, does that make sense? So what you could do here as well, of course, is you could do epochs.baseline to retrieve the exact same baseline as we used yesterday. Maybe that's actually the neater way to do it. Let's just do it that way. Okay, where was I? So we had pre-processed raw data and cut it into epochs. Now we want to use ICA to correct artifacts. Research shows that to get the optimal results out of ICA, the data should be processed slightly differently. Namely, we should apply a high-pass filter with a higher cutoff frequency of about 1 Hertz. Now, we cannot simply run a filter step again on the epochs we already have. Or theoretically, we could do that, but practically it would lead to issues. You typically don't want to filter epoch data again because you get artifacts at the edges. We want to avoid that. Therefore, we have to bite the bullet. We have to go back all the way to the raw data. We start with the raw data again. We apply a different set of filters. We slice this data into epochs. But now we might end up with different sets of epochs actually than we retained yesterday, because yesterday we already looked at the epochs and maybe threw out a bunch of them because they were so bad or were obviously something that we don't want to keep. So we might end up with, let's say, 300 epochs of the data we produced yesterday and 320 epochs for the ICA. And that's not a good idea. So we should use the same set of epochs to run our ICA on as we used or as we retained from our yesterday's data. Does that make sense? And to achieve that, we had a look at the epochs.selection attribute, which essentially is it's just an array that contains the indices of all the epochs that we kept. And now we're using this selection. It's a bit of a workaround, okay? We're using this selection to select a subset of the events that we are going to use to create new epochs, which we will then feed into ICA. Whew. Okay, if there's any questions, feel free to ask. I hope this was kind of understandable. Okay, let's have a look at the epochs we just created. So we can see there is, it still contains our MEG channels, stimulus channels, the EEG channels, and the EOG channel, which is 376 channels in total. Okay, let's get ready to actually run our ICA. The ICA, here the indentation is a bit off. Let's just move that a bit to the right to make it prettier. To run ICA, we will use the MNE.preprocessing.ICA object. And it takes actually a number of parameters. And some of them have similar names, and then there is interactions between them. For example, you have N components, max PCA components, N PCA components. And there was so much confusion around these parameters and their interactions that we actually decided to get rid of all of them except for N components. I think this is the last one remaining in the development version of MNE, and in future, meaning that in the future, I think with the next release already, the max PCA components and N PCA components parameters will either disappear, or if you use them, you will get a message that says, hey, this parameter is scheduled for removal, please do something else. However, we still need to use some of those. Namely, we need to use max PCA components and N components, which will remain part of the function. Okay, but what the heck does all of that mean? N components, it's just the number of ICA components you intend to fit. This can be either an integer, which is typically your number of channels, minus if you have EEG data and you have applied, for example, an average reference, you would subtract 1 from the number of channels, and that would typically be the number of ICA components you intend to fit. So say you have 64 electrodes and you have applied an average reference, then you would want to set N components to 63. If you have MEG data and you have applied an SSP projector, this would also reduce the number of components by 1 because it reduces the rank of the data by 1. Instead of passing an integer, you can also pass a floating point number, namely a number between 0 and 1, which will be the fraction of, let me try to get this right. It's going to be the fraction of total explained variance you want to keep. Or it will, or let's put this another way. MNE will then just use as many components as are required to explain this much of the total variance in the data. So in this case, 80% of the data. So MNE will determine how many components am I going to need to explain up to 80% of the total variance I can see in the data, and set the number of components accordingly. You can set a method parameter. MNE offers, I think, currently 3 different methods or algorithms for performing ICA. There is fast ICA, there is Infomax, extended Infomax, and there is PCAR. Most of you probably have never heard of PCAR. It's an algorithm that essentially it can produce results similar to both fast ICA and Infomax, but it achieves that with fewer iterations. So it actually converges faster. It was developed by Alex Kramfor and some colleagues, and it has been part of MNE for quite a while. And for whatever reason, it's not the standard yet. But we're working on changing that. So I always use PCAR. I'm very happy with it. It's fast and produces good results, for me at least. But since it's not the default yet, I have to let ICA know specifically that I want to use this very method. There's a max iter parameter, which specifies after how many iterations ICA should be aborted, essentially. So
hopefully it will finish before reaching this number of iterations, and it will say, hey, I've finished converging, we're done, here are your results. But to prevent it from running forever, we can set a maximum number of iterations, and it will just stop when it reaches this number of iterations, no matter whether it has already converged or not. By default, max iter is set to 200 in MNE, which in my opinion is too low. So for my research, I typically set it to 500 or even 1,000, like quite high, sometimes even 10,000, just because I don't want MNE to stop calculating, just because it thinks it takes too long. For the sake of this demonstration here, I set it to 100, just to make it finish earlier. This means that it will probably not converge properly, which will produce a warning, but we don't have to wait for, I don't know, half an hour or so on my old computer. There's a parameter called fitparams, which allows you to pass some additional parameters to the specific method you're using. So since we're using Picard, I have checked the documentation, and Picard accepts a keyword argument, which is fast ICA it, meaning that it will use that many fast ICA iterations before actually starting the proper Picard algorithm. So this is just intended to find a more efficient starting point. That's I've never evaluated whether it actually changes anything. I just, at one point I discovered this parameter and I added it, and I just kept using it. Okay, I mean, you're totally free to also just drop that one. And then there is a random state parameter, which you may also omit, but setting the random state essentially initializes the random number generator to the same value every time you run the ICA, which will ensure that you always get the same results. If you don't do that, the results might differ slightly between runs. Most notably is that sometimes the sign of the results flips. So a component might, in the first run, have a positive peak, and in the second run it might have a negative peak. It's still the same component, but it just looks different when you plot it. And to prevent this kind of confusion, you can set a random state. This is, for example, helpful when you're currently preparing figures for a publication, and because you will most likely have to rerun this thing over and over again to get the figures right, and you don't want the graphs to change every time. Okay, so now finally, let's run this. So we're going to run this, and I emphasize that again, we're going to run this on the epochs we produced just now, based on the raw data that was filtered differently than the raw data we processed yesterday. So it's our new epochs. It's not the epochs we loaded from yesterday, it's the epochs we just produced. Running that. Now I don't know how long this is going to take on my computer. Hopefully not too long, maybe 30 seconds or so. I'm gonna grab myself a glass of water in the meantime. Okay, I'm back. While we're waiting, are there any questions so far? Did I totally confuse you? While talking, I sometimes had the impression that I'm losing track a bit and I'm producing confusing gibberish. So if you felt like that, do let me know, and I can try to explain myself again. Okay, time's up because my ICA has just finished. Maybe I should emphasize that the first step is always to produce this ICA object, okay? And this only takes a fraction of a second. Like, it really sets these parameters and stores them somewhere. Conducting the actual ICA takes place when you call the fit method.
and pass it the epochs, or you can actually also run it on raw data, but we're using epochs, and pass it the epochs you want ICA to be performed on. Okay, now let's view the fit components, or maybe first, let's see here. It says here, it used the Picard algorithm. It recovered 27 components. Okay, remember we had more than 300 channels in our data. But we told ICA to only keep as many components as required to recover or explain 80% of the total variance. So MNE decided, okay, 27 components will do. And there is 3 channel types, magnetometers, gradiometers, and our EEG sensors. So this is actually, I think, a pretty cool feature. So if you have combined data that contains EEG and MEG recordings, you just fit ICA on, on the compound, on this entire thing. You don't have to first run ICA on magnetometers, then on gradiometers, then on the EEG, and then somehow figure out how to combine all that stuff again. But MNE does all of that for you. Okay, enough talk. Let's look at the components. I'm going to go back because I want to show you that this parameter here, inst equals epochs. I think you can also just type epochs. Completion isn't working. If we pass the epochs to the plot components function, it will make the resulting figure interactive. We'll see that in a second. So these are the first, actually I have to count 1, 2, 3, 4, 5 times 4, the first 20, first 20 components. Remember we had 27. These are the first 20. They are labeled. Sometimes the labels overlap a bit with the figure. I'm not sure if that's the case on your computers, but definitely happens here on my small screen. Okay, and I just mentioned something regarding interactive figure. Well, if you click on one of these components, and it takes a few seconds, you get a pop-up. You actually get more info about this component. You see the spectrum that is involved, you see the topo plot again, and you get this, this ERP image. So lots of diagnostic info you can use to decide whether a certain component is potentially an artifact or not. Let's maybe check out this one. And you see there's a repeating pattern here. And let's I mean, no, I'm not an ICA expert, but this looks like something. Okay, I'm not going to teach you how to identify whether these are actually, oh, now I close these windows and now the next set of components is popping up. These are the last 7 of our 27. So now I'm not going to teach you how to find out which of these are actually artifacts and which are maybe not. But I'm going to show you a convenient way to let MNE do the work, or at least part of the work. Yesterday, we used SSP to detect ECG and EOG artifacts. And we're going to do the same today for ICA. The way we do that is that first we create epochs. So we're going to start with ECG here. So first we're going to create ECG epochs. MNE already has a convenience function for that. It's called mne.preprocessing.create ECG epoch. We feed in the raw data, and this time it's the raw data we produced yesterday. Okay, it's not the raw data we filtered again for the ICA. It's the raw data we actually used yesterday before cutting it into epochs.
We feed in the raw data. These baseline and Tmin and Tmax settings I just took from the MNE documentation. They seem to work nicely. So what's going to happen is that MNE is going to try to figure out where the heartbeats are. It's going to extract these events, essentially, and then based on these events, it's going to produce epochs. And in this case, we will say, or we tell MNE to start the epoch half a second before the heartbeat and to end it half a second after the heartbeat. And the baseline period starts with the beginning of the epoch and ends half a second before the heartbeat. Ends 0.2 seconds before the heartbeat. Now, I'm not sure why these values have been chosen, but they seem to work nicely. Then we're going to produce an evoked, and then we're going to run a small tool, actually, some kind of classification algorithm that tries to find ECG artifacts in our ICA. Okay, so we're going to produce these ECG epochs. We're going to create an evoked, which we're going to need later. And we're going to ask MNE to look into our ECG epochs and try to find the components that are actually related to the ECG activity in these epochs. Okay? And down here, we're going to do the exact same thing with EOG. First, we create epochs around eye blink artifacts. Then we're going to create an evoked of these eye blink epochs. Not going to use that for now, in a second. And lastly, we're going to tell MNE to please try to recover or uncover rather the ICA components which are hopefully explaining this eye blink artifact. Once we have all of these components, so the ECG components and the EOG components that explain the respective artifacts, we're just going to concatenate them, and we're going to add them to ICA.exclude. ICA.exclude is just a list of components, component indices actually, we intend to exclude as soon as we apply ICA to clean our data. Okay, let's just run that and you will see what this is all about in a second. It's done. I'm getting a deprecation warning here because the default for one of these functions will change. This is something you will see quite regularly when using MNE because you're constantly changing stuff. But typically nothing changes in a backward incompatible way from one version to the next. Typically what will happen is that if you update to the next version and you're using a feature that is going to be changed, you will first see a warning like that. And then only in the next release after that one, this is going to change. Okay, let's have a look at the components that MNE proposes to exclude because they are most likely associated with EOG and ECG activity. In this case, it's components number, or with the indices 1, so it's not ordered here, 1, 0, 14. Okay, well, what does that mean? A bit difficult to tell right now, right? So let's look at some graphics. When we asked MNE to automatically find components coding for ECG and EOG activity, it actually calculated some kind of score, and we can now visualize these scores. Let's just do that for ECG. What you see here is on the x-axis, that's all the components
from 0 to 26, so it's 27 components in total. And on the y-axis, you get the score that was calculated in whichever fashion. You can actually select how to calculate the score and which kind of score, which is sticking with the defaults that work best in most cases. So I don't want to go into the details there now. And you can see that there's 2 bars sticking out here. That's the one for component number 1 and for component number, I guess that's 14. So these are very likely to be associated with ECG activity. Let's have a look at a similar figure for the EOG plots, EOG scores. So I'm just going to call the same function, ICA.plot scores, and I'm going to pass in EOG scores instead of ECG scores. And we can see, so it's somehow flipped. And the first component has a pretty visible red bar. So this indicates that, yeah, it's quite likely that component number 0, the first component, is associated with EOG. We can also have a look at the ICA sources. Looks like this. And as you can see here, this is our 3 components that MNE thinks might be related to artifacts. The red line corresponds to the artifact which might be EOG related. And I'm not sure which color that is actually. Turquoise, no, it's lilac. These are found to be likely associated with ECG. So this is a good diagnostic plot. So this is why is the red one so flat here? Well, it's because we just plotted this for the ECG. Therefore, we see strong activity here on the ECG components, but not so much for the EOG component. If we were to call the same function, ICA.plot sources for the EOG evoked, and this is, by the way, the only reason why we produce these evokeds up there. Okay, it's just for visualization. So now we're going to plot the evoked of the EOG, and we're going to look at the ICA sources there. And here you can see that now the ECG-related components are rather flat, whereas the red one, which is likely related to EOG, really sticks out. So this shows that this component really captures something here. Okay, I'm going to close. Is that clear until here? Oh, I'm just seeing there's messages in the chat. Okay, there's 2 questions. Does ICA here go off template bad components? Actually, no. Actually, it uses different algorithms to try to detect which components are bad. I think it can use correlation, and for ECG it uses CTPS, but I'm not an expert in that kind of stuff. If your question is whether, I think for EEGLAB there is an extension that allows you to match your ICA or your data to certain artifact templates. I think this doesn't exist for MNE yet. At least I haven't seen anything like this yet. Okay. The second question, will the new version drop PCA completely, or will it apply PCA automatically to compress the data before ICA? Okay, this question is in regards to this parameter, I suppose. And I just realized I skipped the explanation of this one. Okay, this parameter, max PCA components, it determines,
I have to go a bit into detail here. So ICA in MNE is a multi-step process. I think if you do ICA in EEGLAB, you just it's just one step. It does ICA. Correct me if I'm wrong. Whereas if you do ICA in MNE, it first conducts a PCA, and then it allows you to only use a subset of these PCA channels to run ICA on. And there's lots of, and this is where all of these different parameters I mentioned earlier came from, and where all of the confusion of our users came from, because there's so much flexibility that nobody really understood what the heck this is all about anymore. The max PCA components parameter determines how many PCA components to fit during the first step. So if we set max PCA components to 300 here, it means that MNE is going to generate 300 PCA components from our data. And from these 300, it's going to use a subset which explain 80% of the total variance of the data. This is then how the max PCA components and the N component parameters interact. In the newest versions, the newest development version, in the upcoming stable release of MNE, the max PCA component parameter will be gone. The only reason I'm using this here is because there is a mathematical, let's put it, there's an issue here in the maths that are used in the current version. If I don't pass this parameter, MNE will by default, I think, use all, we'll try to estimate PCA components for every single channel in the data, which in our case, it's 300, I don't know, 66 or so. However, there have also been SSP projectors applied, reducing the rank of the data. So now we're running a PCA on data which is not of full rank, and we're asking it to produce a bunch of components which explain the variance of essentially 0. Now, when we conduct the ICA and try to reconstruct the signal in the end, these pretty much 0 components, this handful, will essentially impair our result. I know this is super complicated, but this problem will be gone in the next version of MNE. So in the next version of MNE, you can just drop this parameter here, okay? I'm just gonna write this here. Remove with MNE 0.22. And in the future, if you only want to keep a subset of components, you just stick to N components, and this will do the right thing, okay? I hope this explains the issue here. If you didn't get a word of what I just said, the only thing you need to remember is max PCA components, this thing you should just drop from your ICA call as soon as MNE 0.22 has been released. That's all you need to remember for now. And you can control the number of ICA components and the amount of variance you would like to have explained purely through the N components parameter. Okay, I hope that clarifies it a bit, at least for some. Okay, where were we? Oh yeah, right. So let me go back here. We looked at the ICA sources. We figured that, okay, if we visualize the ICA sources on the EOG epochs, we can see this nice peak here, meaning that component 0, the first component, has most likely captured eye blinks, actually. Super nice. And if we do the same thing for the ECG epochs, we can see that component 1 and 14 are most likely associated with ECG activity. So it should be safe to drop those. But before we do that, let's just do another verification. ICA has a function which is called plot overlay. And this shows what the data looks like after removal of these of the ICA components, which are scheduled for removal as soon as they have been added to ICA.exclude. Okay, remember ICA.exclude contains the first component which codes for EOG activity, and component 1 and 14, which are probably related to ECG activity. So let's call plot overlay. Let's first call it on the ECG evoked. And this is, I think, a very nice demonstration. So what you can see here is obviously the evoked signal based on the ECG ECG epochs that MNE created automatically for us. You can see these epochs go from 0.5 seconds before the ECG till 0.5 after the signal. And in red here, you can see what the signal looked like before running the ICA. Okay, so that's essentially the input signal. And then after excluding these 3 components that we added to ICA.exclude, the signal looks like what can be seen in black, as the black traces. And we can see it here for all 3 sensor types, EEG, gradiometers, and magnetometers. I think here it's most pronounced. And you can, again, click and drag a certain area to get a topo plot. So that's super cool. So it seems the ECG signal has been removed properly. That's produced. So I'm not sure why it produces so many warnings here. Wow, it's full of warnings. Just going to pretend we didn't see that. And we're going to, let's do the same visualization now for the EOG evoked. And I think this is also really nice, and it demonstrates that it's doing the right thing here. So this is these are the it's the evoked signal based on the automatically generated EOG epochs. And again, in red, you have the original signal, and in black, you have the signal after removal of the EOG and ECG related components. Okay, somebody's saying there is nothing red in the graph, it's all black. Okay, if it's all black, the first thing you should check is, you should check if ICA.exclude contains anything at all. Is that the case? Or is it not the case? Does it contain anything? Let me rephrase the question. Does it contain anything? Do you get an output which is similar to this? Yes, so you do get something. 0, 1, and 16. Are you on Mac OS? You're on Windows. Is it all black both for when plotting the EOG and the ECG, or is it just for one of them? If you run this again, plot overlay with EOG for both. That okay, let's take one more step back. If you run this, plot sources with the EOG, does it look something like this? It looks the same but flipped. Okay, this is already good. This means that the ICA sources have been discovered and have been associated with respective artifacts. And you also get this. You get 3 components here in the legend, and one is associated with EOG and 2 are associated with ECG. Is that right? Okay, that's good. The graphs being flipped is totally to be expected. Okay, this just happens. If you were to run ICA again a few times, sometimes it would point upwards and sometimes it would point downwards. Okay, so this is good. So the issue you're experiencing seems to be solely related to the visualization of this what MNE calls an overlay. So old data and new data. When you say it's all black, do you see these, like, is it all flat like this, or do you see these peaks that go up to like more than 100 microvolts for EEG? Looking at the EOG evoked now. I mean, this actually sounds like some sort of bug, but I'm not sure how we can tackle that now. It's kind of odd. And do you get these plots inline in your notebook, or do you get a pop-up like I do? Pop-up, which sounds, I mean, this all sounds correct, you know? So I'm super confused as to why the color seems different. Okay, so Pierre, so we're just going to move on for now, but maybe that's something we can look into later again. It really should work. It's not complicated code. So, but and it works for all the others? Does it work for everyone except for Pia? Do you get these figures where some traces are red and others are black? Just want to ensure that's not a common problem. Okay, I'm getting some yeses. Okay, that is great. Okay, and I just realized I forgot actually the most important part. So we actually calculated the ICA and we verified that it will hopefully do the right thing, but we forgot to actually clean our data. So let's just create a new cell here. And what we're going to do is we're going to call ICA.apply. For whatever reason, I'm not getting tab completion anymore. I'm not sure if that's the same for you. So I don't remember how ICA apply works. So I'm just going to add a question mark at the end. Oh, I'm not getting tab completion because this window is still open and seemingly it blocks something here. Yeah, okay. Now it works again. So that's another pointer. If you have the impression your notebook is kind of stalled and won't respond anymore, maybe there is still an open plotting window somewhere that prevents you from interacting with the notebook again. Okay, so we need to apply this ICA. There is this method or function called ICA.apply. It accepts as first argument an instance of an epoch, evoked, or a raw. We want to apply this to our epochhed data, and I think that's it. I think the rest we can actually exclude. I think the rest we can actually skip. So, and now the last thing we need to figure out is it going to change our data in place, or is it going to return a copy? Let's see if this is somewhere hidden in the doc string. It only says it's going to return the processed data. Ah, here it says the data is processed in place. Okay, it's always important to check that. So for that reason, what we're going to do is, so we're going to pass in our epochs. Now we're going to pass in the epochs we produced yesterday. Okay. So this needs to be really clear so you don't get confused and end up working with the wrong data. So we ran ICA on the epochs we produced specifically for ICA. But now that we have discovered which components are related to artifacts, and we want to remove these artifacts from our data, we actually apply ICA to the data we produced yesterday, which is the data we are analyzing while conducting our study. Okay, so we're going to call ICA.apply epochs, and we're going to do that on a copy of epochs because as we just figured out in the doc string, ICA.apply is going to work in place. And so we can work with the output. We need to assign it to a variable name. So I'm just going to call that epochs cleaned. That's a common way to name this. Okay, let's see. Let's hope it works. It may take a bit. And there we go. Let's visualize that. Just calling the plotting function here. I'm scrolling down to the EEG. I mean, the data is still not very pretty. And it's really difficult to tell the effect of the ICA right now, right? Without a direct comparison. But I think we can see, if I'm going back up to the MEG again, I think we can see that the heartbeat artifacts are already a bit reduced. Let's maybe try, I'm not sure if that works. Let's maybe try to open these, the uncleaned and the cleaned data side by side. So I'm just gonna call epochs clean plot and epochs plot, and let's see if we can set a title so we can actually differentiate the two. So this is after ICA, and here we would say title equals before ICA. And I'm going to run that. It's a bit difficult to arrange. My computer is struggling. Oh, I think my computer can barely handle it. It's kind of stuck. Not sure if it's the same for you. Okay, so they don't have the same size. Let me try to move that here a little bit. Actually, oh, what's going on? Okay, so now let's change the zoom factor a little bit. On my Mac, I'm going to press function and cursor key left a few times, and I'm going to do the same for the right window. And I guess one can already see a small effect here. For example, in epoch number 4, just going to highlight that. It's this one here. We can see that here, that's a heartbeat artifact. You see that here? This is pretty much gone. Yeah, I think it's entirely gone. Yeah, so it actually worked. Yay! And if we were to go down to the EEG, we should see the same happen for eye blinks. Although it's a bit more difficult. We would have to change the zoom factor again. But I just wanted to show you that there is actually an effect, and you should have a look at the clean data after applying ICA. It's really important to see that to ensure that nothing fishy is going on. Okay, I'm going to close that again to help my computer recover from this shock. And that's it. So if we wanted to, we could also, of course, save these cleaned data, right? We would just call, epochs_cleaned.save, and insert a file name here. I'm just going to skip that for now. Okay, all clear up until here? So now you know how to conduct ICA. Oh, there's a question. How are other artifacts removed? For example, motion artifacts. You have to remove those manually. So currently there is only automated detection of ECG and ICA, ECG and EOG in ICA, and other types of artifacts you have to remove manually. I think there is now a way to at least it's not directly related to ICA, but it's related to artifact detection. I think there's now a way to detect certain types of muscle artifacts, but I would have to check out the docs myself. Let me just do that. It might only be in the development version. I think it's called annotate muscle or something. Here, annotate muscle artifacts. I'm going to switch back to the documentation of the stable version to see if we have the same thing there. Yeah, it's already in the latest stable version. Okay, so there is a way to automatically detect and annotate movement or muscle artifacts. I've never done that myself. So essentially one would use the function annotate muscle z-score. And I'm not sure which kind of annotations it actually adds. Okay. So it first tries to discover, okay, there is muscle activity in the signal. And then you can add this information to your raw data in the form of an annotation. Remember these annotations when we read our data from, we first converted our data to bits, and then we read it back in, and then we were like, but how do we get the events now? And I told you that, hey, the events are now stored as annotations, and we use this events from annotations function, which would go into the annotations and convert them back to events. I also showed you how to add annotations to your raw data by pressing the A key, and then you could select stuff. And essentially the same thing can be automated for muscle artifacts if you follow this, this tutorial. It will add these annotations. They start with bad underscore. And annotations that start with bad underscore are automatically ignored by MNE when you process the data. So this means that by default, if you have muscle artifacts that are marked that way in your data, and then you epoch the data, and MNE detects that there's going to be a muscle artifact inside the epoch it intends to create, it would just not do that for this one epoch. It would just drop it. It would say, hey, there was a bad annotation, there was a muscle artifact in would have been epoch 25 or whatever. I'm just going to skip that one. Okay, so that way you wouldn't even come into a situation where you would need to desperately try to remove muscle artifacts or the like with ICA. Yeah, of course, this would mean you would lose an epoch in this case. Okay, but that's the current approach I would take personally, because there is, like I said, there is no automated way using ICA to recover muscle artifacts. I think ICA is not a suitable method for that anyway, because these, I think muscle artifacts are just not suitable, like movement artifacts. Just not suitable as opposed to regular ECG and EOG artifacts, which, repeat. They don't occur just once or twice, but they occur throughout your data. Whereas a muscle movement artifact might only occur a few times. And I'm actually, I mean, maybe there's ICA experts around here, but my impression is that ICA typically won't capture this kind of artifacts, and they should actually be removed before running ICA, because it might just throw off the algorithm. But please correct me if I'm wrong. Okay, does it answer your question, Borja? Okay, you're welcome. Good. Yay. Okay, so don't forget the last step, okay? Where is it? Here. ICA.apply it's the important step to actually take advantage of your ICA and clean your data. Okay, I'm going to close that one now. And we're going to do a brief excursion to EEG and EEGLAB. So I'm going to use notebook number 5, and we're going to need the data that I put into the subfolder or subdirectory, which is called EEG_montage_data. If you haven't downloaded this folder from Google Drive, please do so now, because yeah, we're going to need this data. Do you all have this data? And maybe I can share the Google Drive link again. Well, if I only knew where I put it. Just share the Google Drive link again. So you're going to need this directory as well, okay? EEG_montage_data. All right. So yesterday a question came up, and the question was pretty much, oh, sorry. Need to have a sip of water. Yeah, excuse me. So, and the question was pretty much, what if I have data that was pre-processed in EEGLAB, and I want to work with that, I want to continue working with that? Just, yeah, download the file and, well, unzip it, obviously, and then it should contain an EEG_montage_data folder, and you should put this into the same folder where you have stored all your notebooks. Okay, I'm just going to move ahead now. Okay, I just received another read first. Okay, so one of the participants currently is experiencing issues with plotting, so plotting won't work at all anymore. I don't know. I may have an idea on how to approach that, but let me first go over this notebook. So what we want to do is we want to load EEGLAB data. Actually, this is a bunch of epochs that were pre-processed by a colleague of mine using EEGLAB, and we wanted to continue analysis using MNE Python. So first, obviously, we're going to import all the stuff we need. And to read EEGLAB epochs, we first have to specify their file name, obviously, and location. And then we can just use the read epochs EEGLAB function. So let's just run that. I'm just going to insert a new cell here so we can view the epochs object. And you can see here that we imported 335 events or 335 epochs. These are the trigger numbers that we used. And this here on the right side, this is the number of corresponding epochs or events. So first thing you may notice is that there's just event numbers, there's not event names. We can work around this by passing a parameter here, which is called event_ID. This can be a dictionary, which again just maps certain event IDs, event numbers, to actual experimental conditions or proper names. I'm just going to skip that here now, because what I actually wanted to show you is that if we try to look at the sensors now by using epochs.plot sensors, you're in for a surprise, because something is off here, right? I mean, we do see sensors, and they have labels, but where's the head? Like, something is clearly off. So gotta close that. And what we want to do is we want to ensure that we use the correct montage. This is also something you will have to do when you're working with EEG data that has not been exported from EEGLAB. Setting an EEG montage, telling MNE where the sensors, where the electrodes were located during recording, is an important step as you process and analyze your EEG data. So I'm just going to show you how that works. So what we're going to need is what MNE calls montage file. I think outside of MNE, this is it's just called sensor layout or something. It's this file I put here into the EEG montage data folder, and it has this super long name, in this case, ActiGem, 64 channels, whatever. And let's have a look at this one. And it's just a text file that contains channel names and locations according to the theta phi coordinate system. And the way I got my hands on this file, if I remember correctly, is I just Googled. Like I just Googled. I know we used a brain product system, an ActiCap or something. So I just Googled brain products, and then electrode locations or something. Let's see if I can find anything here. And here it says montage. Oh, they also call it montage. Okay, second hit here. And it says here, this is for passive electrodes. We used active electrodes. And here there's actually a way to download this. And it should be very similar with other manufacturers as well. I'm just gonna have a look here. I'm gonna unzip. And I think it comes with like lots of different montages for all their different systems, and you just have to pick the right one. Okay. And once you have this file, you can tell MNE to actually load it using the function read custom montage. Once you have loaded this montage, you can visualize it. So MNE calls this thing a DIG montage, digitization montage. And this also comes with a function called plot. And to make it look nicer, I'm passing the parameter sphere equals auto. Let's just use it that way for now. I'm going to run this. And yay, can you see? The head is back. The head is back. This is great. And the electrode locations do make sense. So FPZ is at the front, OZ is at the back, ZZ is at the apex, vertex, sorry. And FT10, TP10, FT9, TP9 are outside of the head circle, but this is all right. We discussed this yesterday. It's just one of the many different ways to map 3D sensor locations to a 2D plane. You can also see what happens if we don't pass the sphere parameter. Just going to remove it, plot again. Now all electrodes are squeezed inside the head circle. I think it looks bad. I like the previous visualization more. So therefore, that's the only reason why I'm in favor of passing this auto parameter here. Okay, that's really the only reason. Okay, now that we've read the montage, we need to tell MNE to please apply this montage to our data. And we're just going to use the set montage function. And we're going to pass the montage we just created. This works in place. And if we now call the plot sensors function again, we see the head. We see again the not so nice visualization of the sensors. But if we click here now, we can see that actually all sensors are there. Oh, in fact, oh, it's all sensors. All sensors we had previously. Okay, so this is important. This is an important step when you do EEG analysis. Otherwise, your topographic maps will not look right, and when you then start doing the core registration, which is a required step in preparation for the source analysis, this will also not work nicely if you have, will actually not work at all if you don't have the correct montage set. So in this case, we simply used a template montage, which was provided by the manufacturer. So these electrode locations were actually not measured on any participant's head. These are just the default locations that the manufacturer said the electrodes would have as soon as the cap has been placed on our participants' heads, given that we're picking a cap of the correct size. If you have, I mean, ideally, you have digitization points. So you would have montage files with actual digitization points for your individual participants. We never had that in our lab, and I therefore always work with these template locations, template montage. Okay, any questions? It was just a brief excursion just to show you how to read EEGLAB data and set a montage. Okay, if there's no questions, let's move on to the next notebook, number 6, temporal decoding. So when running this notebook, oddly, I experienced some issues with my graphics output, which is that either I didn't get any figures at all, or what would pop up would be empty figures or transparent figures. And the way I managed to work around this is by adding this line here. Let me zoom in so you can see it in its full beauty. Just added a cell, and it says $ matplotlib qt, and this helped. So if you're experiencing issues with your figures, maybe add this one at the top of your notebook, and it might help. Okay, so what we want to do now is we want to use some fancy machine learning to see if there's any differences between two selected conditions in one of our experiments. So first, I'm going to run this important line here, which hopefully fixes my plotting. It switches to a different desktop. I hope that's a good sign. Let me switch back. And next, we're going to do a bunch of imports. You've seen all of this before, or at least most of this. I'm going to load the epochs that we produced yesterday, the epochs before running ICA or SSP. Should be good enough for now. And again, we are going to apply a different base, or a more sensible baseline. Okay. And during this tutorial, I would like to focus only on the auditory epochs. And specifically, I would like to look at the differences between auditory left and auditory right stimulation. So going to select only a subset of the epochs. You remember how that works. Just going to use these square brackets, auditory, which should fetch the auditory right and auditory left epoch. In my case, because I played with this a little bit, I have 72 auditory left, 73 auditory right. Your numbers might vary slightly. Okay. And before we get started with all of that fancy machine learning stuff, let's maybe see if we cannot calculate an empirical difference of the neural responses to auditory left and auditory right stimulation. To do that, we are first going to calculate the evoked responses to auditory left and the evoked responses to auditory right stimulation. And then we're going to calculate the difference of the two. And lastly, we're going to visualize auditory left, auditory right, and their difference using GFP plots, which we produce using plot compare evoked. Okay, to calculate the difference of 2 or more. Actually, I think there is no limit. You can combine more than just 2 epochs, but to calculate difference of just 2 epochs, 2 evoked, you use the function MNE.combine evoked. You pass in a list of evokeds. So here we're creating the evoked objects in place for auditory left. So we fetch auditory left epochs, then calculate the average, and we fetch auditory right epochs, calculate the average. This produces 2 evokeds, and we combine them using the weights of 1 and -1. This just means assign a weight of 1 to this, to auditory left, and a weight of -1 to auditory right, and essentially sum them up. So this means auditory left minus auditory right. And we're going to save this difference to a variable called evoked diff. We're going to plot the GFP of this difference, and then we're going to compare this GFP to the GFP of the input data. Okay, let's just run that and hope graphical output works. It doesn't look, oh, okay. There's something happening. Not sure why there's so many output windows here now. Ah, that's what I was actually looking for. So this is just a visualization of the difference we calculated, okay? So this is the difference of the auditory left and auditory right evoked responses. And we have that for EEG, gradiometers, and magnetometers. And again, we can select here a certain time range, and we will get the topographic plots. Okay, well, I mean, there might be something it's nice to see, nice to have, but let's just move on to the other plots. Just going to close that. And here we have the GFPs. One's for EEG, one's for magnetometers, and one's for gradiometers. That's why we have 3 of those plots. And as you can see, there's 3 different traces here. One trace corresponds to auditory left, the other to auditory right, and the green one is actually the difference, okay? And the difference, of course, is calculated on the evoked level, and then the GFP is calculated on that difference, okay? So what you're seeing here is not the difference of the GFPs, but it's the GFP of the differences. Hope that makes sense. You should not use linear transformations like subtraction or addition when it comes to GFPs, because the GFP is, calculation of the GFP involves a nonlinear term, and you would get results that are not very useful. MNE does that correctly, automatically. Okay, so yeah, we see there is, so in the difference here, there is something going on. So there might actually be a difference, but that's all we can tell so far. And looks similar for gradiometers as well as for magnetometers. Gonna close all of these. Okay, but now let's get started with some machine learning. Yay! Before we do that, we have to take care of something, which is, I showed you up here. I have 72 epochs for auditory left and 73 for auditory right. Depending on how I intend to evaluate the performance of my machine learning algorithm, this imbalance, so 1 epoch more for auditory right than for auditory left, this imbalance might have an influence and might lead me to incorrect conclusions, especially if we're using accuracy scoring. So it's always a good idea to equalize the number of epochs. This will keep the chance level at 50% by having the same number of epochs for all involved conditions. So we're going to use the function equalize event counts, and we're going to tell it, maybe I should show you that real briefly. When you have an epochs object, you can retrieve the event ID dictionary that was used when creating it, just by adding .event ID. There's a typo. And here we can see auditory left was coded as a 1 and auditory right was coded as a 2. And equalize event counts actually requires us to pass in a dictionary like this. And so the easy way to do it is to just tell it, hey, please use your very own event dictionary. Okay. So I personally think that's a bit awkward. You shouldn't have to pass in anything, but I think, yeah, it insists. It insists. I think by default it actually should do this, but currently it doesn't do that. So therefore we have to do this awkward thing here. So we ran this function and we end up now with the same number of epochs for auditory right as for auditory left, which is cool because now if we try to evaluate performance of one of our classifiers and performance is 50%, we know it's chance level, and if it's above 50%, we know it's above chance level. Okay, now let's actually create our first classifier. A classifier is relatively, I mean, the detail of the implementation is complicated, but creating one, or using one, is actually not too difficult. A classifier takes an input, typically called X, with an uppercase X. Uppercase because it's typically a matrix. And the classifier produces an output called Y, lowercase, which is essentially the prediction. What we want to do here is we want to first, one second. Yeah, what we want to do is we want to perform something that's called supervised learning. I was actually missing this term now. I'm sorry. Supervised learning. So we already know the correct responses. So we know which epoch belongs to which stimulation. Okay. And what we want to do is we want to train the classifier that hopefully learns. So we tell it, we show it a bunch of epochs, and we tell it, okay, so this is auditory left, this is auditory right, this is auditory left, this is auditory left, etc. And we hope that it learns the spatial temporal pattern of this. And when we then show it some epochs it hasn't seen before, it will be able to tell us, oh yeah, this is most likely auditory left, or this is most likely auditory right. To prepare that, we first need to create the vector y. The vector has a length which is equal to the length of our epochs, or of our events in epochs. It's the same thing. And we're going to code. So we're first going to create an empty vector, okay, with a certain length. The length equal to the number of our epochs. And then what we're going to do here is we're going to code auditory left stimulation as a 0 and auditory right stimulation as a 1. Okay, and to do that, we just create these indices here. So essentially, we say if we go through all events, okay, and we say that if this event has an ID which is equal to auditory left, then please add a true to our index vector. And if that's not the case, please add a false. And here we do the same with the right auditory stimulation. Let me maybe break up this cell to make things easier. There was actually a command to do that. Split cell. Yeah. So we can run this and can have a look at this index left and index right, because I'm right now realizing it might be difficult for you to actually follow through here. So that's the indices for left simulation. Essentially says, okay, the first epoch was not a left simulation. The second epoch was a left stimulation. Is that okay? And we have the same for the right side. I'm going to remove those again. And now we are indexing our empty vector. We're going to pick these indices where we know that there was an epoch, there was a left stimulation epoch. We're going to assign it a 0. And for the right stimulation epochs, we're going to assign it a 1. Let's have a look at this thing. So eventually it looks like this. Okay, it's just 1s and 0s, meaning epochs were right, left, etc. Great. 140 length is 144. I do remember that we had 144 epochs. Yes, 72 times 7 times 2 is 144. Perfect. So this vector now has the correct length. Now it gets a bit more complicated because we need to create the input x. And here we're just going to focus on the data for the gradiometers because it just makes our life easier for now. We're going to use the pick types function. You've already learned how to do that yesterday. And now there's something new. We're going to use a function called getData. GetData gets us an array or a matrix of the actual measurement data stored in our epochs object. You will see what that does in just a second. I should print data so you can. So it's this super complex kind of thing. It's a matrix or an array with shape 144 times 203 times 631 in my case. What does that mean? 144 is the number of trials we have. 203 is the number of channels. And 631 is the number of sampling points or time points. Okay, and what we have to do now, it's a bit awkward, but what we have to do now is we have to reshape these data. And the way we're going to do that is what we want to end up with is something that has in each, where each row corresponds to one of our trials. So we would have 144 rows. And we would in each row just concatenate column-wise, essentially, the data, the time points for all the channels. So in the first row, for the first, we would have 631 columns for the very first channel. And after that, directly after that, still in the first row, we would have another 631 columns for the second channel. Then again, 631 columns for the third, fourth, up to the 203rd channel. So we create a super wide matrix, if you will. Let's just do that. So we end up with something that has 144 rows and a huge number of columns, like almost 130,000, which is crazy. But it is how it is. And now we want to train the classifier on this data, okay? So what we have here is for each, we really have the entire spatial temporal information that we recorded. We have it concatenated in one row, okay? Now, let's build a classifier. And I know this is all a bit, this may seem all a bit complicated, but I just want to go over this with you. And I'm going to show you an easier way to do it in a second. But I just wanted to show this to you because there might be, you might run into situations where you have to deal with scikit-learn directly. MNE has some convenience functions to make your life much easier, but you may not always be able to use them. So therefore, it would make sense to teach you the hard way first, so you know where you can go back to in case you get stuck with the MNE built in. Okay, so now we have this input vector x, and we have the output y. Now we can actually create our classifier. We're going to use lots of stuff from scikit-learn for that. Okay, we're going to import lots of different things. I won't go into details here, but we're going to create a classification pipeline. First, and this is super important, we have to scale the data. If you look again at our vector x, you can see that these values, they're so tiny, you know, 10 to the power of minus 13 and the like. This is something that will lead to instabilities during calculations because, you know, there's floating point inaccuracies. And also certain types of classifiers just have issues with data that is so tiny and doesn't have a mean value of 0 and the like. So we're going to fix that by scaling the data using the standard scaler. This is essentially going to standardize the data. So it will have a mean of 0 and I think also a standard deviation of 1, but don't quote me on. And the actual classifier we're going to use is going to be logistic regression, a logistic regressor. We're going to use cross-validation to actually train and test our classifier. This means that in this case, we're going to split our data into train and test samples 5 times. The classifier is going to learn on the training data, and it's going to be tested on the testing data. And performance will be evaluated based on how well the classifier is able to work with the testing data. This is going to be repeated 5 times, like I said, and the performance of the classifier in this case will be estimated using the ROC AUC, which is the area under the curve of the ROC curve. Okay. Another scoring method could be accuracy, but ROC AUC seems to be more robust in many cases. Okay, so this will return 5 scores because we have 5 cross-validation splits. So we return 5 scores. We will calculate the mean of these scores and the standard deviation, just, you know, to give us some info on performance. And eventually we will then visualize these results. Whew, this all seems so complicated, although it's just a few lines of code. Okay, let's just run that. Okay, and I do acknowledge that I did not provide a proper introduction into machine learning here. I just want to show you how you can do it. You will have to do some reading on this topic yourselves later on, I'm afraid, if you if you have no clue what I just talked about. But what we maybe just to reiterate that. So what we did here is we fed into the algorithm all of our epochs. And for each epoch, we had the entire data of all of our sensors recorded for that entire epoch. We told the algorithm, please take these epochs and split them into training and test data. Try to figure out, or try to learn the pattern of the epochs for auditory left and auditory right. Try to learn it. Try to find something that helps you to tell these two apart. And then after the learning was done, we told it, okay, so and here's some test data that you haven't seen before. Please show us what you can do. Please show us how well you perform. This is then what we get back as these scores. This is going to be 5 scores. Here they are. And then we calculated the mean of these scores and the standard deviation. There is just a question. Yes, Olaf said his AUC is 0.9. And yes, I think, Olaf, you won. You won. Okay, so what we can see here already is that the mean rock AUC is, in my case, 0.88. The highest value you can achieve is 1, obviously. Chance performance would be 0.5. So we are well above chance, which is good. Let's visualize these results. So it's a very simple visualization, I know, but I think it helps to get a better feel of the data. So the whiskers of this box plot, they actually correspond to the smallest or lowest and highest score or highest performance. And this green triangle is the mean. So that's actually quite nice. So we are well above 50%, well above chance. Okay, so what this shows is that most likely our classifier was able to learn to differentiate auditory left and auditory right epoch. Okay? The conclusion we can draw from that is that it is likely that for the data we fed into the machine learning algorithm, it was possible to find some spatial temporal patterns based on which data did we use? Gradiometer data that differentiated auditory left and auditory right brain responses. Okay, so this is all nice, but maybe we can do this simpler. And as you can see here, there's a decoding module in MNE. Let's just use that because it really makes your life easier, like really easier. Okay, first, we're going to grab our data. Okay, we're going to pick the gradiometer data again. Going to extract the entire data. We don't need to, so this time we don't need to change the data shape or anything. If you remember up here, I cannot find it. Here, we had to use this reshape thing, and this is always a bit error prone. So when I tried to do it the first few times, I ended up with weird results, let's put it that way. So it's good if you don't have to worry about the data shape. Since MNE already knows in which shape its data is or is going to be, we can just use the data as is. And the response vector can just be the events, okay, or the event numbers corresponding to our different conditions. Okay, now we're creating, again, a classification pipeline. We're using a scaler here, which this time comes from the MNE decoding module, okay? So it's not a scikit-learn scaler, it's an MNE scaler, which is aware of your different channel types. So it knows how to treat magnetometer, EEG, and gradiometer data and scale them properly. And it just needs the infrastructure of your data. Then we have this vectorizer. Here, this comment says it all. This vectorizer essentially takes care of bringing your data into the correct shape. So you don't have to worry about that. Super cool. And eventually, again, we use logistic regression. Down here, pretty much everything stays the same. We're going to use a cross-validation scheme, meaning we're going to split the data into train and test automatically. We're going to do this 5 times, and we're going to use the area under the RUG curve to evaluate classifier performance. Let's run this again. And okay, so there is a typo here. Oh, okay, so I'm calling this epochs auditory grad. And who can spot the mistake? I cannot find it actually. Ah, here. It must be called epochs auditory grad. Yes. Okay, so here it has to be epochs auditory grad. Sorry about that. And if I run it now, you run the classifier. First gets trained, and it gets tested. It takes a while. Okay, now it's done. And I get very similar scores. Actually, they might even be identical, huh? Yeah, 0.878 and standard deviation of 0.065. The standard deviation is slightly different, but the mean score is the same for me here. Okay, so this is the entire thing. Like, that's super simple. And it's much easier than all the stuff we had to do up here. I just wanted to put you through it, like I said, so in case you ever have to leave the MNE sphere a bit, that you know how to do that. There is a question. Okay, so the question asked was, if you rerun this, will you always get the same results? And the answer is no. I think if I rerun that now, so we might not see a difference down here because I'm only printing 3 digits after the decimal point, but typically the results would change. There are ways to set a random seed. I think you would have to pass it here. No. Well, actually, I'm not sure now. There is ways to set a random seed. I would have to look it up. I don't know. I don't know now. Definitely not here. This is actually weird because I do remember setting. We have to set it for the classifier somewhere. For the cross. Okay. I think I know. We can, if we're using our own cross validator here, I think that's the key. So we tell it to please perform 5 folds for our cross validation, and it will typically do the right thing. It won't allow us to pass a random seed. Now, if instead of passing a 5 here, oh, by the way, this should be n splits, okay? That's why we defined it up here. This should be n splits. But if we defined our own cross validator, now I would have to look it up. So you can import from scikit-learn. I think it's called model selection. I would have to look it up over the break. So you can define your own cross validator, and you can feed a seed into that one. Okay. Actually, now you got me curious. Let's quickly do that. Scikit-learn cross validate. So there should be different cross validators. Where are they? Yeah, it's okay. Why did this not work? Scikit-learn model selection. Import. Here we go. So there's different kinds of cross-validators. Now I don't know which one you should pick for this type of data, actually. But assume you pick a k-fold. Not saying you should use that one, okay? It's probably not the best one here. Then you can define your cross-validator by instantiating this thing. And typically this would accept a random state. And then down here, instead of passing CV equals number of split, you pass your cross-validator. And once you've done that, you should achieve the same results with each rerun of your script. Okay? But I'm going to delete this here again because this is probably not the best cross-validator for this scheme, so one would need to do some reading on that. Just gonna put this back to what we had before. Okay. Oh, it's already lunchtime. It's already 10 minutes past lunchtime. Okay, so I would say we take a break here. Obviously, we should. After lunch, I will show you. So what we did now, and I'm repeating myself, but I just want to make this clear. We trained classifiers on entire epochs. Okay, so classifiers learn to discriminate entire epochs. And in the next step, what we're going to do is we're going to look into how to actually train classifiers at each time point of our epochs. Because this will then help us to answer the question, at which time point precisely were the two spatial temporal patterns that we observed for auditory left and auditory right stimulation different from each other? At what time point did our classifiers perform above chance, meaning we're able to find something, whatever it may be, in our data that allowed it to tell the left and right auditory stimulation apart, okay? And this is really valuable. This can be very valuable to figuring out at which time, yeah, essentially at which time some processes are starting to behave differently in our data. Okay. Okay. There were a few questions that came up during the break. One was regarding, so there was this one step where we equalize epoch counts or event counts, as MNE calls it, but it's actually the number of epochs that gets equilibrated. Is that the right word? Is that the right term? Equilibrated? Yeah. Can't find it. Equalize event counts. And the question was, how does MNE decide which epoch or event to drop? And I have to say, I don't know. Probably it's hidden somewhere in the docs, and it says here, it tries to make the remaining epochs occurring as close as possible in time. So apparently, it tries to do the right thing and it tries to be clever. So it's not random. So the epoch that is being dropped is not selected randomly, but there is some more thinking behind the algorithm that is applied there. Okay. The next question was whether we actually do have to use this function. And my response is, if you use ROC AUC as your score to estimate the performance of your classifier, I would say you don't have to do that, because ROC AUC can deal with unequal numbers of samples, and in this case epochs in the input data. However, if you were to use, say, accuracy scoring, then it's very important that you equalize the event counts before you actually do that. before you actually run the classification, because otherwise the chance level will not be at 50%, and so it will be more difficult for you to interpret what is going on, and there might be also issues in other parts of the calculations you will be conducting. So I would say do equalize event counts just to be sure, unless, of course, there is a strong, imbalance, and you have good reason not to drop. Maybe your data is super valuable and you cannot afford to drop epochs. But if that is the case, please double, triple check that the scoring you're going to use will not be affected by this. I think there was one more question, but I cannot find it now. Maybe these were all. So what we did before the break was we learned how to classify epochs or data, experimental conditions, more generally speaking, based on entire epochs. And we first showed how we can do it using pure scikit-learn. So first extract the data from our MNE epochs and then bring the input vector x, or actually the input array x, into the correct shape, create the output array y. Someone scrolled over that. Where is it? There. The outcome array y, and eventually create and train the classifier. Then we showed how it can be easier using MNE's decoding module, because it's already aware of the data that will be stored within MNE, and we visualized the results. Okay, and now the question is, or actually the goal we have now is to actually perform decoding for not on an entire epochs level, but upon every single time point of your epochs. Okay, so what we want to do now is we want to train a classifier for every individual time point across your epoch. And this will help us see how classification performance changes over time, changes over the course of the epoch, which in turn can help you figure out whether and precisely at which time the spatial temporal activation patterns of your cortical responses started to differ between two conditions. In our case, this will be auditory left and auditory right. Okay, let's get to it. Again, we're going to use some features that are provided through scikit-learn and some that are provided using MNE. MNE comes with this sliding estimator thing. If you check out the documentation, import this, run the import up here, and then I'm gonna put a question mark there. It tells us it's going to fit, predict, and score a series of models to each subset of the data set along the last dimension. In our case, the last dimension is going to be time, but we don't have to worry about that now. It's just to say this is exactly what I said earlier. We are going to fit a classifier at each time point. Okay, so let's look at this code. So first, we're going to focus on gradiometers again, okay? I'm gonna pick the data from the gradiometers, gonna extract the data into our array or matrix X, and we're going to extract the corresponding events. Okay, and again, this is going to be, since we're looking at the subset of just auditory epochs, this is going to be events for auditory left or auditory right. There's nothing else in there. The classifier we're going to use is, again, a pipeline. Here we're going to use the standard scaler. That's the one we used previously in our first example. And as the actual classifier, we're going to use logistic regression. Oops, sorry, I was irritated because some Zoom info popped up. Okay, so that's the classifier where we want to use at every time point. Okay, at every time point. For scoring, we're going to use ROC AUC. And now, this is the interesting part here. We're making use of this sliding estimator thing that comes with MNE. And we hand it this classifier pipeline, which first applies the standard scaler and then conducts a logistic regression. And we tell it to apply the following scoring and some more parameters, for example, n jobs, which allows you to specify how many CPU cores to use, essentially. I have set this to 1 here to speed up things. And if you have more than 1 CPU core, you can set it to a higher number. I think if you set it to -1, it would try to use all of your cores. My computer is old. I don't want to do that to this old machine, so I'm going to leave it at 1. Here again, we have to specify the number of cross-validation splits. And now I remember this other question. It was regarding reproducing results and submitting a random seed to our classification. And I showed that you could actually, instead of passing a number, which is the number of splits, one could also pass a cross-validation scheme, a cross-validation object from scikit-learn. You could instantiate that manually and then pass it in here. And then I looked at the MNE documentation again, and to me, it's not so clear whether it even shuffles the data before running the classification. So that's something we could try out. You could try out, just run one of these classifications, these decoding code blocks multiple times and see if the results actually change. I couldn't make this out just by looking at the documentation. I would have to look at the code. By default, scikit-learn methods do not shuffle. At least these cross-validation methods do not shuffle the data before performing the cross-validation split. So it might well be that you don't even have to pass a random seed and would still have reproducible results, okay? Okay, yeah, and then we call this cross-val multiscore function, which is just gonna do, yeah, the cross-validation actually. This is going to return us, again, 5 scores because we have 5 splits. We're going to calculate the mean score. And yeah, and so just to be clear, this is going to be 5 scores per time point, okay? So since we have 600 something time points, it's going to be 5 times 600 something scores. And we're going to calculate the average at each time point. And let's just do that, and let's hope my computer won't die. So this is a bit ugly. You can see here it says hbox, whatever. Normally, and if you were to run this in the Jupyter Notebook and not in Jupyter Lab, you should see a pretty progress bar here. Obviously, that's not the case. I think it's a bug in Jupyter Lab, which has already been fixed in the latest version or so. But yeah, that's just to say, normally there should be a nice graphical representation of a progress bar. So this is going to repeat another 3 times because there's going to be 5 progress bars in total, 1 for each cross-validation split. For each split, it's going to fit a classifier at all the time points in the test data, and then going to try to match, to hopefully correctly tell apart auditory left and auditory right in the test data. Are your computers faster? Anyone already finished? And again, I would like to point out just how little code that is. Okay, if you remove the comments and this summarizing code down here, it's just 1, 2, 3, 4, 5, 6, 7, 8, and like around a dozen lines of code. That's quite neat. Okay, we should be almost there. Last cross-validation fold for me now. Okay, Pia is already done. Parfait. I think I always got a similar result. Pia got a mean cross-validation score of 0.567, which is a nice number. I'm getting the same. Yay. Okay, that doesn't sound too impressive, but this is now the mean across all time points, okay? Including baseline, including everything. So let's maybe visualize these results. There we go. So what we can see here during the baseline period, the decoding performance pretty much oscillates around 0.5, which is chance level because we had 2 conditions, right? 0.5 is chance. After time point 0, which is stimulus onset, suddenly we see a change. Decoding performance reaches, how much is that? Almost 95% or something at about 65 milliseconds after stimulus onset. And it remains, so here it goes down, and here it's up again at about 130 milliseconds, goes up to 84% again. And then it goes down again to around chance level, which is great, I suppose. This is just for one participant, obviously, right? There are currently no error bars whatsoever. So what one could do, for example, is add an area plot, which shows the range from the smallest cross-validation score to the largest cross-validation score, or the standard deviation of these scores, or something like that. So you get a better feel of how strongly the cross-validation scores actually varied for each time point. But I think this is a pretty good indication here already that there might be an actual effect at about 0.70 milliseconds and about 130 milliseconds. Okay, I'm gonna close that here. There is a question. The question is it possible to extract which channels actually drive the classification accuracy or the weights? Yeah, that must be possible. I don't know how. It must be possible. You would have to. I'm not sure. I'm not sure, but this data is certainly there somewhere. I just have never touched it, so I cannot answer that question. Sorry. Okay, great. Is there any other questions? One question is there something we need to add to change end jobs? It says it requires joblib. Oh yeah, you need to install the joblib package. You can do it using pip install joblib, which is into the chat. And then MNE will be able to spread your calculations across different CPU. Okay, welcome. Okay, so this is quite neat. Guess we can move on to the next notebook, unless there's any more questions. Oh yeah, what I wanted to point out is the epochs we used here were the ones before actual pre-processing, right? So we simply, they were based on the data that was just bandpass filtered, and that was it, right? So we didn't run ICA or SSP on these. We didn't, in my case, I didn't reject any epochs because of bad data quality or anything. So I essentially just fed them in as they were. And yet the classification performance is astonishingly high, at least at some time points, which is a good sign because it indicates that there might actually be an effect. And it also shows one of the strengths of certain machine learning approaches, which is that they are able to pick up patterns, even if there's lots of noise all around, they might still be able to extract relevant patterns in your data. So my suggestion, if you're planning to use machine learning or to get a first, impression of your data, don't do too much pre-processing and data cleaning. You can maybe immediately start with running a classification and see how that works to get a feel of whether there might be an effect or not. Not saying you should cherry pick, just saying that, as we mentioned earlier, too much pre-processing or being too strict or lenient about what is an artifact and what not can alter the outcome of your analysis. So, and the beauty of this machine learning stuff is that typically you don't need to do too much opinionated stuff to your data. That's all I'm saying. Okay, let's close that. Let's move on to notebook number 7. And I want to briefly show you that and how MNE can do time frequency analysis. Now, I personally, I have to admit, I'm going to be totally open with you, I have never really done time frequency analysis on my data. So I can just give you a brief overview, but I don't have in-depth background knowledge. So in case questions pop up that are too specific, I probably won't be able to answer them. You can ask anyway, and maybe there's someone else hanging out here who can jump in and save me. Okay, but that just said, we're going to start with our epoch data again. Of course, going to run our imports, and it's taking a while. Okay, now it's gone. And if we look at the info structure of our epochs, scroll down here, it says the projectors are on. Actually, I expected them to be off. I'm not sure why it says on here, because I just wanted to show you that I just wanted to suggest that we apply these projectors which are saved inside the data because they remove some of the artifacts that are already there. Okay, at any rate, so I'm gonna apply these SSP projectors, and then we're going to pick the auditory epochs. Okay, now let's have a look at the info structure of our epochs again. You can see the projectors are still on. Okay, I don't know what I did do that. Are they also on for you? Or did I do something odd when I saved these? Because I expected, instead of saying on here, I expected them to say off. Doesn't matter now. Just in case you want to do your time frequency analysis and you have added projectors to get rid of certain artifacts, be sure to apply them before actually conducting your analysis. That's all I wanted to say. Same with ICA, be sure to apply your ICA to the data to clean it before you start doing the time frequency analysis. We're going to work with our relatively unclean data. Okay, and we pick only the auditory. First thing we're going to do is we're just going to visualize the power spectral density. And we can tell MNE which frequencies or which frequency bands actually to use when creating that plot. And by default, it starts at 0 hertz and goes to, I think, infinity. But we're just going to limit ourselves now to 2 hertz to 40 hertz. And we ask MNE to calculate the average across all channels. You will see what that means in a second. Takes a bit, of course. And there we go. So for all our 3 channel types, we now see the PSD. So this black trace in the center of the gray is actually the mean across channels. I cannot tell you from the top of my head what the gray is, whether that's like 1 or 2 standard deviations, or whether it's the entire range or confidence interval. I would have to check the documentation. You can select. Oh no, you cannot select here. Okay. Well, okay, that's all we can do with that plot. You can see there seems to be some kind of peak at around, how many is that? 7, 8 hertz. Okay. And let's move on. So we passed here also a bandwidth parameter, and this bandwidth parameter essentially controls the resolution of the multi-taper that is used to generate PSD. We just picked the value of 2 here because it provides an okay-ish runtime. You can also play with this parameter and pass something else and see how it affects the runtime and the visualization that you eventually get. Okay, now let's have a look at what MNE calls a PSD topo map. Wow, that sounds fancy. Let's just try it out. Okay, so it's a bunch of topographic maps, and cool thing here is that it already has certain frequency bands that might be of interest predefined, and these color bars that are interactive. Okay, so if you click on them and drag your mouse, you can actually adjust the color scale. And yeah, it's quite neat. So we have delta, theta, alpha, beta, and gamma. That's for EEG now. We can, of course, plot the very same thing for the gradiometers and magnetometers. Let's do it for the gradiometers here. It's a bit small on my tiny screen, and also it doesn't scale with the size of the window. I think both these issues will be pretty much gone with the next MNE release. Okay, so that's quite neat because you see the distribution essentially of the power in certain frequency bands across the head. I'm just gonna skip the magnetometers now. Oh yeah, one thing I almost forgot, that's why we wrote it down here into the remark. So I just passed normalize equals false, which gives us the absolute values of the power, but we can also have it normalized, and that might be useful in some cases. Let's just change normalized to true here, and let's plot this thing again. And there we go. There is okay, 2 questions. Are these baseline corrected PSDs? Can I not interact here? And the honest answer is, I'm not sure, actually. I would have to check the documentation. There are ways to calculate the PSD with baseline correction. I'm not sure if that has been done when plotting these topo maps. I would think that not, but we will look into that in a second. Okay, we will extract the power into a separate object. So currently we are still using the epochs, and then using the epochs plot PSD topo map function. We will just in the next section create an object which is a power, and this has then the option to apply baseline correction in different ways. Okay, there was another question. can we change the range of the frequency bands again? For a plot PST topo map, I would have to check the documentation. And it seems that not. Oh, no, wait, there is a band parameter. There it is. List of tuples. The frequencies or frequency ranges to plot. So obviously, yes, you can do that. So, and here's the default. Okay, it's a list of tuples. So we can, let's just try something. We pass a list, okay? And the list contains of a tuple or multiple tuples, and the tuples 3 items. So let's define 2 custom frequency bands, okay? Let's start from 0, go to 10, and call this custom 1. And let's go from, I don't know, 7.5 to 15, and call this custom 2. And I've never done that before, so let's see if it crashes now or whether it produces something useful. But as you can tell, there is like the number of things you can do with MNE is so huge that even I use MNE daily. Even I don't know everything, especially in regards to time frequency, because I don't really touch that stuff. Okay, but now here we have our 2 frequency, 2 custom frequency bands, and the corresponding powers. Okay, happy? And yeah, I don't know why I get these warnings here. You can just ignore these. Okay, it seems the question was answered. Perfect. Let's move on. Power and inter-trial coherence. Now this is really stuff where I don't have much experience. Okay, so what we're going to do here is first, we're going to create an array of frequencies of interest. Maybe I should split up this cell into 2 to make it easier for us to figure out what's going on. I just copied these lines from another tutorial. I didn't write them. Full disclosure here. And let's just see what this does, okay? So obviously it creates log-spaced frequencies, and 20 of those, and they range from 2 to 30. And the step width is logarithmic, or it's a, how is that called? Geometric order or something. Okay, we want to use Morley. Do I pronounce this correctly? Is it called Morley? Time frequency decomposition. And this TFR Morley takes a number of parameters, and one of them being the frequencies for which we want to compute it. This is what we just created up here. The n cycles, I'm actually not really sure what that means. It's probably related to some specifics of this algorithm. I've never touched that before. We want to return the intertrial coherence as well, in addition to the power. That's why we said return ITC to true. We decimate the data to make the computation a bit faster, and we're going to use 1 job. So this here you can also change to 1. So we give cleaner results. However, it will take much longer. So I'm going to change it to 3 because my computer otherwise won't be able to handle it. What use FFT does, I'm not sure. So I'm going to check the documentation. Okay, it tells me that whether this controls whether it's going to use FFT-based convolution or not. I have no idea what that means. Okay. Okay, Olaf knows more. N cycles defines the length of individual wavelets and should be at least 2, and at higher frequencies, it should be frequency divided by 2. Perfect. Thank you so much, Olaf. Do you know, can you comment on the use FFT parameter? Do you know what?
I'm afraid no. I'm afraid not. But just for the number of cycles, it is actually an important parameter. It determines. I mean, a wavelet, as it's defined, is infinite. You know, it goes from minus infinity to plus infinity. So you have to make a choice there. And the number of cycles is the number of cycles that you basically chop off. So at what point, how many cycles of the wavelet do you want to include in your analysis?
Okay.
Obviously, if you only choose half a cycle, then you can't estimate the wavelet accurately. If you use too many, you don't have. I mean, it's overkill, or you have. Your wavelets might not fit into your epoch. So there are certain heuristic values, and those are the ones I mentioned. But better to look them up if you find examples elsewhere.
Okay, great. Thank you. Thanks for helping me out here. Okay, so let's run that. Let's calculate some wavelets. I hope this won't take too long. Dum, dum, dum. Are your computers still processing as well? Or is it just my old box again? I probably should have increased the decim value to 10 or so. Okay, it took about 1 minute for Olaf. Actually, I didn't time that when I ran it yesterday. I started preparing dinner and then returned. So I never realized how long it actually took. I just give it another minute here. Oh, it's done! Yay! Okay, and now that we're set, we're going to remove some. We're going to crop the results a little bit, okay, to get rid of potential edge artifacts, because there might be some at the very edges. So I'm gonna, we can maybe have a look at time points are currently covered. Does tmin exist? No, it's times, I suppose. The first time point is about minus 250 milliseconds, and the last time point is about 800 milliseconds. So we're just gonna crop it to minus 100 to plus 700. Okay, this cropping here works in place as we know from our previous work with epochs, etc. We get some info about the frequency range that was involved, number of channels, the number of epochs actually. Okay, and now it's time to actually have a look at the power we just calculated. And this is, so there was a question previously, can we apply different baseline? Yes, you can. So you can specify the baseline period. And here we follow a very similar approach, actually the exact same approach as with epochs. So we set a tuple, and a value of none is replaced with the smallest or the first time, and if it's the first element, and the last time if it's the last. So here we're going to use the beginning of our data until time point 0. And then there's different baseline modes. We can have a look at this in a second, but for now we're just going to leave that log ratio. Okay, specify that. And for starters, we're going to create a topo map, which looks like this. It's a bit sparse. It's because here, as you see, I only plotted the topo map for EEG data. We can also do it for MEG, but here I opted to do it for EEG only. So each of these small squares corresponds to an EEG channel. So this would be OZ, which is channel number 60, and here are the frontal channels. Here on the side, we can see a color bar which codes for the power. And if we click on one of these little, actually not squares, it's rectangles, right? If we click on one of these rectangles, we get the frequencies I'm sorry, we get the power across time for each frequency. Okay, we can actually select a range. Oh, my computer is so choppy. Okay, let me try it again. So I'm going to select frequency from about 5.4 hertz to 2 hertz down. I'm going to include a bit of the baseline period up to about 0.3 seconds. And this is what it's going to look like. Okay, this is then the average across this time and frequency. Okay, makes sense? So now let's switch back. Let's decide, well, assume we decided we want to look at a specific channel, and maybe we also want to apply a very specific baseline and baselining strategy. We can use power.plot function. We can add picks using the picks parameter. So this can be a string for an individual channel. It can also be a list of multiple channels. And we can pass a baseline and a mode, which is the baseline mode parameter. And there is actually a number of different modes available. Let's see if I can find it. There's mean, ratio, log ratio, percent, z-score, z-log ratio. And then it explains here what all of that actually means. We have opted to pick log ratio here, and the baseline period, like I said earlier, is from the beginning of our data until time point 0. So let's just run that and see what happens. Et voila. So here, again, on the y-axis, we have the frequency. On the x-axis, we have time, and this is power. And we can also, again, this is interactive here. Yeah, you can click on the color bar and actually drag it and can see how that changes the outcome. And I thought I could get a topo plot by drawing here, but it doesn't work. Okay. And look at all of these messages. Nice. So I think I was supposed to get a topo plot, but there seems to be a bug somewhere. Fortunately, it did not crash. I mean, that's already great. Okay. Let's assume we want to create a bunch of topo maps for different and custom frequency ranges. We can just use the plot topo map function. So here we use it 3 times, and let's just run that and see what it is. So we pass here the frequency ranges, right, and the name of the respect. Let's just see. Isn't that cool? So now we got custom labeled topographic maps that for now also look sane because we get the MEG helmet here, you know, this helmet shape. And again, the color bars make this plot interactive. Oh, there's another one. It seems to be stuck. What you can also create is joint plots. You remember these joint plots from our evoked responses, where we had topographic maps, and these topographic maps would be linked through a line to the traces that show the activation of all the channels. And by default, the maps would be plotted for the peaks in the data. We can do something similar here, and we can also specify at which frequencies and which time points to plot topographic maps. So let's just see what that does. Okay, nice. So we get this for all of our 3 channel types. It's difficult to arrange on this small screen, but you get the idea. So we get time, frequency, color codes, power, and we selected to to show the topo plot at 0.05 seconds for 2 Hertz, that's down here, and at 0.1 seconds for 11.1 Hertz. And a plot like this also allows you to easily compare activation patterns or frequency patterns across different recording modalities, okay? So this is what MEG picked up, and this is EEG, looking a bit different. Okay, so we've not only computed power, now we've worked with power, but we've also calculated ITC, intertrial coherence, and we can of course also visualize that by just running ITC.plot topo. Again, get a figure that looks pretty much like I don't know. It's very reddish, that's for sure. And again, we can click on one of these small rectangles and it pops the respective channel. Again, it should be interactive. And there we have for all of our sensor types. Neat. Okay, that's all I can show you so far for time frequency analysis. If you need to know more, there's lots of tutorials on the MNE website, and I bet there's one of them that covers time frequency analysis in depth. Otherwise, it seems that Olaf is an expert on this and might be willing to help. Okay. Oh, there is a question. Oh, it's a well. Okay, I take that back. Sorry, Olaf, I didn't mean to put you in this situation. Or maybe I did. Okay, so are there any questions regarding this? Because if that's not the case, we can then move on to a super fancy part, which is, oh, Olaf says you can always ask. Perfect. We can move on to the next part, which is a fancy part, and it's source estimation. And I just realized I haven't uploaded this file yet, and I will need to run a final test on my computer to see whether everything works correctly, because I did prepare this on a different machine, and I tried to run it on this computer, and it didn't work anymore. So I had to adjust a few things. Long story short, I will need about 5 minutes to run the final test and upload this thing. So I suggest we take a 5-minute break now, grab a coffee, go to the bathroom, and we reconvene at 2:15 London time. And we will then enjoy source estimation. Okay? Okay, I'm back. I'm now just it seems to work. I'm now just going to upload the last notebook we're going to need. 1 second. Okay, it has just been uploaded to the Google Drive, and I will also put it on GitHub just now. Okay, it's on GitHub as well. I'm going to share the Google Drive link again. So please, everybody, download this notebook, number 8, the last one, and open it in your Jupyter Lab. Okay, are you all ready? I'm just going to start, and if you're having difficulties, if you cannot find the notebook or if it won't open or whatever, just let me know. Okay, source estimation. It's tough, but I will show you how to do it. There is this, this schematic. I always only find it using Google. Not sure how to find it from the MNE website. If I go to overview, there's so much. Ah, here it is. Go to overview, and then the typical MEG workflow. Then there is this schematic. And it's essentially divided into two sections or two sides. This one is concerning the electrophysiological data, and this one is concerning structural information, meaning MRI scan. The processing flow on the left side, I think we've done almost all of that. So we started with raw data, pre-processed the data, we epoch the data, we average the data to create evoked objects. That's it. This one we haven't done yet, noise covariance. And actually everything that is in, is that red, is that orange? You get the idea. That is in this color, we haven't touched upon yet. But as you can see, to get a source estimate, we need all of that stuff. Structural information, we need a BEM, whatever that is. We need the source space thing, forward solution, inverse operator, a noise covariance matrix. Oh my goodness. But I'm going to show you how to do it. So go to your Jupyter notebook, Jupyter Lab for that matter, and let's get started. Because I was experiencing issues again with my plotting, I'm again running this line here, this percentage sign matplotlib qt. I hope this will fix the issues I had. We'll see. So what we're going to do now is, at least the last steps require that you have working 3D visualization. Actually, also some of the early steps require that you have working 3D visualization set up. If you have followed my installation instructions, the chances are that this is working. If you haven't quite followed the instructions, the chances are quite high that it's not working properly. So that's just to say that this is still kind of delicate. There's lots of dependencies involved, and it's tough to get it right and get it working on all supported platforms, Windows, Mac, and Linux. But we're trying to do our best, but we cannot guarantee that it will always just work out of the box. If you experience issues, do let me know. We may be able to come up with a quick workaround for at least, that will serve at least for the tutorial here. Okay, enough talk. Let's start running the first few cells. Again, it just switches desktops. I don't know why. Okay, let's run this. And just so that we start with some, let's say, fresh data, what I'm going to do here, what we're going to do here in the first, in this third, it's the third cell, is we're going to load the raw data again, and we're going to create some fresh epochs. Okay, this is really just to ensure that we don't carry with us any wild changes that we may have done by accident or entirely willingly during the previous parts of the. Okay, so we're just gonna create epochs from -200 to 500 milliseconds, and we're gonna save them at a spot where we can find them later. Processing for me done, and what is going on? Oh, I have run this previously, and the output file already exists, and I didn't pass override equals true here, so I'm just gonna remove these two lines, gonna add them to a new cell. I'm gonna run the saving, the save command here, and I'm going to pass override equals true. I forgot to add that, so it's gonna save that file, and we're going to have a look at the info structure. We already know that there's two bad channels. We know the sampling frequency. And one thing that's important now, these projectors here, they're off. And this is something that is important when you start doing source estimation. I think MNE might allow you to run source estimation on data where projectors have been applied and spit a warning, or it might not allow you to do it at all. I don't remember now, but it's just to say, if you are working with projectors and want to do source estimation, make sure they have not been applied to the data. First thing we're going to need is called the BEM, boundary element model. What is that? Well, let's just plot this thing here, because fortunately our sample data set comes with a BEM readily. And it's just, so what we see here is essentially the T1 scan, the structural scan, different slices. And you can see these, I'm not sure which colors these are. It's red, yellow, and I think orange, which essentially indicate what is the gray matter, what is the white matter, and where's the bone, essentially. So this is a parcellation that the free surfer software actually produces. Now, we're not going to create this parcellation ourselves now during this workshop. First, because it takes a while to run, and secondly, because it can be super painful to get FreeSurfer to run properly. There are no ways which make it easier, but it can still be a delicate thing. So we're going to use this BEM model that comes with the sample data set here. And yeah, I just got the message regarding the projectors that if you apply inverse modeling, the projectors will be automatically applied. This is entirely correct. But if they have been applied previously, MNE, I think, will throw an error message, or at least a warning. So you can have projectors in your data, they just shouldn't be applied. Where was I? The BEM. So we're going to use this pre-calculated BEM for this subject. But of course, you can also create your own, and you will have to create your own. No, I think I accidentally closed this tab. Oh, wait, where is it? Give me one. This happens when you have too many tabs open, and then just as your computer gets slower, you desperately try to regain some memory, closing some tabs, and end up closing the wrong one. I think I just found it. Yeah. Okay, let me turn on the screen share again. So there's two functions you can use to create this BEM. One is called make watershed BEM, and the other one is called make flash BEM. Make watershed BEM works with T1 weighted images, and make flash BEM works with flash images. I'm just mentioning this here because you should take note. I'm also going to add this to the notebook because this is definitely a step you will have to run if you have your own MRI scans. So I'm just gonna add this here. Normally need to create the BEM via create the BEM first, and the other one was make flash BEM. It's not mentioned here. Make flash BEM. It's really important. Otherwise, you will end up halfway through. Where's this now? I again closed. I think I again closed the tab that I wanted to keep open. Overview, a typical MEEG workflow. Otherwise, you don't have this thing here. And if you don't have this, you won't be able to calculate the forward solution. And without the forward solution, you don't get the inverse operator and no source estimate, okay? So you need this. Okay, so we created the BEM. We visualized the BEM. And like I said, this really just describes the different layers of your slices. Okay, what is white matter? What is gray matter? Where is the bone? Now, difficult thing, co-registration. MNE needs to know where the sensors were located on your participant's head. Positioning these sensors on the head, this process is called co-registration. And MNE has a nice graphical user interface that makes it easier, not super easy, but easier to get this done right and to visually inspect that everything went correctly. So let's just run that. So we're going to use, this is, by the way, the reason why I saved the epochs in the cell further up and didn't just keep it in memory. This MNE co-registration command, it requires that we pass the file name of an epoch or of I think a raw instance might also do it. But it needs to be a file name and not the data to work properly. Subjects here is also important to specify. That is the free surfer subject there. Not sure if you've ever worked with free surfer, but let me briefly show you. So here's the MNE sample data. And there is a subdirectory called subjects. And there is a few subdirectories. Again, one is called sample, which is going to be the subject we're using. It has the subject name. And then there is FS average and a few more. Actually, this is just one more. And you need to tell MNE where this free surfer subject directory, which contains the BEM and the MR data of your participants, where this directory is located. Otherwise, MNE doesn't really know what to do. It needs to have access to all that data when you want to perform source estimation. Okay, but now, finally, let's run this co-registration thing. Taking a while to start up on my computer. And it's also important, say you're working with EEG data, it's also very important to ensure that you have set the correct montage, which tell MNE where the sensors ought to be located on the scalp, that you've set the correct montage before running this co-registration step, because otherwise everything will either not work at all or will be weird. You will run into weird issues. I'm not sure it just has been done already. Okay, so here we have a head, a 3D representation of our sample subject. I think there is a beard down here. And these 3 points there, these are the fiducials. These are LPA, RPA, and the nasion. So we can ignore most of this stuff for now. We can click on lock up here. Once we click there, so we haven't entered proper locations of these fiducials, okay? So normally you might want to adjust these, but if you need to adjust these, you would typically know. Okay, so here we can just live with the defaults. We click on lock, and then what we get here is a representation of all the sensors that were involved during recording. And as you can see, they are not placed on the head properly, and we would like to change that. So what we can do is we can click on the right here, we can click on fit fiducials. That already did something. So now the fiducials of the MR seem to match the values from our sensors much better. And if I click now on fit ICP, this is going to run an iterative approach. As you can see, it tries to bring the sensors closer to the scalp. Okay, I think this worked actually quite nicely. It's not perfect. You can see these green, the green point here, the nasion. Normally these the same colors should match up, you know, for the Fibonacci. So RPA here, that doesn't really match. This doesn't really match, but it's good enough for our demonstration here, I suppose. What you can do is you can also change the shape of the head. For example, so I click on scaling mode 3-axis, and now you can move things. Whoa, my computer is really unresponsive right now. Whoops. So like this, let's turn it up to 100 again. So this compresses or expands the head in X dimension, and we can do that for all 3 dimensions. And we can also move the head relative to the sensors using these buttons down here where it says translation and rotation. Okay? And you would have to ensure that ideally ideally all the sensors are placed in this 3D representation exactly the way that they were placed when you recorded the data. This is oftentimes very difficult to achieve, but you should ensure that you don't end up with sensors that are sunken into the skull or are floating multiple centimeters. Okay. Oh, there's questions coming up. Okay, so one question is, Saskia, the error message you are seeing, did you change? Yeah, you changed something. Okay, yeah, it already looked like you were not running the original notebook I had provided. Okay, so for Saskia, it's working. Borja, there was an issue there. You get an error message. Runtime error, no PyFace toolkit plugin found. I'm not sure what that means. And it shouldn't happen with the MNE environment created according to my installation instructions. Are you sure you're running everything in the correct environment? Okay, I'm just gonna proceed here for now. Okay, so let's assume we are happy with this co-registration. Oh, by the way, there's lots of, this is one thing I love. You can have this tool make your participant grow hair. Isn't that cool? Oh, I love it. Okay, let's just return to 0. There's also some display options you can change down here. And for example, you can change the opacity of the head, which may or may not be helpful to find out whether electrodes might be, sunken into the skull. I think you can even color them. I cannot find this value right now, this setting right now. Yeah, but at any rate, this is how you can do the co-registration. So we can click on Save, because we're happy. So I haven't seen this before. I'm just going to click Yes. And we can I want to call the subject. We have to give it a new name, so I'm going to call it sample_new. And we're going to save. Now it offers us to save this trans file, and we're going to just save it in the out data folder. I guess that's the best place. Oh, it's still saving, taking a while. Now I'm not sure how to figure out when it's actually done. It says down here, scaling, computing. So unfortunately, this step also takes a while. And if we have a look at our MNE sample data directory, we can see that a new subdirectory has been created here. In this case, sample new. Is it still calculating for you too? Or is it just my computer again being slow as hell? Because if it's taking too long, we can also just skip this step and use some pre-calculated. Has it been finished for you? For Olaf, it's been done. Okay, so let's give my computer another minute maybe. I mean, I already have this trans file here. I guess is it done? It's done. Whew. Okay, it's done. Great. So, co-registration is done. We wanna view the results of the co-registration, and so what we did is at the end of the co-registration, when we saved the data, as I showed you, MNE created a new folder here inside the subjects directory inside the MNE sample data folder. Normally, the location of this subjects folder is someplace else. Okay, it's somewhere else on your hard drive, because this is normally where you put all the structural information about all of your participants. Since we don't have working free surfer installations here, and we don't have a, if you will, global subject directory, we just used the one that was located inside the MNE sample data. And therefore, so we essentially loaded this data here, performed a co-registration, and saved it, and MNE created this here. So essentially, these must be, I think these are all copies of what we had before. But actually, we don't need this. We need this because that contains, we only need this because that contains all the structural information we want. I just wanted to show you how to perform the co-registration, because the co-registration eventually helps you generate this trans file. In this case, it's called sample_new-trans, and we want to use that now. Okay, so I have to adjust here the file name. Obviously, I mistyped that. So the file name was sample_new-trans. Okay, and we are asking, so we also need an info structure. We're asking MNE to read the info structure from our epochs. That's the ones we created at the top of the notebook. And then we just ask it to please visualize the co-registration. For that, it needs that info. It needs a link or the name, the path of a trans file, a co-registration file, which, by the way, just contains a matrix, okay? This is just information on how to translate, rotate, expand, squash, whatever, the head, such that it eventually matches the sensors. We need to tell it which subject we used, so it will pick the correct MR information. And we need to tell it which subject steer to use. So subject is just to help you with your understanding. The subject name is always the name of a subdirectory inside the subject steer. Okay, subject steer, I repeat myself again, is currently under our MNE sample data subject. Okay? And here there's a bunch of subjects, and we want to pick the subject, not this one. We don't really need that. Is this working now? I have the impression my notebook is stalled. Yeah, it seems this step up here never properly finished for whatever reason. So I'm just gonna press the stop button and hope that it helps, but it doesn't. Okay, let's see, is there any Python window still open? It's not the case. Okay, so what I'm going to do now is I'm going to restart the kernel. I'm going to run the first few steps again. Is anyone else experiencing this issue? No, it's stuck entirely here. Same for Pia. Okay, let me try it again. Restart kernel. Maybe even shut down kernel. So now it has been shut down. So now I click in the top right here, where it says no kernel, and I select the first kernel that it suggests, Python 3 in my case. Clicking select, and let's see if we can run this stuff again. And this seems to be working now. Okay, if you follow the PI, if you follow this, it should work for you as well. Okay, perfect. So what I did again was I clicked on kernel, shut down kernel, and then in the top right here it says no kernel, and then you click there and select. Okay, so we have to run all that stuff again. Creating epochs. Be sure not to run the co-registration again. I have to say that the co-registration up to now still uses some old toolkit to produce this 3D interface, this 3D visualization. And Guillaume Favellier is currently planning and will soon start working on moving that to a more modern toolkit, which should make lots of these small issues, like the one we just experienced, simply go away. Okay, so I created the epochs. Okay, this line here this cell here I can skip. View BEM, I'm just going to skip that. Co-registration, the first cell I'm going to skip. The second cell I'm going to run. I dare. And why does it say, epochs fname. You see, we define the file name of the epochs here in this cell. Of course, if we don't run this cell, this variable will not be available. So I'm just going to copy this line into this cell down here. I'm going to run this cell. And I'm going to, and now I realize the subject there has also not been defined. Where did I leave that? It's been defined up here. Just going to copy that. Okay, does that make sense? You copy these two here. We need a definition of subjects here and the epochs fname. And if I run this now, oops, why does this not display anything? This used to work yesterday. Okay, let me quickly try something out, please. There is a message. It works for Pia. Why does it not work for me then? Oh my. Just need to try something. This is disappointing. Is it working for anyone? I wonder if I ran this line. Yeah, I did do that. I don't get it. Is that working for anyone? Does any of you see a figure coming up? Might just be my computer having an issue right now. Okay, it's my computer. I mean, that's great. That's great. So it's just me, so we can proceed and you will still have a good experience. All right. And what does it look like? Would any one of you care to share a screen? Maybe for those where it doesn't work, maybe for me included. Olaf, would you be so kind to briefly share your screen so we can see it? Great, exactly. That's exactly what we wanted to see. Thank you so much, Olaf. So that's the MEG helmet and the MR image superimposed. Thank you. Thank you. It's great. Thanks. Okay. How can I get back to sharing my screen again? Share screen. Okay. So the next thing we need to do. So we're done with the co-registration. Okay. This is this part here. Head MRI transformation, essentially from head to MRI. We have this BEM. We looked at it earlier, and I showed you which commands you can use to create it if you don't have it. Now, we need to look at these guys here, source space and forward solution. Okay, first on our plate is setting up a source space. There is a source space, essentially, it's just a, if you will, a mesh grid that represents potential origins of brain activity. Sorry, I just got a message there. That it's a mesh grid that represents potential origins of brain activity. Okay, so typically when we think of the magnetic or electric field that the brain actually emits while working, by processing data, just like our computer a little bit. It creates this magnetic field that we can measure with the MEG, with EEG. And we essentially just assume that we put a very, very fine grid onto the white matter surface, where we assume that there's the origin of this electric or magnetic signal. And of course, the finer the grid we put on there, the more the better, essentially, the later source estimate we can calculate will be, the higher the resolution, given that our measurement device provides data that actually benefits from that. However, this will also require more memory, will require more computational power, etc. This just said there are different grid spacings available that you can choose from in MNE. And one that I typically use during analysis is called oct6. Now, I have to admit, I again don't remember what oct stands for, something like octaedric or something. I don't know. But it contains, if I remember correctly, about 5,000 or was it 10,000 points for each hemisphere. You see, I will also have to check the documentation. The important thing here is that for this tutorial, we're only going to use a spacing which is called oct4, and it has a reduced number of grid points, if you will. This is just to speed up the computation time. But when you perform an actual analysis, you should not use this. And I've actually put a comment here, okay? You should use oct6 or something. We can also look this up in the MNE documentation. Maybe we can just do that, because there is a nice overview. Again, I don't remember where to find it. I'm just going to search for oct6 and see what comes up. It says here, head model. Let's just open these two. There was a nice table somewhere. It's not here. Somewhere buried in the documentation, there is a nice overview which compares how many grid points there are in these different models. Maybe if we search for setup source space, spacing can be ICO whatever, oct whatever for recursively. Oct is for a recursively subdivided octahedron. Of course, right? And maybe there is also a list. No, there's no list where there's not a table here. Okay, like I said, there's different grids you can choose from. Somewhere in the docs, it explains what they mean specifically. Cannot find it right now. Okay, let's create a source space. Also, we're going to set this one, add this to false. This is also something that you should not do when conducting an action analysis. Okay, this is also something that massively speeds up the computation for now. Otherwise, we would wait quite a long time on my computer to finish. So I'll create this source space. And as you can see here, it says surface, left hemisphere, 15,500 vertices, surface right hemisphere, and oh, it's actually not 15,500. It's I'm sorry, it's 155,000 vertices, and on the right hemisphere, it's pretty much the same. Okay. Also tells us which subject this is based on. So this is a good. It helps us to ensure that we're not using a source space that was made for one subject and try to apply it to another. Okay, so now we can run this plot alignment again, but this time we can pass the source space, which is calculated, and we can ask to plot different surfaces, white matter and the head dense. I think this was what we saw previously, and you would have all of them now in one view. Again, I don't dare to run that because my computer is going to. No, actually, it didn't crash, right? It just didn't show anything. So let me try to run it. Maybe it works this time. No, there's nothing. So hopefully it's working for you. Okay, so we've got this source space thing. This is super cool. So we've got this. This is all set now. Now we need the forward solution, the noise covariance, the inverse operator. Let's start with the forward solution. What is the forward solution? Well, the forward solution just means that based on the grid, essentially, based on that grid we just calculated, we're going to place an electric dipole there. And we're going to think, okay, if there is a dipole there, how would the electrical or magnetic activity from that dipole be projected to the outside of the brain? In EEG, that would of course be how would it be projected to the scalp, and for MEG it would be how would it be projected to the outside of the skull. And to be more specific, we will of course not calculate it for every possible point in the room, which is an infinitely large number, but we will only calculate it for each sensor. So that means given a known activation inside the brain, how would the sensor signal? Okay, that's why it's called the forward problem. We know which activation takes place, and then it's relatively easy to calculate how the sensor activation would look like in that specific. It's called the forward problem. Later, when we want to do the source estimation, we have the inverse problem, meaning we don't know what was going on inside the brain. We only have the sensor readings and want to go the opposite way. That's why it's then called inverse modeling. Okay, but for now, we want to calculate the forward solution. So, and for that, we need to create first the BEM conductivity model. And so the only thing you need to know here is that if you're working with MEG, you assume that you only have a single layer. And the conductivity of that layer by default in MNE is 0.3. Why only one layer? Well, because one assumes that the way that a magnetic field expands through the let me start that again. We know that magnetic signals, which we will measure with EEG, are being attenuated by different structures, like the bone in your skull, and maybe certain fluids and your skin. So there is attenuation taking place, and this attenuation is different for different layers while we are on the way from your brain to the outside of the skull, where we can actually measure the signal in EEG. Therefore, in EEG, it is recommended to specify conductivity for 3 of these layers. We assume there is 3 layers, and this works well, and conductivity is different in these different layers. For MEG, we just assume, oh, everything is the same, and there is a certain attenuation taking place, but we can work with assuming that there's only a single layer. So there is inside the brain and there's outside the brain. Therefore, when we ask MNE to calculate the conductor model, which is here just called the BEM model, and we're working with MEG data, we tell it, okay, conductivity is, there's only one value for one layer, which is 0.3. That's the default. I think there's lots of literature which can be used to verify that this actually makes sense and works. And if you were working with EEG data, you would use 3 values, 0.3, 0.006, and 0.3 again. Okay, this is just to say, if you're not working with MEG data, but with EEG data, remember this, pass in 3 values. Okay, I think we have to set the variable subject. Subject, in our case, is sample. And the other variables should have been set already. And this is what is being generated then. Not of that much use for us now. And now we're going to calculate the BEM solution. This probably also takes a bit on my computer. Ah, no, there it is. Okay, so the BEM solution here, the conductor model, 1 layer, okay? Because of this, we only specified conductivity for 1 layer. So this looks good. We also want to save that because if you have like a larger source space and maybe more layers, then calculation can take quite a while. That's my experience from working with EEG data there. Okay, so we have this BEM solution. Finally, we can create our forward model. So it needs as input the file name of a raw. Actually, it needs an info structure, so we can pass in a raw file name, but we can also pass in the info we defined earlier, if I remember correctly. Let's just see if this runs. And a file name to the path to our trans file. It needs the source model that we generated earlier. It needs the BEM solution. That's a mistake, so it should be BEM solution. We only want to analyze MEG data for now. We want to exclude the EEG data because it wouldn't make much sense, right? Our BEM solution was only 1 layer, and so we shouldn't really use that with EEG data. It will not work nicely. And we can pass this min-dist parameter, which means that, so in this case, it's set to 5. And this means if MNE finds a potential source that is super close to the inner skull, meaning less than 5 millimeters, or closer than 5 millimeters to the inner skull, it will just remove that. Okay, it will assume no, that's not very likely to be a proper source, and it will just drop it. Okay, let's see. Okay, so what I changed here again, this said raw f name at first. I just changed it to info because we read the info earlier, and that's all we need here. And here it said BEM equals BEM, and I changed it to BEM equals BEM sol. So sorry, I kind of, it seems I missed a few spots by copying and pasting. Okay, that's also done calculating. It tells us how many channels there are. And our source space is super tiny, okay? It's just 474 vertices. I mean, that's pretty much nothing. but it's okay for now, I suppose. And of course, we should save that to disk. We have the forward solution. Yay! Now we're getting super close, okay? We're almost there. Now we need the noise covariance and the inverse operator. Noise covariance, what is that? Well, it has something to do with noise and something to do with co-varying data and co-varying sensors. Essentially, we need to enable MNE to figure out how to properly regularize the data in order to perform a useful source estimation, if you will. And MNE figures out how to regularize the data properly by looking at the covariance of noise we provide to it. So now what is noise? And probably one could talk hours about what is noise and what is not noise, and whether noise is spurious or whether it actually might contain valuable information in certain situations. Let's talk about MEG data first. In MEG data and MEG setups, oftentimes you have empty room recordings. If you have empty room recordings, typically you would use these as your source of noise, because the empty room recording, for those who don't know, is you essentially start an MEG recording, but there's no participant in your experimental chamber in your lab. So you just record the noise that the MEG sensors themselves, or the building the MEG is housed in, produce. And you would then use this data and let MNE know, okay, this is my noise, please calculate the noise covariance of that. If you have an evoked design, and this also works in MEG and is the only thing you do in EEG, if you have an evoked design, you would typically use the pre-stimulus baseline period and would say, okay, this is noise. Use this and consider that as noise, and calculate the noise, the covariance of the pre-stimulus baseline period, and use that as my noise cover. Okay, and this is exactly what we're going to do here. So although it's an MEG recording, we're going to use the pre-stimulus period. I'm going to tell MNE, okay, please consider that noise, and please consider that as, please calculate the covariance of that. So we're going to use the function compute covariance. We're going to pass in our epochs. We're going to say, okay, please calculate only until time point 0, which is the stimulus onset. We can pass a number of different methods to calculate the covariance. And it also needs to know how to calculate the rank of the data. There's also all kinds of switches you can flip there, and I don't want to go into details. I just want to show you in a second how you can verify that actually the estimation of the noise covariance did make sense, or does make sense. Okay, so let's run that, and I hope it won't run for too long on my computer. Okay, it just complains that no average EEG reference has been set. I don't think that's an issue right now, because we are not going to look at EEG data for now. Okay, but this is also something important. If you analyze EEG data, you should use an average reference before running source estimation. Okay, so these figures, actually there should be two figures. Where's the, there's the first one. So these are the noise covariance matrices. On the X and on the Y axis, you have the number of sensors, so magnetometer, radiometers, and this is EEG, which looks a bit odd right now, right? And And what you want to see is that you want to see this this stripe along the diagonal, essentially. Okay, but yeah, I'm gonna spare the details now. This is really something we should put into the MNE documentation to make it easier to interpret. But this one here, these figures, they are really, really helpful. So what you can see is how much variance essentially is being explained by, start that again. So what MNE does here is it essentially performed a PCA, a principal component analysis, and decomposed the data into as many components as there are sensors. And for each of these components, it plots here the percentage or the amount of noise that it explains. And as you can see here, there is like a sharp drop. Okay, there's a sharp drop. This is because we have 3 SSP projectors in the data, which reduce the rank of the magnetometers from 102 to 99. And now the important thing here is to see that this rank estimation, here it says rank equals 99, and there's this, this dashed line. This dashed line should be precisely where this drop-off occurs. So should you ever see data, plots of data where the drop-off maybe occurs here, this can happen, okay? The black line would go like this, but this dashed line would not be placed at this drop-off, but would be placed, let's say, right of the drop-off or left, then something is off. Then the rank of the data hasn't been correctly identified by MNE, and you can stop right there. You don't need to do any more, you don't need to experiment with source estimation or whatever. You need to go back and figure out what was going wrong. Because if you continue working with data that produces a figure like that, you won't get any usable results, okay? So summary, if you see a drop-off like this, then this, this dashed line should be placed exactly at the drop, not right and not left of it. Okay, here there's not a drop-off like this, therefore the rank is placed, this dashed line, which signifies the rank is placed to the very right of the x-axis in both cases here, which I suppose is fine. I'm seeing time is running out, but we are also almost there. And hey, aren't these figures? Aren't these the figures that I couldn't produce earlier? Why are they here now? I mean, what's going on? It's magic. Okay, so we have the covariance now. Actually, this cell you can remove. This shouldn't be here. No, actually, we can use it. What we can do is we can do epochs.plot white, and we pass the noise covariance that we just calculated. And it will use the noise covariance to whiten the data. You will see in a second what that means. Okay, we're doing here epochs.plot white noise cof. Why is this not? Ah, no, it must be, I think it must be an evoked. Sorry, we do dot average. Oh, there we go. Hey, sorry about this confusion. This cell actually, it snuck in here accidentally, but since it's there already, we might as well use it. And now I'm getting inline plots and a pop-up. I mean, that's neat. Okay, so what do we want to see here? This is also a very important, so it's actually good we have this cell. It's a very important diagnostic plot here. What you want to see is that this, in the, oh, so far down. What you want to see is that during the baseline period, which here expands until time point 0, the signal, or the large majority of the signal of the traces you observe, should be between these two red dashed lines. Okay, So these red dashed lines are at +1 and -1. And you want the majority, I think actually 95% to be precise, of your pre-stimulus signal to be in here. If that's not the case, something is. And down here, we have the GFPs. And here, the red dashed line is at 1. And again, what we want here is that the GFPs in the pre-stimulus period are pretty much 1, okay? And this seems to be the case here as well. So with that info, we can be pretty sure that the noise covariance we estimated of our data is actually reliable, and we can proceed now with a good feeling to doing the source estimation. Okay, we're almost there. I just had a look at the clock. We're almost there. So we have the noise covariance. So now we need, the last things we need is the inverse operator, and then we can produce the source estimate. Okay, we need to import a bunch of stuff, a bunch of modules. Damn, I'm sorry, I accidentally screwed up this cell. So actually we don't need any of these here. You can remove all of these lines. The only one we need is make inverse operator. So delete all of these. There are certain parameters, again, you can set that specify how the sources should be oriented, and you know, should they be like perpendicular to the cortex, or do you allow more flexibility and the like. This can be controlled using the loose and depth arguments, but really we don't need to touch that now. The defaults are typically good. So let's run this. So now we have our inverse operator, which means we're just one step from getting our source estimate. Okay, we can pick from different methods to do the source estimation. There is DSPM, there is MNE, which gave MNE the name. There is S-Loretta and E-Loretta. I typically go with DSPM, it's relatively fast and gives good results. So what I'm going to, so this I don't know, something went wrong here while copying and pasting. I'm super sorry for that. So what we want to do is, you need to change here something, okay? What you want to do is you want to apply the inverse to our epochs, and you want to have the auditory left, for example, and you want to take the average of that, okay? So you want to apply it to the evoked. Pass the inverse operator, you pass this lambda 2 and signal to noise ratio, SNR signal to noise ratio. The default is 3. That's all parameters one can use to fine tune the analysis a little bit. In my experience, so with the data I've worked with in the past, this has never changed much of anything. So I don't know, I'm just sticking with the defaults. This is what the documentation recommends. So let's run that and have it visualized. Again, I cannot see anything. Oh, there it is. Why is it I cannot see anything here? Okay, what we do have is a brain. The surface is inflated, okay, which allows you to, there is a message. Oh, thank you so much. Yes, of course. Of course, I'm not seeing anything because I was looking at the wrong time point. Okay, so we have an inflated cortex. Actually, that's the white matter here. We have an inflated brain, which allows us to easily also look into the insula here. By clicking and moving the mouse, you can rotate this thing. On the Mac, I'm using my using 2 fingers for which emulate a scroll wheel. So I might also work with the scroll wheel to zoom in and out. And we can, if we press this play button up here, we actually see how the brain was activated, or which sources our algorithm calculated for us. And of course, we have these, it doesn't look very pretty. This is because our source space was so limited. If we had used a higher resolution, this would look much nicer. But yeah, so we have this. Let me zoom in a little bit. Down here, this time course, that's the peaks actually for the left and right hemisphere. Actually, I'm not sure why there is 3 now. Maybe I clicked somewhere. Ah, yeah, right. If you click somewhere, you add one of these spheres, and this adds a trace to the time viewer down here. And by default, it shows the peak activation in the left and right hemisphere. You can also play with these settings here, smoothing, for example, or you can change the color bar, the limits, There's lots to explore. There's also a help. So if you press I, this is super nice, by the way. If you press I, it toggles the interface. So now, theoretically, if I wanted to, I could take a screenshot of that, and I could publish it in my next Nature paper or something like this. And if I press space, the animation runs again. Isn't that nice? We can also use other surfaces. I think I'm going to use this one. Let's see if that works. So here, I now have a cortex. So the surface argument I passed here was pile. I hope I'm pronouncing that correctly. And see what happens. So now we have a colorful cortex. And there's many more options. You can also create multiple plots. You can link them. But this would lead a bit too far now, I suppose. But with that, I guess, we've managed to get through it, even though there were some rough edges. And I, again, have to apologize for these broken cells in this notebook. Like I said, I had some issues running the notebook I had prepared on this computer, which I have to use now. And I copied over from one notebook and one computer to the other. And in that course, it seems that I made some small mistakes. But I hope you eventually, all of you, got the thing to run and got some nice visualizations. There's a chat message. Okay, it's just some thank yous. Yeah. Did all of you manage to get the source estimate and the visualization of the source? I think white is also a valid name for a surface. Whoa. Yeah, so now I have white matter. That's really pretty, isn't it? Yeah. So one thing we didn't cover, it's group analysis. And I actually haven't checked the MNE documentation. I'm not sure how well it's explained there. But essentially what you do is, of course, you perform analysis for each participant individually first. And if you're on the sensor level, then there is a function which is called, oh my, I forgot the name. Is it called average? Average for the solutions, that's not what we want. Grand average? Grand average. Yes, so you process each participant until you have an evoked, and then you can use the grand average function to produce a grand average evoked. Okay, that is one thing you can do. If you want to conduct statistical analysis, there are examples, maybe even tutorials on the MNE website. If you want to do a group analysis on the source level and have actual MRIs for your participants, and individual MRIs for your participants, what you need to do is you need to morph all participants' brains essentially to the same brain, to the same reference brain, which is typically the FS average brain that comes with FreeSurfer, so that essentially you project the activity in each individual's brain to an average brain. So you have the same brain for each participant, if you will. And only then you can start doing some actual comparisons and running statistics. Did that make sense? So what FreeSurfer does is it will first take an individual participant's brain, it will inflate it to a sphere, and then it will map this sphere to a target brain, which will typically be the FS average brain. And you do the same thing for all of your participants. So say you have 20 participants, you start with 20 different brains. In the end, you have 20 brains that are the same. And then you can start to calculate an average or contrasts or whatever. Yeah, I guess that's it. Be sure to check out the MNE documentation. It is extensive, and sometimes the examples are not the most straightforward ones, but it's definitely a gem to have all of that. There's so many issues are being covered here. Oh, and maybe one final thing. We have been working on a processing pipeline called the MNE study template. Just gonna bring that up here. Oh no, that's just the GitHub repository on our website. mne.tools/mne-study-template. Why is this not? Did I just mistype that? There we go. I'm currently working on improving the documentation, but essentially what we have there is a collection of scripts that start with bits formatted raw data, filter the data, epoch the data, run ICA and or SSP, calculate contrasts, perform machine learning, do time frequency analysis, do group statistics, source estimation, and in the end create a quite a large report actually with all processing and analysis results. And this is still a work in progress, but it may be worthwhile to check it out. Also because you can just have a look at the script and can see how we're doing things. For example, so I'm just following this link to GitHub here. And if you go to scripts, then you have a collection of scripts that do different things. Importing max filter. Let's maybe have a look at, I don't know, sliding estimator. That's the part that does the machine learning. You see we have quite a number of imports, so it's not always super straightforward to understand how things work, but I think you will still get an idea, because here you see, remember that? We create an X, we create a Y, and then this pipeline, then we use a sliding estimator and do stuff with that. So this can also be a source of inspiration, because we're, of course, trying to follow best practices in the study template. For example, when we perform ICA, we first high-pass filter the data. Things like that, tiny bits like that. So yeah, do check it out. There's some more questions. Okay, the question was, if we want to work with MNE on our data, would you suggest we look into the MNE study template or try to work on our own scripts. So I would say, as long as you're a beginner, you should write some of your own scripts, definitely. And you should explore what MNE has to offer for you. Once you have created a script that works, then maybe it would make sense to have a look at the respective script or respective processing step in the study template and see how the problem you were trying to solve was solved there. Maybe you will discover a certain parameter that is used in the study template and might turn out to be important or of advantage that you've never heard of before. So that could always make sense. If your question is whether it can make sense to entirely skip your learning phase, essentially, and immediately start using the study template, I would not recommend that, because if you don't have any experience with MNE, you won't understand what the study template does in detail. And I think you should understand, or should be able to understand what's going on under the hood. It shouldn't be a black box. That's my personal view. So yeah, I would say first, do some hands-on practice yourself. See if you get it to work. See if it seems to do what it ought to. And then, only then, would I have a look at the study template for this respective part of your analysis. And that's my opinionated personal response right now. If you ask me again next week or in 2 months, I might say something different. Yes, but if anyone is willing and wanting to try the study template and running into issues, or like I said, I'm reworking the documentation right now, so there's probably also certain things that are still unclear. If you check out the current documentation when you get started, you might get stuck somewhere. But if you experience any of that, you can also just drop me a line or open an issue on the GitHub issue tracker. Yeah, I guess that's it. Oh, and maybe one last closing remark. I mentioned it, I think, at the beginning of yesterday's session, getting help with MNE. So there is currently a mailing list and there is the GitHub issue tracker. So now the GitHub issue tracker is actually meant to report bugs or things of which you think there might be a bug. It's not so much made for user questions. Then again, the mailing list, which is intended for just that, it's not very appealing, right? I mean, who wants to use a mailing list? And for that reason, we've been thinking about and discussing ways to make it easier for users to get help, and also for users to engage in helping other users more easily. And we've been in touch with Discourse. So this is it's actually a company that produces an open source forum or bulletin board software, which is super popular and super nice to use. And it has gamification. For example, if you help someone, you get an award. And if you have your 10th posting or something, you get another award. And it's super neat. And we will most likely switch to that very soon. There are still some details to discuss, but it looks as if within the next few weeks we will do the switch. And if for whatever reason we decide not to switch to this specific software, we're going to use something else. But we will offer a user-friendly way to, and a modern, not only user-friendly, but also modern and accessible way to get help and potentially get in touch with other users and exchange thoughts, tips, ideas, and once in a while, probably also a bit of frustration, but hopefully not too often. Okay, oh yeah, and if you don't do that already, maybe you should consider following the MNE News Twitter account. So it's just, let me see, I think it's just MNE_news, MNE_news. Yes, there you go. And anytime something notable happens in the MNE universe, it will be announced here. So yeah, I guess that's it from my side. Thank you for taking the time.